Shweta, Ed and Assistant Professor, Department of Biotechnology, MMT and SDM MMT. We welcome you, ma'am. I request Srimati Meghana M. Head and Assistant Professor, Department of Biochemistry, MMT and SDM MMT, to welcome the co-chairperson, Dr. Shweta S. Lodin. Thank you, ma'am. Now I call upon Nidhi from 2nd BMDB to formally introduce our esteemed chairperson, Dr. Anil Kumar KM.
Good morning, everyone. It is an honor to introduce our esteemed guest, Dr. Anil Kumar Kheran. Dr. Anil Kumar Kheran is an associate professor at the Department of Higher Education and Research. He has extensive experience in teaching and research with a strong focus on water and health, water management, biodiversity conservation, and sustainability. He holds a PhD in environmental science from the Department of Higher Education and Research. His research interests include environmental technology, integrated water source management, biodiversity and conservation, and water and wastewater management. Dr. Anil Kumar has published over 60 research papers in international journals, including significant contributions in photoclimate degradation of Antarctica and sustainable water management. He has received several prestigious awards, including Insights Award from the Global Research Foundation and the University of Toronto University, as well as the NCMR Senior Research Fellows Award. He has also established international research collaborations with universities in the UK, Thailand, and Iran. His research expertise covers wastewater treatment, water reclamation, sustainable water resource management, biodiversity conservation, environmental data analysis, sustainable development, and the synthesis and application of nanomaterials for environmental remediation. Through his work in teaching, research, and community service, Dr. Anil Kumar has made significant contributions to environmental science and sustainability. We're glad to have you here, sir. We welcome you. Thank you.
Thank you, Nidhi, for your warm introduction. I request Dr. Anil Kumar KM to introduce our esteemed resource person, Mr. Vinay Aradhya.
Good morning, everyone. These are the things that Sri Government of Krishna. Thank you for giving me the opportunity to share the scientific session. Good morning, everyone. Now, the scientific session, we have a session in the product management, and technology council, Toronto, Canada. So he is the director of AI project and CM to setting AI project development, and he is associated with Government and Quantity School of Business and Technology. He is a member of the technology, and he is board on strategy in accelerating AI strategy, functioning in laws on water development and workflow. Platform. So here's the AI, that is how training AI, artificial intelligence, drawing, leadership, and he has many awards and recognition. He has recognized by Government Council, and City Award for Government Excellence, and Community Award Business, and the in product management for the AI. He is going to talk about AI as a co-founder from capabilities to product and commercialization. He is well experienced in AI
Thank you, Rest. Can you hear me okay?
Building on what has been shared, I now invite Mr. Vinay Adada to take over the session on AI as a co-founder from hypothesis to product and commercialization.
Thank you so much. Can you guys hear me okay?
So you are audible.
Okay, thank you. Thank you, respected Chair Dr. Anil, Dr. Shweta, Professor and Head of the Department. Thank you so much for inviting me to speak to all the audience who are so much participating at 8 o'clock in the morning from past couple of days. A big round of applause to all of you for participating at this event, an international or national conference. Today I'm going to speak about what today I'm going to speak about what is possible from a life science perspective to bring a product from zero to one, meaning zero customer to one customer, and to scale to an enterprise. Meaning, how can you be a founder of your own product and build your own company? Eh, We've been hearing from our uh, Prime Minister of India, by 2047, India will be a developed country. And there is a lot of rush in terms of supporting entrepreneurship, startups, building in India or make in India. What is possible today? Why this talk? What is the reason of this talk? I'm going to start off by explaining a little bit, just giving you a little bit of background about myself, and then I'll give you what's possible today in the current AI era. I see that you've already seen one of the talk yesterday on generative AI. Okay, let me give you a little bit of glimpse of what I'm going to speak about. What's possible with the AI? The talk is more on AI as a co-founder. Let me screen share and then let me know if you can see my screen. Is it visual? Is it visible?
So it is visible.
Okay, perfect. Thanks so much. The topic is AI as a co-founder. We know that to start a company, it's very difficult to begin understanding the market, understanding the customer, understanding the competition. More importantly, building something from zero to one ground up, whether if it is an AI, it's a software product or it's a hardware product. But let's think about focusing on life sciences. I'm going to give you a little bit of background about what I've done. I finished my master's in University of Mysore in biotechnology, like most of you are studying biotechnology and life sciences here. I moved to Canada for business side. I wanted to explore the, hey, what's on the business side of things? How can I commercialize what my knowledge I've learned into building products? So that's the beginning of my journey. Luckily, I founded a company on cloud computing and technology space and moved into multiple enterprises, whether it's telecommunication, fintech, startups, e-commerce, gaming, currently SaaS products, business to business product. So that's the journey, and which the reason I'm giving you the journey is anyone can do with the life sciences in different industries, different domain, but you need to tap into your own skill set. What is possible with your skill set? What is your passion? What is the industry looking for? What is the market looking for? What is your customers or target users looking for? And where you can make money. That's the commercialization part of it. So the topic is from classroom to co-founder. How can you do that? Is it possible? When we start any education, as kids, when we go to school, all we thought about was, can I get a job after I complete my school, university, graduation, what have you? And many parents and your relatives ask, hey, what kind of job do you get? But now you can confidently say, I can create jobs as opposed to getting jobs. That's the premise here. The power of AI as your co-founder, it will handhold you to get you that co-founding experience and also starting your product or company from zero to one, and then you can scale to enterprises. And there are multiple ways of doing it. Whether you focus on business to consumer, directly going to the customer. I'm going to give you some examples, specifically AI products in healthcare, built in India, and I'm using some of the products. It's amazing. And there are other side of product which is you're building for the company. Let's say you have some life sciences experience, you build a product and you sell that services or product to a company. So that's a business to business model and business to consumer if you go directly to consumer. Let's more on that in the upcoming slides. It's easier said than done. You can say, hey, how is it possible? I'm in life sciences and I'm in school, and you're saying that I can be an entrepreneur. Many of them have done that. And if you think about Facebook and what have you, all the founders, they've started a company when they're in school. They're bright, they're ambitious, they find co-founders in the school and then build their product. What are the commonalities here? You guys do research, you find, you work on projects. Entrepreneurs do validation. They are pretty much coming with a hypothesis whether my target customer, my user base like it or not. That's a hypothesis. Hey, you know what, my uh, problem statement or hypothesis is, hey, so many of those people have issues finding knowledge about naturopathy, for example, or he getting kind of healing from ayurveda or herbal medications and what have you. It's hard to find because a lot of information out there, but how can you validate that this is actually a problem? That's the hypothesis. You guys present in your projects, in your school. Entrepreneurs sell that. They'll pitch their idea to raise capital, they'll pitch their ideas to get customers. So they're always selling. That's pretty much you do that in school in presenting. You work on projects, entrepreneurs work on shipping those products, whether it's physical product or it's a digital product. There are like parallels you can think about. And how do I start now? Yeah, it's cool to know you've been using, some of you or many of you are using ChatGPT or similar, or many other LLMs to get your job done on a daily basis. How do we start the product discovery? When I say product discovery, anything is a product. And I'll give an example. What is a product? Product is pretty much like a solution to a problem. If people have a problem, you work on that, solving that problem, which is viable. When I say viable, it has to make sense to the company. When you start a company, it has to make sense in terms of financially viable, legally viable, ethically viable, many parts of the viability part of it. And usable, can it be used by my customer segment? Is it usable? For example, we've all, I don't know how many of you guys know Nokia as a phone. It was a really wonderful phone in my university's time. It was really hard to break. Even if you drop it, it's almost like a brick. It was wonderful at the time, but it is non-existent right now because of iPhone's innovative, 10 times better than a Nokia or other competitors. What happened? It's revolutionary design, user experience. Is it usable? iPhones or many other smartphones are much more user-friendly. So you need to think about viability, meaning can I make money? It's not just like solving problem. You need to make money after solving problem. Is it usable? That's the user experience part of it. It should not be very clunky. It's hard to use, cannot be that. It's very easy to use. And most importantly, can I build it feasible? Meaning, is it possible to actually build it? I can't think about some time machine right now. It is a cool idea, but is it possible to build? Is it feasible? I don't think so right now. But in the future, could be. You can build a time machine. Similarly, there are the three-legged stool here: viability, feasibility, and usability. These are the things that I need to think through. So what do you mean by product discovery? I just want to check time. Product discovery is just understanding the problem statement. You need to understand, like you might have heard about Einstein's quote, if you want to solve a problem, you have to spend majority of your time on with the problem, not finding solution. Let's say out of 100%, I'll spend 95% with the problem. I deeply understand the problem and then I can think about solution, because solutions pops out like that after knowing the problem. So product discovery is like that. When I know that, let's say, take an example of naturopathy, for example, I want to like heal my body using herbal medications than English medications. If that is the problem statement, how many people have that problem? How many are willing to even pay for it and get that use out of it? So I need to talk to minimum of 30 users or my target users, and I need to understand their pain point. Is it actually a problem? If I get signals, excuse me, if I get signals that there is a problem, That's a qualitative study. And then you need to do a quantitative study. You may have seen these surveys, your MBA students or commerce student might have sent you surveys. Hey, can you take a survey? I'm doing some research. Or you might have done some surveys too, where, hey, do you like this? Do you like that? What is your pain point? Like few questions to understand user sentiments, user problems, excuse me. And finally, understanding from the qualitative to quantitative to really find statistically significant number, meaning I need to be really sure that this is actually a problem so that I can go and solve the problem. Meaning, I don't want to spend so much time and money to a problem that is not a problem. You go and that's what majority of the companies fail. Excuse me. Startups fail because they find wrong problems because they jump into solutions too quickly, because they get excited. Human brains are wired that way to solve problems. Once they get into it, you get emotionally attached to the solution. Oh, this is my product, it's my baby, I'm going to bring this out to the customer. Eventually 2 3 years after, nobody buys your product, and then product doesn't exist anymore. So what I'm trying to say is product discovery is very essential. Talk to customers or users of your target segment. 10 to 30 is the ideal number. Understand their pain points. What is their problem? What is their frustration? Why is that a frustration? So on and so forth. And then you go and send survey to at least 300 to 400 people to get like a really good number, that it is not like 10 15 year, it should not be biased survey. You cannot just go to your friends and family and then they just say yes to everything you say. You need to get random sampling. It's, you can read more about statistics on that, or you can reach out to me. I can, go in depth on that. So that's product discovery. Now, how do I use AI here? So I've spoken a lot about product discovery. Where is AI coming in? That's classical way of doing things. How do I amplify using AI? This is a prompt I've been using for myself. You give a persona in the prompt engineering and you give a task, you need a context, and you need a format that the AI should reply back to you. Perplexity is an amazing research tool on AI, built in the US by an actually Indian CEO in the US. The problem here is the persona here is you need to tell the AI what persona it is actually embodying. Like in this example, hey, you're a startup founder evaluating 0 to 1, meaning zero customer to one customer, telehealth opportunity, like a doctor on demand, for example, on the phone. And what is the task? Conduct a structured discovery research following an idea. Just go and search on the internet, Reddit, and what have you. Come back with your own understanding. Come back with your own research. And here is the context, and here is the format. Format could be come back to me with a tabular format or an executive summary. However you want to read it, make it make sense out of it. You can do that. PTCF is the framework which is widely used, and it's very beneficial for me. When you do that in chat GPT or Perplexity is pretty good for research. It comes back with a phenomenal results for you. It can do a computation analysis. It can give you a target market. Who is that? What is the total addressable market? Can you actually solve it? All that stuff in matter of seconds. It would have taken weeks in traditional way of doing things. You need to go to an agency, spend thousands of rupees or hundreds of thousands of rupees, and get that research done by an agency. Now with AI, you can just do that in 2 3 minutes, or maybe let's say 15 minutes. Once you get that, now you can also do a trick here. I don't want to go like really take a lot of time. The trick is you can ask the AI to behave like a customer. Hey, can you go and behave like the customer of this persona so that I can interview you like a human being? So you can just go have a conversation with AI. How does AI know the target user's persona? It can go and research about it. For example, I want to know about what does SDM university students like. And you may have done some Facebook, chat or Instagram feed or Reddit or what have you on some social content. So now the AI will go and discover what you have mentioned or collectively as students, then it'll bring back that persona to itself and behave like all of you. And I can chat with, hey, what does SDM university students like or dislike? What kind of content they like for me to go and talk to them. So it will just go back and forth with me as a students of the university. And that's how you can do interviews with AI. Perplexity is amazing with that. So discovery is done. Now we know exactly who is our customer, what are the pain points, is it actually valid to spend time and money to invest building product. Again, this is the user interviews I was talking about. Do a lot of user interviews. So what happens after the user interview? Like, let's say you figured out a problem. And how do you go and validate that? Like, how do you know that users actually like it? You cannot visualize it. You need to build a prototype. You need to something similar to a closer product. And going and getting that done with the technology team, it's going to cost you a lot of money. It was costing a lot of money before. Now, with the help of Lovable, that's a prototyping tool, Claude Code, Replit, and so on and so forth, it is in a matter of minutes you can build products. That's the prototyping with AI tools. I'm going to just give you, and again, I'm not affiliated to any of these products, I'm just giving an example of what's available out there. When you go to Lovable, you may get free credits when you log in. You have to just chat like a chat GPT. Hey, here is my problem statement. Here is what I'm trying to solve for. And here is the solution I'm thinking. Let's say naturopathy here as an example for this discussion. Many of Indian people are not thinking about going with English medications. They want to just use food as medication, and we want to tap into our ancestral knowledge, Ayurveda. How can we get that concise knowledge into one place as an app, and an everyday user I can just go into app and then talk to that AI, so the app can come back and give me prescriptive ways of personalizing that medication for me using the kitchen items as medication. That's the problem statement here. So I'm going to just write that here in this context, in this text box. It's going to think about the UI. It's better for the UI. It does all the magic for you. Come back and gives you really almost closer to a product like prototype. What are you going to do with that? You can just take the prototype, go to your users, and then show it to your user. Hey, you know what? User number one, whatever the name is, say, here is the product, here is the prototype, and here is the problem you mentioned. I can't find all the details of Ayurveda. I can't make sense of everything is in Sanskrit. I don't know Sanskrit so much to understand that. That's the problem statement. Now, if you can give that prototype or product to a user, they can just click through. Hey, I have this problem. Can you give me a remedy for that or a medication? It will come back and say, hey, here are the 5 steps you need to do at home, what have you. So that's like a really cool thing. And then you can see that how the user is interacting with the product, and are they excited about it? What is the wow factor? Can you see their eyes? Can you see their emotions? You can get all of that when the user is interacting with your product. And the more they like it, the more it is better. And then you can interview iteratively, meaning go back and forth with the user to improve the product based on their feedback. That's an iterative improvement. Similarly, there are tools out there like Replit, which does the whole software for you. This is only just a prototype, Lovable. It just gives you, when I say a prototype, it look and feel like that, but it has nothing in it, which means it doesn't work. It just shows you a pretty picture outside, but it's nothing inside, meaning it doesn't actually have brains to work. But in this tool, Replit, it does the whole end to end, meaning it shows you look and feel and the brain of the product, meaning the thinking and all the logic and the data, everything. So it gives you a whole end-to-end product. You can actually deploy that and solve for it. You may need like one engineer with you as opposed to 15 engineers or like thousands of lakhs of rupees spent on developing the product. You can just do it in like thousands of rupees here, giving you a little bit of context of how AI can help you become a founder, an entrepreneur, CEO of your own company. So hey, I'm going to give you some of the examples which I like. I've discovered one of the cool product AI products in healthcare in India is called August AI. I don't know if you have looked into it, heard about it. You can go chat with it via WhatsApp messaging. I can chat with him. Hey, and I have this personalized questions as well. Like, hey, I have this problem. What do you think? Talk to AI doctor. It will come back to you with assessment of based on what the content it has been trained on LLM, comes back and say, hey, for this problem, here is the solution, here is what you need to do. It is just like easy for you to discuss with AI doctor, not replacing the actual doctor, but it is really good to know than asking a friend or Google research yourself. It gives you a more content curated by doctors. This tool was built by a doctor in India called August.ai. You can There is a number, you just have to text it via WhatsApp. It will reply back and say, hey, what's your request, and it'll go back and forth with that. It's amazing product. And many products on oncology, AI powered tool, and we worked on some of the cancer research as well. Like it identifies what is a cancer, a skin cancer cells much better than a doctor's eye, because it goes with pixel, very accurate, precision is really higher compared to doctors. That's the power of AI in India. That's how doctors have built. And the reason I'm saying this, you are in the right space at the right time. AI being helping you to build products with the knowledge, with the professors around you, with the support in India. Everyone in who's listening here can be an entrepreneur. That's the future of India, and that's the future for the new students and upcoming generation. I hope you can take this. How do I launch 0 to 1 product? Again, I'm going to go back here, just going to high level, very high level. Find what is the problem in the customer space. It has to be big enough. It should not be like 5 people have this problem. It needs to be millions of people have that problem. You're going to go with 5 to 10 customers or 30 customers. You can interview them, and statistically significant, you need to send that to 400, 500 users. To reject the null hypothesis, you need to have the confidence of 95% that that is the problem. And you need to think through, brainstorm solutions, prototypes like the Lovable and what have you, and quickly build the product using Replit, tools like that, and have one engineer if you really need one. And then that's why you need a co-founder after the AI's initial heavy lifting is done. Once the MVP, when I say MVP here, minimal viable product. I've mentioned what is viable. Viable has to be, it has to be usable. It has to be valuable to the customer. If it's no value, nobody wants to use it. You can take about any example. If the marketing is fantastic, let's say take a simple example, shampoo or whatever you buy. If the marketing is fantastic, you're like, oh wow, this is amazing, I'm going to bring it. Like it doesn't, when you buy that, you use it once, it doesn't smell, it doesn't give you any of those things. It actually makes it worse for you. Then you're not going to buy that again. You'll say, hey, this is really not a really good product. So that value should be really, really high. You need to have significant value to the customer. Customer should be in the center. You need to solve the problem because you're passionate about this problem to be solved. You know the industry well, you know the knowledge, you know the domain knowledge. And the byproduct of making money and all that, it's just for the byproduct. You should not make or be an entrepreneur just to make money or fame or rich. You really care about the problem space. You really care about the society. You really care about having a positive impact there. That's the reason to be an entrepreneur. And you also have the passion and knowledge to have an impact. So that's the MVP. Then once you have that, you will learn. By that time, you will have a lot of knowledge of how do I iterate, improve the product over time. And then you can hire first few founding team, and then you can go from there. I've looked into some of the support in government of India. It's amazing out there. Like I wish I was born now. It's so much in India happening on startups. I was blown away by learning a lot on biotech, ignition grant, and thousands of grants available, specifically like women entrepreneurs. Government of India is actually pushing for women entrepreneurs. This is a right time for everyone to think about what problems I need to discover, talking to the customer to see that is actually a problem, and then solving that problem because those customers don't know how to solve that problems for themselves, and then making money out of it. Let's say I'll give an example of creation of value to the customer and capturing of the value. That's the commercialization part of it. That's the end talk or end part of the talk. So now you build the product. Like, how do you commercialize? You have to keep that commercialization in the mind throughout the journey. It's not like at the end, it's just in the center of it. So when you create value, for example, I'm going to just give you an example. Let's say if I uh, charge you 1,000 rupees for consulting or coaching you guys on something to start a company, and it will generate you, let's say, 10 lakh rupees. You should be able to give me 1,000 rupees, because that's what, hey, you know what, he's generating me 10 lakh rupees. I don't mind giving him 1,000 rupees. So that's the value I've created to you. You don't mind giving me 1,000 rupees. That's the capturing the value. So I've created the value. Now I'm capturing the value, what I've created for you. So any customer who are in that pain, any user who are having that unmet needs or pain points, when you solve for that, they are willing to pay you some for that solving the pain point or solving those unmet needs. That's the creation of value, and capturing the value is the commercialization. There are multiple ways of monetization. I can't get to it all of it today, but when you get to it, it's amazing, mind blowing. You may have seen subscription basis. You know, you pay monthly this much, or you one time cost. There are a lot of ways of SaaS products out there. You can explore more on that pricing. There is a book called Pricing Monetization. It's an amazing book. You can give it a read on commercialization. To wrap it up, what is needed? Find the problem, talk to the users. That's your hypothesis when I say find the problem. Talk to users, validate the problem actually is a problem. Go back and prototype with lovable replets and the tool AI tools. Then talk to users and show it to them. Hey, here is the solution. I've built it for you. Is it solving your problem? If they say yes, amazing. And then build that, deploy, go iterate. Then you'll be either B2C, B2B, depending on the model you want to choose. That's how you'll be an entrepreneur. I feel India's future belongs to those who build. It's not make in India, that's the reason. It's just the future will be more entrepreneurs, because the conventional way of getting a job is getting slimmer and slimmer over time. You need to think through broader, how can I be an entrepreneur? How can I use AI to the best advantage I have? How can I be a founder? How can I start something in my school days as a side hobby and become that entrepreneur? With AI as your co-founder, you have everything you need to start. Money is not a challenge at all. You need to find the right problem to solve. You need to have the perseverance, you need to have the passion. You cannot have something easy. You need to persevere really hard until you actually accomplish that. I think that's the future for those in India as a builder. With that said, thank you so much for your time, and a great round of applause for all of you to be here at this time in the morning at 8 o'clock in Indian time. I thank you so much for inviting me for this conference. Thank you, Dr. Shweta. Thank you, Dr. Everyone. Thanks so much, STM. Over to you. I look forward to hearing for anything. Please connect with me on LinkedIn if you have any questions.
Thank you for spending your valuable time to share your knowledge with us. We are deeply honored by your presence. I humbly request Chairperson Dr. Anil Kumar K to share their valuable thoughts.
Thank you, sir. So it's a wonderful presentation. These students to craft the resources, like Mr. Person and your wonderful presentation about the AI. Actually, sir has mentioned about the hypothesis HPC, that is the name of concept, hypothesis and modeling and then commercialization. So now students are like in the AI, now it is a training. So before we have to write the legal documents, we have to take to help of the AI. Now within 2 seconds you will get 100 varieties of letters writing. Apart from letter writing, we can use for the research purpose also. If you are like a review article or research article, so how we can get the acceptance, we can participate in any of this. So we can use that and we can get any information about the review article. So when I was studying PhD, I used to read 100 to 200 research articles in the library. So now we can worry about this. So we can just write everything, creativity, in get any article. Okay, so we can read. And I have mentioned about the many softwares and the tools. Okay, there are many tools are there. So recently, I have visited Indonesia in the month of September 2023. It's about AI. International workshop. I represent India. I gave a presentation on artificial intelligence and climate learning futures. Okay, how the climate learning futures. generation players and futures. So there is really wonderful presentation there. So definitely you are the resource students are going to be retain it.
Thank you, sir, for sharing your valuable thoughts. Now it's time to express our gratitude to a small token of appreciation. I thank our esteemed resource person, Mr. Vinay Acharya, for the moment as token of gratitude for the session. I request Dr. Shweta S. to thank our chairperson, Dr. Anil Kumar Jain, with a moment as a token of gratitude. I request Srimati Meghana M. as an assistant professor, Department of Biochemistry and Chemistry, to facilitate Dr. Shweta. Once again, we extend our sincere thanks to our distinguished resource person for the enlightening session and to our respected chairperson and co-chairperson for guiding the session so effectively.
This is a short video.
ಆಡಿಯೋಗೆ ಏನೆಲ್ಲ ಕನೆಕ್ಷನ್ ಕೊಡಬೇಕು ಇಂಟರ್ನೆಟ್ ಇಲ್ಲ ಸರ್ ಡೌನ್ಲೋಡ್ ಆಗಿಲ್ಲ ಸಿಸ್ಟಮಿಂದನೇ VLC ನೋಡಿ ಓಪನ್ ವಿಂಡೋಸ್ ಮೀಡಿಯಾದಲ್ಲಿ
ಹಾಕಿದ್ದೇನೆ ಬರ್ತಿಲ್ಲ ಅದಕ್ಕೆ ಸರ್ ಇದು ಇದು ಸರ್ ಇದು ಕೊಡಿ
congratulate the Department of Life Sciences. Hello everyone. Department of Opportunity to congratulate the Department of Life Sciences in association with SDIM, SDM Reps, SDM University, under the aegis of Mr. 2.0 program for organizing this mega event today National Conference on Research and Innovation in Life Sciences. I'm sure that the event has been organized effectively under the able guidance of the principal, Professor M. Bharti, the advocacy coordination with the K. Sugita, Associate Professor, Head of Department of Computer Science, and Professor K. Satyamurthy, Research Director at SDM University, Harvard. I'm so happy that the students have also participated. Our college students have wholeheartedly given their volunteership towards this program, and I encourage that they will continue doing the same because not only will they be exposed to the latest developments in the field of life sciences, but also through this participation, they will definitely develop their personalities. I admire the participants who have registered for this program because it is evident that they have a thirst for knowledge and that they are willing to sacrifice their time and energy to gain the knowledge and also share knowledge. And hence I expect and I also encourage that there will be meaningful collaborations created through this platform and that whatever knowledge or whatever expertise that they have gained or acquired through these talks and sessions would inspire the participants to think out of the box and come up with solutions that address the various problems faced in our society. So, as personally, I miss you all. I wish I was there, but I'm sure that the time will come when we will all meet again. So, for this, I heartily congratulate and appreciate the efforts of the staff, Athiya Madam, Shweta Ma'am, Rajeshwari Ma'am, Meghana Madam, Abhishek Sir, Kantashri Ma'am, for their love and passion for their profession and for their heart that they have for their students to ensure that they get the best of education through these kind of programs. So I wish all of you success in all of your life's endeavors and I miss you all. I look forward to the day when we will meet again. God bless you all.
P314, I repeat, P314. Please do enjoy the breakfast and please report here by 10 o'clock so that we can continue with the call.
Sir, our problem with the ನಂದು ಬೆಳಗ್ಗೆ ಬಂತಲ್ಲ ಬರುತ್ತೆ ಸರ್ ಅದು ಕನ್ಫರ್ಮ್ ಓಕೆ ಅಲ್ಲ ಅಲ್ಲೇ ಲ್ಯಾಪ್ಟಾಪ್ ಕನೆಕ್ಟ್ ಮಾಡಿದ್ರು
ಅಷ್ಟು ಉದ್ದ ಇಲ್ಲ ಸರ್ ಕೇಬಲ್.
ಆಪರೇಟ್ ಆಗ್ತಾರೆ
ಕರ್ಸರ್ ಪಿನ್ ಕ್ಷಿತೀಶ್ ಸರ್ ಒಟ್ಟಿಗೆ ಕರ್ಸರ್ ಒಟ್ಟಿಗೆ ಪಿನ್ ಇದೆ. ಎಮ್ಮ ಇಲ್ಲ ಇದೆ ಸರ್.
ಹೇಳಿ ಮೇಡಮ್. ತಿಂಡಿ ಆಯ್ತು ಮೇಡಮ್, ತಿಂಡಿ ಆಯ್ತು.
Take one share, no? You have it? 11 today.
Floor 3 that's correct ಆಗಿ ಬರಲ್ಲ most of.
And
Connect this page. Patience.
I don't have that.
It is way faster.
Mine is way faster than that.
So remove this also.
How many members are watching this?
Devyashi.
ನಮಗೆ ಕನ್ನಡ ಮಾಡೋಣ
ಅಂತ ಮಾಡ್ತೀವಿ.
ನೀವು ದೇವ್ರು ಹೇಳ್ಬಿಡ್ಬೇಕು ಅನ್ನೋ ಅವ್ರಿಗೆ ಇಲ್ಲ.
You open my first.
ಆತನೋ
ಇರ್ಲಿ ತೆಗಿಬೇಡಿ ಈಗ ಪುನಃ ಇರ್ಲಿ ಇಲ್ಲ ನೋಡೋಣ ಇರ್ಲಿ ಕಣಮ್ಮ
ಮೊನ್ನೆಯಿಂದ ಹೇಳ್ತೀನಿ ರೆಸ್ಪಾಂಡ್ ಮಾಡ್ತಿಲ್ಲ ವೈಫೈ ತಗೊಂಡು ಹೋಗ್ತೀವಿ ಅಂತ ಅಲ್ಲಿ.
ಇನ್ನೊಂದು ಹೇಳಿದ್ರು ಇಂಗ್ಲಿಷ್ ಡಿಪಾರ್ಟ್ಮೆಂಟ್ ಅವರು ವೈಫೈ ತಗೊಂಡಿದ್ದಂತೆ ನೋಡೋಕೆ ಮಾಡಿಸ್ತಾರ ಹೆಸರು ಶ್ವೇತಾ ಚೈತನ್ಯ there was an issue on our end. We don't know. Drive through water. ಇರಿಸಿಗಳು
ಕನ್ನಡ
I will search making myself only I will search. It's on there, bro. You download Chaitanya video. I miss her so much.
spell her name.
Chai Hug.
Not on 2 minutes, 1 minute video. She even got Chaitanya Ram's video. He needs her to people.
Maybe.
I have to skill
ಹೋದ್ರು ನಾನು ಕೊಡಲ್ಲ ಕಣೆ ಅದನ್ನ ಯಾರು ಎತ್ಕೊಡೋಗ್ಬಿಟ್ರೆ ವ್ಯಾಲ್ಯೂಯಬಲ್ ಮೌಸ್ ಒಂದು
ನಾನು ನಂಬಲ್ಲ.
ಯಾರಾದ್ರೂ ಪಿನ್ ಕದ್ಕೊಂಡು ಹೋಗ್ತಾರೆ
ಎಷ್ಟು ಡಿಲೀಟ್ ಮಾಡೋದಿದೆ ಗೊತ್ತಾ?
ಅಲ್ಲೇ ಕಾಪಿ ಮಾಡಿ ಹಾಕ್ಬಿಡೋ
ಇಲ್ಲ ನಾನು ನೋಡ್ತಾ ಇದ್ದೀನಿ. ಅಲ್ಲಲ್ಲ ನಾನು ಹೇಳ್ತಾ ಇರೋದು ನಿಮಗೆ.
ಡೆಸ್ಕ್ಟಾಪ್ ಕಾಪಿ ಮಾಡಿ
ಇದೆಯಾ ಬ್ರೋ ನೀವು
MMKSDM this one
ಸೊನ್ನೆ
हाँ
My brain is not braining.
Forest
Hey bro,
ಅದನ್ನ Forest
ಹಾಕ್ಬೇಕೀಗ.
ನಂಗೆ ಬೇಡ ಅದು ಕಾಪಿ ಆಗುತ್ತೆ ಅದು ಬರೀ ಸೆಕೆಂಡ್ ಇದಕ್ಕೆ ಒಂದು ವೇಳೆ ಅವ್ರ ಹತ್ರ ಮಿಸ್ಪ್ಲೇಸ್ ಆದರೆ ನಾವು ಕೊಡ್ಬೋದು
ಮಿಸ್ಟೇಕ್ಸ್ ಹಾಕಿದ್ದೀನಿ. ಏನಿಲ್ಲ ಕಣ್ಣ. ಟೇಬಲ್ ಕವರ್ ಆಗ್ತಿಲ್ಲ ಬರೀ
ದಿಸ್ ಪ್ರೆಸೆಂಟ್ PPT ಅಲ್ಲಿ ವಾವ್
ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್.
ಇದೆ ಹೋಗುತ್ತಾ ಅಂತ Like beetroot juice is good, bro. It is good, but it does not. Some people it does not suit. Inauguration ಹಾಕ್ಬೇಡ ಡೈರೆಕ್ಟ್ ಅಜಯ್ ಕುಮಾರ್ ಓದಿಲ್ಲ
ಅಲ್ಲಿ ಹೋಗ್ಲಿ ಮುಂಚೆನೇ ಹಾಕ್ಬಿಟ್ರಾ?
ಸೆಕೆಂಡ್ ಹಾಕ್ತು ಆರೋಗ್ಯನ ಇವತ್ತು
ಚಂದು ಇದೊಂದು ಸರ್ಪ್ರೈಸ್ ಇದೆ ಚಂದು. ಇದೊಂದು ಸರ್ಪ್ರೈಸ್ ಇದೆ ಚಂದು. ಇನ್ನೊಂದು ಸರ್ಪ್ರೈಸ್ ಇದೆ ಎಲ್ಲರಿಗೂ. ಬರಬೇಡ ಸಿಗಲ್ಲ ಈಗ. ನೀನು ಬರಬೇಡ ಗೊತ್ತಿಲ್ಲ ನನಗೆ. ಸರ್ಪ್ರೈಸ್ ಮೈ ಸ್ವೇರ್ ಹರ್ ಸ್ವೇರ್ ಯೋರ್ ಸ್ವೇರ್
ಬೇಡ ಬೇಡ ಪ್ಲೀಸ್ ಬೇಡ ಬೇಡ ಒಂದು ನನ್ನ ಹತ್ರ ಇಲ್ಲ ಇದು ಚೈತನ್ಯ ನನ್ನ ಹತ್ರ ಇಲ್ಲ ಎಲ್ಲರನ್ನೂ ಕರಿತೀನಿ ಅದಿಲ್ಲ ಎಲ್ಲರನ್ನೂ ಕರಿ ಅದು ಬೇಕಾಗಿರೋದು ನಮ್ಮ ವಾಟರ್ ಬಾಟಲ್ ಎಲ್ಲಿದೆ
ಅಲ್ಲಿ ಇಟ್ಟಿದ್ನಲ್ಲ ಲಾಸ್ಟಲ್ಲಿ.
ನೋಡ್ತೀನಿ ಅಂತ
ಓಕೆ
ಮಾಡಿ ಮಾಡಿ
ಏನು ಮಾಡಕ್ಕೆ ಇಷ್ಟ ಇಲ್ಲ.
ನಿದ್ರೆ ಮಾಡ್ತೀನಿ ಆ ಟೈಮ್ ಯು ಆರ್ ಅಂಡರ್ ಹರ್ ಯು ಆರ್ ವಿತ್ ಹರ್ ಯು ಆರ್ ಗೋಯಿಂಗ್ ಫಸ್ಟ್ ಚೆಕಿಂಗ್
ನಾನು ಪ್ರಾಜೆಕ್ಟ್ ಮಾಡ್ತಿದ್ಲು ನಾವು ರಿವ್ಯೂ ಮಾಡಿಕೊಂಡು ಅದಕ್ಕೆ ನಾನು ಬರಲ್ಲ ನಾನು ಇದಕ್ಕೂ ಬರಲ್ಲ.
ಮೋಸ್ಟ್ ಪ್ರಾಬ್ಲಮ್
ಈ ಕಡೆ ಬಾ.
know. Maybe they didn't like for me too. Just to keep like this.
Day-2 edit ಮಾಡಿಲ್ಲ
ಏನು ಮಾಡಬೇಡ you put I will take your password ಇರಲ್ವಾ
ನಾನು ಅವ್ರು ಕೊಟ್ಟಾಗ ಏನು ಕೊಟ್ಟಿದ್ದು ಅದನ್ನೇ ಪಾಸ್ವರ್ಡ್ ಇರುತ್ತೆ ಅದು ಚೇಂಜೇ ಮಾಡಿ ಮಾಡಕ್ಕೆ ಮುಂಚೆ ಬರ್ತೇನೆ ಕನೆಕ್ಟ್ ಮಾಡಿದಾಗ ಮಾತ್ರ ನೆನಪಾಗುತ್ತೆ ನನಗೆ ಅವ್ರು ಪಾಸ್ವರ್ಡ್ ಚೇಂಜ್
ಏನು ಎಕ್ಸೆಪ್ಷನ್ ಅಂತೆ ಕನೆಕ್ಟ್ ಆಗ್ತಾ ಇಲ್ಲ
ಹಾಂ ಸರ್ ಮಾಡ್ಬೋದು ಸರ್ PPT
Final year, working in the college, studying the last 3 years, and this is a very ಇಲ್ಲೇ ಬಯ್ಯೆ
I seriously have not seen.
ಥ್ಯಾಂಕ್ ಯು ಸರ್
Check in.
ಯಾವುದಲ್ಲೂ ಇಲ್ಲ
Extra water bottle by any chance here?
ಮೈ ವಾಟರ್ ಬಾಟಲ್
Where's it?
ಇಲ್ಲ
This is with
ಇಲ್ಲ
Slide ಇಲ್ವಾ?
ಅಲ್ಲ ಇಟ್ಟಿಲ್ಲ ಸ್ಲೈಡರ್ ನೋಡಿಲ್ಲ
We are supposed to
It took to change the inauguration to RFQ. No, we have not made it.
add one slide here. In the way. You love me. Yeah.
Click ಮಾಡಿ
BA college for computer innovation.
ಇದೆಲ್ಲ ತಗೋ ಫಸ್ಟಲ್ಲಿ ಇದೆಲ್ಲ ತಗೋ which PPT ಪ್ರಾಜೆಕ್ಟ್ ಮಾಡಿದ್ರೆ ಗೊತ್ತಾನೆ ಅದನ್ನ ಎಕ್ಸ್ಪೆಕ್ಟ್ ಮಾಡೋದು ಆಯ್ತಾ ನನಗೆ ಗೊತ್ತಿಲ್ಲ ಜೀವ ಇದೆ ನೋಡ್ಬಿಟ್ಟು ಹೋಗ್ಬೇಕಾದ್ರೆ ಎಲ್ಲಾದರೂ ಹಾಂ ನಾನು ಇವಾಗ ನಿಜ ನಂಬಿಕಾಗಿ
It is there, Chandana. It is there. You will gain 1 second. You wait. No, please. Not that. Wait. ನೀನು ತಡಿಯಮ್ಮ ನೀನು ಅಯ್ಯೋ this you keep different that you keep different ಇದು ಅರ್ಥ ಕಣಮ್ಮ ತಡಿಯಂತೆ really like Anusha. There is a special video we will show. ಅದೊಂದು ಹೇಳುವಲ್ರಿ
ವಿಡಿಯೋ ಇದೆ
there is
ಚೈತನ್ಯ
a
ಮ್ಯಾಮ್
video, a message we would like to present to all the students. Please view that.
ಮ್ಯಾಮ್ ವಿಡಿಯೋ ಇದೆ
ಚೈತನ್ಯ ಮ್ಯಾಮ್ ವಿಡಿಯೋ ಇದೆ
ಆಮೇಲೆ ಪ್ಲೀಸ್ ಸೆಷನ್ ಲೇಟ್ ಆಗುತ್ತೆ
ಸರಿ ಮ್ಯಾಮ್.
Present morning to annual presenters, respected dignitaries, our esteemed resource persons, chair, co-chairpersons, faculty members, participants, and students. I extend a warm and hearty welcome to one and all present here. I request our dignitaries to settle on the seats. I call upon our resource person, Dr. Ajay Kumar Odi on the dais. With the immense pleasure, I extend a heartfelt welcome to our esteemed resource person, Dr. Ajay Kumar Odi, Additional Associate Professor, SDM Research Institute of Biomedical Sciences, Sri Dharmasthala Manjunatheshwara University, Harvard, for this session. We welcome you, sir. I request, I request Chairperson, Dr. Shiva S. to welcome our resource person, Dr. Ajay Kumar Odi, slowly. Thank you, sir. I also welcome our chairperson, Dr. Shiva S., Assistant Professor, Department of Biochemistry, School of Life Science, JSS AHER Mysore. We welcome you, sir. I request co-chairperson, Dr. Shivshankar P., to welcome the chairperson, Dr. Shiva S. Lodis. Thank you, sir. I would like to extend a warm welcome to our co-chairperson, Dr. Shivshankar P., Assistant Professor and Head Department of Environmental Science, MSc and SDM. We welcome you, sir. I request Srimati Kantan Sri, Assistant Professor, Department of Biochemistry, MSc and SDM, to welcome the co-chairperson, Dr. Shivshankar P. Global. Thank you, ma'am. Now I call upon Rati Jain from second PTDM to formally introduce our esteemed chairperson, Dr. Shivaras.
Good morning, everyone. It is an honor to introduce our esteemed guest today, Dr. Shiva S. Dr. Shiva S. is currently serving as an assistant professor in the Institute of Biochemistry, School of Life Sciences, at Basis Academy of Higher Education and Research Science. He obtained an MSc and PhD in biochemistry at the University of Kansas, Mexico area. Dr. Shiva's research focuses on the standardization of taxonomic applications programs and a various biological solutions what were existed on post-test international studies. He has recently grant from his post-execution of his research efforts and is currently mentoring three PhD students. He has contributed several book chapters and published multiple research articles in different national and international journals. It's an honor to have him here, sir.
Thank you, Rati. I request our chairperson, Dr. Shiva S., to introduce our esteemed resource person, Dr. Ajay Kumar Odi.
Namaskaram. Om Shri Manjunath Namaha. Thank you for the nice introduction. It is my great pleasure and privilege to introduce our next speaker, Dr. Ajay Kumar Odi. Dr. Ajay Kumar holds MSc and PhD in biotechnology. Currently, he serves as in-charge principal and associate professor at SDM Research Institute for Biological Sciences, Sri Dharmasthala Manjunatheshwara University, SDM College, Australia, Harvard, Canada, India. He has 14 years of experience at both UG and PG level of teaching, 2 years of postgraduate teaching at Department of Biotechnology, Gulbarga University, 4 years at Department of Biotechnology at BG Shivlal College of Art, Commerce and Science, and 8 years at SDM University. Dr. Ajay Kumar has an impressive research profile based around 51 research publications in both the national and international journals, 8 book chapters to his credit. He has received 3 awards in recognition of his academic and research excellence in area of research mainly involved in medical biotechnology. Dr. Ajay Kumar has participated in more than 20 training programs, workshops, conferences, and delivered 15 specialist talks across various scientific platforms. In addition to his teaching and research responsibilities, Dr. Ajay Kumar served as Board of Studies member at Colorado University and also an external and internal examiner for MSc programs at several reputed institutions, including Gulbarga University, Nalanda University, ESK University, Kalani, Karnataka University, and Oman University, Vijayawada. With this introduction, let us warmly welcome Dr. Ajay Kumar and request to deliver his talk. So, welcome.
Good morning, everyone. Om Shri Manjunathaya Namaha. I thank you, the organizer, me to handle part of this 2 days national conference. I also thank Dr. Shiva to introducing me to this all occasions. So the main topic for presentation is, I think you heard about inflammation. Yes? Inflammation is simple. The inflammation means like if you have a bite, so your skin will become a redness. So that is nothing but a simple inflammation. Or when you are affected from viruses or bacteria, you get some fever. That is nothing but an inflammation. So what is the reason? Inflammation can lead to the chronic inflammation. That chronic inflammation leads to the disease. So that is the main focus. Inflammation leads to the other form of diseases. So the main thing is immunology part. So we have immune cells. So we have immune cells like neutrophils, macrophages, monocytes, natural killer cells. So these are nothing but immune cells. So these immune cells are mainly plays an important role to have a defense mechanisms. Same thing, they have a role in case of inflammations. Like for example, like there are two types of cells, that is intracellular and extracellular. So the intracellular are nothing but neutrophils, natural killer cells, so like this occurs. So these are the three major cells which play an important role in case of inflammation. So extracellular cells are nothing but coagulation proteins. You know that coagulation protein is the input for the blood clot, and they have a C protein that is nothing but complementary protein which plays an important role in alternative therapies or alternative monoamine pathways. So where they play an important role for defense mechanism. And we have cytokines among that in the surround. So these are the major cells which are playing an important role to cause the inflammation. The simplest definition of inflammation is complex biological process by which body responds to the pathogens or usually irritants. So what will be the sign of inflammations? Right? When there is a vascular changes, they will undergo some two types of changes. That is what we call as vascular dilatation Like for example, what type of the sign we get, that is heat and redness, and we have a fever. That is nothing but vasodilation process. When there is an undergo for capillary permeability, it leads to the swelling, pain, and temporary loss of function. So if you see the quarter, so you are able to see that is heat, that is one of the signs. Then we have a redness, then we have a swellingness, then we have a pain, and finally we undergo for loss of function. So information have the two important roles. We call it as good and bad. So what will be the good and what will be the bad? So in case of good, we call it as acute phase or normal phase. So there will be a progression of healing, like through the chemical agents or poor bite or cuts, stabbing. So where cells are able to recover. And even for example, when there is a strain or a broken bone, so there is fever. And most common, the sign of inflammation is allergy. We know that so many people are allergic for even peanuts, some milk products. So that is also a sign of defense mechanism where the inflammation can show in the form of allergic reaction, and you can come to know, okay, this is the allergies. And burn, like even sun burns or fire burns. So these are the names. And another, the most common is infections. The infection will be any type of viruses or bacteria. So that is the first kind of sign where you get the inflammation in the form of a fever or the pain. Suppose this inflammation progress towards the destructive process. So what it will happen when it will undergo for chronic inflammation, it leads to cardiovascular diseases, it is going to make alcoholism, rheumatoid arthritis. Now we know that rheumatoid arthritis is one of the most common autoimmune disorder. So another thing is we have other form of autoimmune disorder. One among autoimmune disorder is inflammatory bowel disease. And chronic inflammation also leads to the neurological diseases, cancer, Alzheimer's. So that is the big good and bad about the inflammation. So based upon the inflammation, so we undergo for one of the most common diseases, or we can call it as autoimmune disorder. So whether you heard about or not, we call it as inflammatory bowel disease. So inflammatory bowel disease is nothing but the inflammation which causes for large intestinal. So that large intestinal cause is called as ulcerative colitis. When it will undergo for the infection in the small intestinal, it will undergo for Crohn's disease. So chronic inflammation leads to this type of two type of diseases. So these are the main two forms of IBDs. What are the other non-form of IBDs? Generally we call it as ulcer bowel disease, functional bowel disease, lymphocytic colitis, infective colitis, bacterial colitis, and eosinophilic colitis. So once there is a confirmation in the colonoscopy and immunohistochemistry, they are going to make a grading whether it is an IBD or non-IBD. So the most we know that another thing is called as irritable bowel disease or a syndrome. So what may be the symptoms or what may be the outcome? So no tissue is abnormality, change in only the bowel habits, the outcome is nothing but diarrhea, constipation, halting bowel patterns, and pain relief with the bowel movement. So these are the major common symptoms for irritable bowel syndrome. If you see the scenario of the incidence, the most common IBD reported has been shown in the United States. Among them, they have shared both the forms of IBD, that is 50% of ulcerative colitis, 50% of Crohn's disease. And if you see the ratio, that is 1 new cases per 1 lakh people per year. Now it is increased to 15%, means 15 per cases for 1 lakh people. And it is mostly affecting for youngsters. So the age between 17 to 28, that is a peak age where we are able to see the most common is ulcerative colitis. What are the factors which influences for causing IBD? So this is, if you see the intestinal, the most common is the environmental factors. So the environmental factors come all type of water, air, anything. It may be even small of the infections which is caused by the water. Medication, now we will talk about more about the medication because antimicrobial resistance. We take more amount of antibiotics. So the main thing is unprescribed antibiotic is causing the problem for the intestine. So without diagnosis, now we are making use of antibiotics. That antibiotics leading the cause for the IBD. So next, the diet, we know that the diet are two types of diet, what we call as the natural diet or the western diet. So the western diet is also making the most positive due to the more fatty acid that replicate making work for the intestinal problems. Smoking is another form of the external factors, hygienic condition, and we have also called as genetic susceptibility. Already, so many people have genetic susceptibility, like especially the GOT1 gene, which is the most common responsible gene for causing diabetes. And nowadays we talk about more about gut microbiome. So gut microbiome is nothing but so the word called as dysbiosis. Dysbiosis is nothing but when there is an imbalance of microbes. When there is an imbalance of microbes, that is one of the most common leading factor for causing disease. Not only for IBD, it is causing for cancer, it is causing for infertility. So there are a number of reported outcomes come across for change in the gut microbiome or dysbiosis. And once the person is undergo for the appendectomy, so this appendectomy has more prone to have causing the ulcerative colitis and Crohn's disease. And sleep, so this is one of the most common. So yesterday even the Srinivasa has been discussed about the sleep. When there is a sleep, we undergo for stress. This stress will undergo for the change in the attitude or physical activity, even that also leads to the incapability to cause the activities. Same thing, like stress and physical inactivity is also causes for the activities. So these are the major factors which causes for activities. So I said that is how this interaction is understood. So like for example, we can take diet. So it is vice versa for microbiota. Sometimes we undergo for tolerance and epigenetic modification and other environmental factors which causes for the impairing the intestinal epithelial layers. So I said that is there are two types of diet. One is called as healthy diet, another one is called as Western diet. So in this diet is nothing but vegetables, fruits, fiber content, and most important is sugar and vitamin D. But they are lacking of this type of healthy diet. But they are more prone to have Western diet like low fiber, high fat content, no importance of PUFA, and they are more intake of sugars. So this imbalance of the diet is mainly impairing the intestinal epithelial cell and leading causing for the chronic inflammation. So there are four stages to cause the inflammatory bowel diseases. So stage 1 or phase 1, phase 2, phase 3 and phase 4. So I said that is in case of phase 1 we call that pre-disease stage. So that will be answered by the genetic factors or antimicrobial factors, autophagy, handling of bacteria, chemokines and cytokines. And if you see the environmental factors, it may be a microorganism, diet, infection, stress, and another thing is non-steroid anti-inflammatory drugs. So this is another most common which is causing for HIV. And appendectomy, smoking, and antibiotics. So these are the first line of causing the infection. When there is an infection, there will be a trigger. There will be impaired barrier function will be open. Then when there is an open up, there will be a translocation of microbial factors. So this will activate for your immune cells. Or it may be dysregulated for immune cells. Like for example, T regulatory cells is being inhibited. Or they are going to produce more inflammatory cytokines. So this will lead to cause acute intestinal inflammation. That is second phase. In third phase, the chronicity, that is visualization is going up and there is a chronic inflammation will be developed. And finally, we call it as phase 4, where it is causing for total tissue destruction, where it is going to cause fibrosis, or it can cause stenosis, or it can cause dialysis, or even it can cause for crystal or cancer. So this is how there is a progression of the first phase to fourth phase. Similarly, this is going to trigger for immune cells. I said that is the most commonly cytokines. Maybe growth, anti anti-inflammatory or inflammatory side effects, which is able to cause the normal functions of the cells. So when there is an inhibition of these cytokines, then this will lead to the chronic inflammation. So this is the simplest mechanism how the factors influences causing for disease. So now we have a diagnostics. There are three major diagnostics are there. One is called as clinical diagnostic, which is most commonly we do like CR, like CIR2 protein, ESR, EGFR, immunoglobulin test. So these are the most common clinical diagnostics. Then later we go for diagnostic imaging. We use ultrasound or we go for colonoscopy or we go for endoscopy. Now we have high end of CT scans also where the exact location of the inflammation can be found out. And diagnostic matters. So diagnostic biomarkers, we have immunological biomarkers, we have serological biomarkers, and we have clinical markers. One of the most common is calprotectin. Clinical calprotectin is the most common what we call as gold standard marker to diagnose for ulcerative colitis and Crohn's disease. And we have also specialized markers like T-lactate. So these are the major common biomarkers are being used for the diagnosis of the AIDS. So similarly, we have a treatment approach. So our treatment approach, we have immunosuppressive, we have anti-sense autoimmunotherapy therapy, we have immunosuppressive therapy, we have monoclonal antibodies. Now we have more is concentrated on monoclonal antibodies because they are very specific. But the main drawback of monoclonal antibodies is nothing but they are cost effective. Even monoclonal antibodies also being tried for COVID-19, for even cancer therapies, there will be monoclonal antibodies. And now we are more prone to have probiotics. So the people are taking more probiotics such a way that your gut microbiome or your normal microbiome can be have a balance. And other medication, so we can go for T cell regulating cell therapy or FMT is nothing but fecal microbiota therapy, fecal. And we have antibiotics, but I said that is even the overuse of medication of antibiotics also leading to cause the chronic inflammation or resistance. And corticosteroids, so this is another one form of steroid, which is commonly used for RB, what we call as a treatment for IBD, but it is leading to the more prone to have side effect for the other organ systems. And finally, they go for surgeries. So if you see the epidemiology, so earlier we see that US is the major the country which have IBDs. If you see now, the India is also beginning for having the more causes of IBDs. So I said that it is any type of factors which may causes for the IBDs. So if you see the trend line, so USA, Sweden, Japan, South Korea, and China, and India. So if you see that USA after that, we have the Indian. So if you see the locations of your niche, that is based upon the locality or the location of the countries, there are numerous changes in the infection. So in case of ulcerative colitis, so we have three types forms. One is called as left colitis, right colitis. So that is the colitis. Based upon that, so they have been differentiated what will be the ratio of causing the IBDs. So this is one type what we have the analysis like based upon the north zone, eastern zone, central zone, western zone, and south zone. So then we have Chennai, Manipur, and Bangalore. So where we have more information about the causing IBDs. So if you compare to the north zone, east zone, or central zone. So same thing we are undergone for the analysis of the drugs for both ulcerative colitis and Crohn's disease. So the common disease is mesalamine. So status is nothing but never reverse and correct. So there is a chance of causing the recurrent also when we have mesalamine. So next we have for 5-aminosalicylic acid. So this is another common drug which is used for IBDs. We have corticosteroids and we have immunosuppressive and infliximab. That is another form of monoclonal antibody, but there is a less causing the recurrence. So next is comes about your probiotics. That is I said that probiotics is nothing but the main thing is to make enhancement of the normal microflora and element diet and have the another form of the thing where they can work on for the IoT information. Anti-disruptive, so this is to reduce the suspension. So based upon the information regarding our form of your IoT, so when we can understand for the studying retrospective analysis. So retrospective analysis we done from our website hospital. The patient was coming for our SDA medical college. We undergone for the retrospective study for 5 years. So that data we have published in the gastrointestinal journal. So based upon that, we went for prospective studies. So in case of prospective studies, we have taken the patient sample like tissue and blood. So we have taken the consent form from the patient who have undergone for this particular type of a diseases. After consenting from the patient, so we have gone for the screening for immunohistochemistry, normal histochemistry, and we went for gene expression level, whether any changes are there. So we want to compare between the dead and tissue samples. So these are some of the images where we are undergone for the immunostaining staining, that is endoscopic reversing staining. So there is a confirmation that there is a change in the observations of the change in the cell when they are exposed to different magnification. That is indicates that is between the control and the disease samples. So this is the study I said that is we went for the analysis of the gene expression level. So one of the ideas we have one is nothing about stress protein which is causing for the IgE. So that we have checked it. And we underwent for the inflammatory markers like TNF beta and alpha and interleukin 6. So this is the changes where we are seeing between the blood and tissue samples. Similarly, we went for the correlation between the blood sample and tissue sample comparing the clinical outcomes. Like I said, that is ESR, CRP, albumin, alkaline phosphatase, age. So based upon that, we undergone for the analysis of the gene expression level comparing with the clinical outcomes. Based upon that, further we went for study called as MSI marker. So I think you heard about MSI marker, that is microbial stability markers, and we have MMR, mismatch references. So these are the two important genes where you can come to know whether information can be equal to colorectal cancer. So I said that is the MMR genes have 5 proteins. So when there is a deficiency in that particular 5 proteins, that can cause colorectal cancer. So among that is muta homologous 2, muta LS homologous 1, muta H6, and 4 meiotic stimulating 2 genes. So among these 4 genes, any genes like for example, the 3 genes are expressing or not expressing. Based upon that, we can grade whether it is a moderate high or whether it can able to have colorectal cancer. So this is one of the images where they are done for immunohistochemistry using these all 4 biomarkers with comparing with the control of the diseases. So based upon assignment, so immunohistochemistry, so we have MSH1, MSH2, MSH6, and PMS2. So we have undergone for both immunohistochemistry and we have undergone for PCR. So when there is a deficiency of these genes, and we can come to know that how that can develop for the colorectal cancer. So for MMR genes, we have used 5 genes, that is called as BAT25, BAT26, which are nothing but mononucleotides, and D2S123, D5S346, D17S2720. So these are nothing but dinucleotide genes. So this is one of the pattern where we are going to do the analysis of all 5 genes. So this is one of the page analysis. It's not a double development process. It is a polyphenol development process to have a low molecular weight. So this is how we have been differentiated the 5 gene distribution for this for blood sample. And similarly, we also carried for the tissue samples for analyzing these 5 genes. And finally, we have interpreted that based upon the immunohistochemistry and gene expression level. So we got 15 cases that is having microsatellites disabled, and 3 cases have only microsatellite that is low, and 7 cases they have chances of getting more instability high, the chances of causing the colorectal cancer. Similar this other developer process confirmation. So among that 8 cases are MSS, 5 cases are MSI, and 12 cases are IMSI. So finally, our team, so which is being headed by Dr. K. Satyamurthy sir. So we have around 8 faculties who are working in the different area. So like last I want to conclude that this is the thought obviously given by excellent our past president. So your education is the foundation, but your passion and hard work will create the masterpiece of your career. Never stop learning and always strive to make a positive impact with your skills. Thank you. So we have only about
questions from students? It was indeed nice presentation.
thank you.
I have a question. I mainly work on right now we are looking into the areas of very well known that pesticide causes that environment. And is there any relation between pesticide use
As of now, that pesticides even provide a of major irrigation. And most importantly, even the people who are selling the pesticides and the residues, what concentration is there, you are unknown. So among the examples are considered like 1%, so they will go 1%. Among 10%, 0.5% of residues will go in the common crop or any vegetables. Even after growing also, the residues are left will be there. That residues may inhale and is going to affect from starting from bottom of the water and leading to the other form that is in the sand. So even there is no that we call it as a the second grade. So where it is affecting for normal microbial or these. So that is leading to other form of diseases also that we are not able to detect whether it is causing lung cancer or esophageal cancer or IBD. But we have to rule out which type of causing it is. And most importantly, we have to find out which is the pesticide, whether that pesticide residues is there or not. So simple that more people are working that pesticide can be used for model the fish model that is which like good molecular. So generally fish model they use for cytotoxicity. So that we can study about how it is being effective with the standard pesticides and the normal pesticide which is you are getting. After we have DC analysis, so we come to know that what is it.
In that particular treatment impact more this particular process regarding activity. It is common or geographical factors or any seasonal impacts.
So it's all depends upon the season, city, geographical, everything works and everything matters. So that is why individually to individual relates. So not whatever it is what everything does in each patient and their population. So that is the change in the individual. Even it matters about the age, it matters about what they have died. So that is why we are discussing that while collecting the information we have bias because we not get all from the data from the patient. for example, I am now I have before, so I'm not this was a mistake of the habit. So in such cases, how it is very difficult to predict, which is the environmental factor.
Thank you for spending your valuable time to share your knowledge with us. We are deeply honored by your presence. I request Dr. Shiva S. to come forward and facilitate our esteemed research person, Dr. Ajay Kumar Oi, with a memento as a token of gratitude for the session. Thank you, sir. I request Dr. Shiva Shankar S. to thank our chairperson, Dr. Shiva S. with a memento as a token of gratitude. Thank you, sir. I request Srimati Rajaratneshwari, Assistant Professor, Department of Microbiology, MMK and SDM to felicitate Dr. Shiva Shankar. Thank you, ma'am. Once again, we extend our sincere thanks to our distinguished resource person for the enlightening session and to our respected chairperson and co-chairperson for guiding this session so effectively. Thank you.
There will be a short video presentation right now, so I hope you guys will enjoy.
All of this as a participant and congratulations to the entire team of the students and it is rather an emotional overwhelming time to me that I'm part of this as a participant and not part of my community. I genuinely feel that all the students as part of my community. Whoever it is to enjoy organizing for such a wonderful and brings together celebrations and I hope we all parents and that pushes us to explore and keep Wishing you many best.
I request the resource person, Dr. A. Satyamurthy, chairperson, Dr. Anil Kumar, and co-chairperson, Srimati Ramya Ma'am, to occupy the seat on the dais. It gives me the immense pride to introduce and welcome our resource person, Dr. K. Satyamurthy, Professor and Director, Sri Danuttara Manjunatheshwara University, Dharwad, for the session. We welcome you, sir. I request Chairperson Dr. Anil Kumar to welcome our resource person, Dr. K. Satyamurthy. Thank you, sir. I also welcome the chairperson, Dr. Anil Kumar KS, Associate Professor, JSS ASPR Mysore. We welcome you, sir. I request co-chairperson, Srimati Ramya SK, to welcome Sir Global. I would like to extend a warm welcome to our co person, Srimati Ramya SK, Associate Professor, Department of Computer Science, MSc and SDM MSc. We welcome you, ma'am. I request Dr. Shweta S. Head and Assistant Professor, Department of Biotechnology, MSc and SDM to welcome ma'am slowly. Thank you, ma'am. Now I call upon Tanushree from 2nd DPPM to formally introduce our esteemed chairperson, Dr. Anil Kumar.
Good morning, respected dignitaries, faculty members, and dear students. It is my great privilege to introduce our esteemed guest, Dr. Anil Kumar KM. Dr. Anil Kumar KM is an associate professor at the Academy of Higher Education and Research, Mysore. He has extensive experience in teaching, research, with strong focus on water and health, water management, biodiversity conservation, and sustainability. He holds a PhD in Environmental Science from JSS ASPR, Mysore. His research includes environmental radioactivity, integrated water resource management, biodiversity and conservation, and water and wastewater management. Dr. Anil Kumar PM has published over 60 research papers in international journals, including significant contributions in photocatalytic degradation of contaminants and sustainable water management. He has received several prestigious awards, including the Young Scientist Award from the Global Research Foundation and University of Puno Geo, Indonesia, as well as the ICMR Senior Research Fellow Award. He has also established international sustainable development, and the synthesis and application of nanomaterials for environmental remediation. Through his work in teaching, research, and community service, Dr. Anil Kumar PM has made significant contributions to environmental science and sustainability. He continues to inspire researchers and students alike. We are truly honored to have him with us today. Have a wonderful day ahead. Thank you.
Thank you, Manshi, for your introduction. I request Dr. Anil Kumar K. to introduce our esteemed resource person, Dr. K. Satyamu.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ಓಂ ಶ್ರೀ ಮಂಜುನಾಥಾಯ ನಮಃ. I'm honored to be here to introduce our resource person, Dr. K. Satyamurthy sir. I welcome you, sir. Sir is the Director and Research at Sri Dharmasthara Manjunatheshwara University, Harvard. So, sir completed his MS in biochemistry at University of Mumbai, and he continued his research PhD in oncology and cancer biology. So it is very much required and it is very essential. So that Dr. sir was administrative and research position, especially in director position at the University in Harvard. Um, and the certificate that is building research and innovation institutions are acute and corporate number of research and academics. Sir has published more than 340 publications in his field and several patents that patented many patents and applications and has been recipient of several awards. Majorly, Satyamurthy sir was honored with the Dr. Raja Raman Award in the category of Science and Education Research in the year 2022. It is a great honor, sir. He received the award from the Honorable Chief Minister Siddharamayan sir in the function held in Bangalore in the presence of CNR, Professor CNR Rao. Okay, and sir, currently he is working as Director and Research at SDM University. I welcome you, sir, for this debate. Thank you.
Thank you, sir, for your introduction. Building on what has been shared, I now invite Dr. K. Satyamurthy to take over the session on relevance of microbiomes in cancer progression.
At the very outset, at the thank the organizers for giving me an opportunity to speak to you something about relevance of microorganisms in cancer progression. I don't know how many of you really think that microorganisms will have anything to do with the cancer. You think the viruses cause cancer? My dear students? Yes or no? Yes? No? No idea. Viruses don't cancer. Don't cause cancer. Very good. About bacteria, do they cause cancer? No? Truly amazing. On the contrary, I I think it has been amply established that I think he's just provoking me. He's not necessarily giving me the right answer. He's trying to provoke me to give me the answer. Is that what you're trying to do? So there has been ample evidence to demonstrate that that the viruses are in fact one of the major living organisms which cause cancer. And there are a number of examples. And more recently, there is a lot of emphasis on how the fungi, in fact, are also responsible for genesis of many cancers. And more recently, we have been hearing quite a bit about what microorganisms, which will include only bacteria, for example, many different types of bacteria, which inhabit the tumors, and then they probably would potentially promote cancer progression and not necessarily initiate. They may be one of the factors which are responsible for initiating, but not necessarily on their own, as we know of now. But they definitely are responsible for many, many different types of cancer. So I'm going to give you, keeping the audience in mind, very brief overview of how, what is the role of microorganisms, what are the different microorganisms, and how they in fact can promote immunogenesis process. And then I'll give you some examples of our own results for the last 2 years, which will, I hope, convince you that microorganisms have anything and everything to do with the human population. So, how many cells are there in the body? Okay? Our body has how many cells? Any guess? About 30, 35 trillion cells. You know how many zeros the trillions has? Trillion has? Can you tell me how many zeros are there in trillions? No? So that's quite a bit. That's quite a lot. Yeah? So if you think that there are about 30, 35 trillion cells in our body, and they are distributed across various organs within our body, as indicated in this slide. And then some cells, some tissues, some organs have more cells, some tissues, some organs have less cells. If you look into the erythrocyte, which in fact makes the largest portion of cells in our body, followed by the bone marrow cells and skeletal muscles. In fact, about 10 to the 11 cells which are there, and their specific cell types are indicated in the slide. I hope you can see. And more importantly, the microbiome is very famous, especially for its growth. What happens in the gut, in the stomach, small intestine, large intestine. There is also significant number of cells which are in fact indicated in the slide, which is primarily made up of cells and emergent processes can be indicated from this slide. So, what are the different types of bacteria that which are normally present in these organs and tissues, and that is what is indicated. And if you ask us, if you say that we have about 35 trillion cells, and the question also would come, how many bacteria are there in the body, right? Not about how many cells would be in the body, approximation, and how many bacteria in the body, and then they have nearly 38 trillion bacteria in the body. You would imagine how much could be, and the maximum you can find, as I said, in the stomach, in the gut, in the colon, large intestine, small intestine, nearly 10 to the power of 13. And there are some very beneficial bacteria which commonly reside in this organ, which includes formicutes and bacteria, which are approximately about 90% of the microorganisms which are populated in our gut. Similarly, we have skin, which is in fact the major epithelial tissue, covers approximately 80% of our body, has high number of bacteria which are formulated by actinobacteria and formicutes, and oral cavity, of course, the oral cavity is chock full of microorganisms, whole lot of bacteria, Streptococcus, Prevotella, and so on and so forth. I'll be talking a little bit more about cervical cancer, which also has a significant role. Now, coming back to cancer as an hormone, way back in 2020, we have Anahan, which is first
able to provide a broad characteristics of what we call it as harmless and it is subsequently updated in the recent years. And as recent as last week, as we started updated, the vast array of events which happened during the cancer progression have been broadened to an extent that is almost impossible to assimilate everything at one single time. So way back in 2020s when Hanahan and Steve proposed that there are major properties which are attributed to the progression of the cancer. Some of the most important properties are illustrated in this slide. These include the cancer cells being able to grow in an anchorating fashion. They don't need anything to adhere. They can just grow. They can pile up on each other and then they start growing. And similarly, they have a continuous proliferative signal. In other words, they produce their own proliferative signal. We call it as autocrine signaling, which will enable them to grow. And they don't die very easily. There is no programmed cell death. In other words, they are not programmed to die by themselves. They cannot program their own death. It's a very common phenomenon which we see in normal tissues and cells. There is a significant amount of genome instability. What I mean to say genome instability means frequently we find that the genome in a cancer cell are mutated in the form of either translocation, amplification, large deletion, so on and so forth, which provides the property we call it as an aneuploidy or chromosome magnetism, in which it acquires the property of being able to proliferate. Similarly, cancer cells also promote vasculogenesis because for any organ activity to grow, you need to have proper nutrition supply, proper oxygen supply, and these are provided by the blood vessels. But cancer cells have acquired the property to be able to make their own blood vessels. In other words, cancer cells convert themselves into the blood vessels, anastomose in the normal blood vessels, and facilitate the flow of the blood inside the body. And that is what we call it as inter-conversion phenotypes, which are frequently occurred in many cancers. And they have an ability to invade and metastasize. They can come out of their site of origin, they can invade into the dermis, and metastasize into the different organs which are compatible for them to grow. That's why you frequently hear that a cancer, let's say the blood cancer, is frequently metastasized to the lymph node, or even to the brain cell, or even to the kidney. So these are the frequent phenomena which we see from the primary side the cancer cells can progress. More recently in 2022, and then lastly in 2026, Anahan has incorporated many more properties for these cancer cells, which were learned over a period of time. A lot of research has happened, and they have added many more phenomena to that. One of the major one is this infection with the polymorphic microorganisms. In other words, various forms of microorganisms are also responsible for the emergence process. Now, what are the events which are in fact are happening from the cellular perspective or from the genomic perspective that is in fact is promoting the human cells? Because normally you have a diploid cells, and these diploid cells are now converted into a state where we call it a plasticity state, where they can continuously change in many different forms, both genetically and epigenetically, and it would give us properties that is detrimental to the normal cells, but are beneficial to the human cells to continuously grow and acquire all those properties which we described in the previous slide. So, what are some of the major changes which occur in the human gene, in the human DNA, over a period of time, which include mutations and single cell nucleotide polymorphisms, which are responsible for providing specific properties and specific functions of a given gene within the cell so that they can acquire the physiologic state. And as you know that nearly 70% of our genome is full of repetitive sequences, and they are in certain functions. And these repetitive sequences have lost their normal function, and now they are being able to transpose into inappropriate regions in the genome and provide the physiologic advantage of good advantage. Copy number variation. Copy number number variations occur at a frequency of approximately 45% of the genome, and these copy number variations provide an effect what is described as gene dosage effect. In other words, they can influence the extent of RNA formation, protein formation, which can have drastic effects on the process. Similarly, there are epigenetic diseases which are primarily described as women which does not change the nucleotide, but more by addition of some functional groups onto the DNA, which can change the function of the DNA. And one such functional group which has been added onto the DNA is another group, which we call it as DNA methylation process. And this DNA methylation process has a significant effect on how the cell behaves in relation to transcription can happen, and then thereby being able to provide appropriate proteins in appropriate quantities to promote the immunogenic process. There are several different types of epigenetic mechanisms. One of them is DNA methylation. There is addition of methyl group onto the cytosine nucleotide, and then we have the histone modification, which occur at the nuclear level, where these histones they provide a chromatin architecture, which can be either in what we call it as a chromatic or euchromatic state. The euchromatic state is the active form of chromatin, and this is in fact imparted by the type of post-translation modification which occur on the histone. Similarly, there is also a mechanism of silencing to microRNA sequences, which also, if together, can when they are together, they can provide affinity properties that can alter the phenotype of the cell. So, what are some of the examples of microorganisms which we have across the various organs in the body? So this slide basically tells you that there are every single organ, almost all of the organs which we know of, are in fact can be affected by the inappropriate colonization of microorganisms, which has a profound effect on the structure and the function of the given organ, and some of the examples are provided here. For example, we have a phosphate with the microorganisms which occupy and modulate are indicated here. We also have colon, the series of bacteria which are colonizing the colon tumors are indicated here. And then we have organs such as the stomach and the gut, which also show their mouth, lung, cervical, cervix, and also the prostate, and also the breast. If you see, you know that the cancers can occur in any of each of the organs, and cancers which are affecting these organs are also colonized by the microorganisms. And these microorganisms are very much responsible for imparting the tumor progression in the tumorigenic process, and I hope to give you the next few minutes to tell you how this can happen. So, some of the key mechanisms which can occur and how the microbiota can influence cancer progression is indicated here. First of all, these microbes they make number of metabolites. These number of metabolites are secreted, and these metabolites can in turn enter the cancer cell. In fact, they alter the chromatin to a state of activity. In other words, some of these metabolites, which are the single chain fatty acids, can regulate the functions of the chromatin and make them more active. Similarly, they have this significant property of altering the immune function. It is very well established, and especially when you have an infection or if you have a fever where your immune system is altered, and these microorganisms can alter the defense mechanisms of the immune cells, thereby promoting the tumor growth. Similarly, the bloodstream infections also can promote the way the tumor cells behave. And these microorganisms can contribute the clinical pathological features, metastatic potential. In fact, the infections that have been demonstrated that infection of the tumors with a certain type of microorganism promotes metastasis to the distant organs. Now, it's also been now frequently used as the type of population which represents the tumor now are used as a biomarker. They can indicate whether the tumors are growing or not. And more importantly, these are the very microorganisms which can also bring about resistance to therapy. Resistant to immunotherapy because immunity has already weakened in this patient, partly driven by the microorganisms, and resistance to chemotherapeutic treatment because they can bring resistance. So, specific examples are shown here. Microbiome surprisingly basically is a bacterium which activates the group of transcription factors called NF, the big family of proteins. These are in fact complementary cytokines and then potentially can activate the surveillance because it's very well established that some of these microorganisms through their metabolites can also damage DNA. DNA damage is one of the hallmarks of cancer. It can evolve against certain cancer phenotype. Immune modulation and variant factors are also being amply established, especially in the blood-related cancers and therapy resistance, as I have indicated before, are also can be caused by the bacterial infection. So, they can promote the metastasis problem of formation because one of the features of metastasis is immersion of a particular phenotype into a second phenotype, and that is effectively caused by organisms. Drug efficacy is a specific example of conversion of food drug into an active drug by fibroblast and exemplifying which is given for pancreatic cancer, which has been very well established in the past as a very important paper published a few years ago to demonstrate that the treatment of pancreatic cancer fails because of inactivation of drugs in the body by the invading bacteria. Similarly, it can modify our DNA, create immunosuppressive dish, and foster anti-tumor response. And certain exosomes which are released from the tumor cell can also promote, and that is what it is indicated, number of organs which get transitioned into different organs, and these are the potential organs in which the tumor can metastasize and make new tumors, and that's what the metastasis process contributed by the microorganism. So let me give you a specific example of some of the studies which have done, very basic, very superficial, just without going into great detail, to indicate how microorganisms would contribute, can contribute to the progression of cancer in cervical cancer. One of the major cancer is the seventh most common cancer in the world, and in fact it takes approximately about 60,000 people here in slide, please. One of the major causes of cancer believed to be the human papillomavirus. So viruses cause cancer, and human papillomavirus is one of them. It is very well known that 98% of the humans human cervical cancer consists of HPV virus. That is why HPV diagnosis is done in the women to see whether they are HPV positive or negative. A normal person, a normal woman, a normal lady will have approximately about 20 to 40% of them will have HPV. This subsequently, over a period of time, is a quite a determinated period can be cancer. But every single woman who has cervical cancer, 99% of them will have HPV. That is why it is believed that HPV is one of the major viruses that is responsible for the cervical cancer. Yes, it has been accepted for many, many decades. Yes, Zulhausen got a Nobel Prize for discovering the HPV virus way back, I don't know, in 2006. But we didn't believe that HPV alone is sufficient. It may be necessary, but it is sufficient to be able to cause cancer. So we went on to analyze to be able to see what does the HPV do, what else are the factors which are necessary, and why it takes so long to be able for HPV infection to be by two tests. The HPV is a very small genome, approximately about 6 kilobases. It has two crucial genes, which is responsible for inactivating protein, and it is believed that these two mechanisms are responsible for promoting the immunogenic process, and that is what it is indicated. Next slide, please. But then you can always ask the question, okay, if there is a bacterial infection, you kill them. You can kill them by antibiotics, right? And that is what you would imagine that if you have a bacterial infection in the cervical region, you take antibiotics, you eliminate, and then you are fine. And probably not. And that is what we are seeing in the last about 20 years, that there is significant increase in antibiotic resistance, where we are almost run out of antibiotics to be able to eliminate the bacteria, and the type of bacteria which are handling the cervical region are really difficult to eliminate. Secondly, there are very few think that we can eliminate microorganisms by killing the bacteria is out of the question because they develop resistance to antimicrobials. So we did a large training of people in the regions of Udupi and Karnataka. We looked at approximately 4,000 women. 4,000 women were normal women. Nothing is wrong with them. And we analyzed the cervical microbiome, and this is what we find. Approximately about 20 to 40% of the women had HPV present in them. And secondly, nearly 26% of the women had more than one type of HPV. Suggesting that it is not just one HPV type, there are different high risk and medium risk HPVs can infect these women and which proportionately enhances the risk of acquiring cancer over a period of time. Next slide, please. We also looked at the other viruses which they are present in the lung, and some of them is shown here. We looked at Epstein viruses, a known oncovirus, the EBV causes cancer, especially in the esophagus. We have looked for HSP, cystic fibrosis, which is established initially that responsible for cervical cancer. We also looked at CMV, which is not necessarily a cancer-causing virus, but then it can significantly influence the immune function. And we also looked at EBV, which I mentioned before. What is interesting to know that along with the HPV, we were also able to find other type of viruses in the normal women in equal proportion, almost in the equal proportion, suggesting that it is not necessarily just HPV, the other oncoviruses can also contribute to this cancer. What is shown in the bottom panel is primarily in normal women who are negative for HPV, who are positive for HPV, and the co-infection with other viruses. It suggests that whenever there is absence of HPV, the other oncoviruses are less also reduced, suggesting that there is an interaction between these viruses which may be inhabit the cervical region, subsequently over the period and lead us to cancer. So we did not really believe that HPV vaccine is probably the answer for everything. As you know, the HPV vaccines are very common, which is being prescribed, especially for young girls less than 60 years of age, or maybe about 30 to 22 years of age, saying that, look, this is going to prevent the progression of cancer. Primarily because there are at least 40 different types of high-risk HPV. Any one of them can infect the women. At the same time, there are some of the very common varieties which are present in India. The vaccines which have been made is only for the specific group, maybe HPV 16, maybe HPV 18, maybe it is a bivalent, and in one it is for Garrison 9, which is supposed to be effective against 9 different types of HPV. But then, what about the other 6 types of HPV? Whenever we take a vaccine, it has been proved as early as just about 2 months before that whenever you make a vaccine, whenever you suppress these 9 different types of HPV, invariably other HPVs take up, they start growing, and they start operating the region. Suggesting that even if you take HPV vaccine, one needs to be very careful to be able to prevent the growth of other oncogenic HPVs in the society. So we will about sequencing the women who are normal as well as cervical HPV themselves, and also people with the tumor using the traditional sequencing. I will not go into details, and this is what we do. You look at the kind of distribution of bacteria which you have in the normal women, in the women in the pre-cancerous or pre-malignant region, and in the women who have full-blown cervical cancer. And this is what it is. The black box indicates lactobacillus, right? Lactobacillus is a beneficial bacteria that is present in the cervix of the women, which is completely or very little which is present in the tumor. In other words, the normal bacteria is completely absent, or it is almost gone in the tumor. In other words, this beneficial bacteria, the loss of which can promote the pre-malignant and malignant. Similarly, there are other type of bacteria which are shown here. Serratia, then we have Staphylococcus, Bacillus, Alkaligene, Streptococcus, Klebsiella, Sporidium, so those pathogenic and non-pathogenic bacteria distribution are completely different. And this is what the process is called dysbiosis. From probiosis to dysbiosis, a completely bacterial population is change, and this was analyzed in 200 different humans. It's not just one sample, two samples, but this is an average study from nearly 200 humans. Just think that under or dysbiosis of microorganisms, the service is in fact is one of the hallmarks of the cervical cancer progression. Next slide, please. And the different type of bacteria and the relationship in the various stages of progression which is shown here. We call it as a plot. The red line primarily indicates those bacteria which are in high abundance in the squamous cell carcinoma or the tumors of the cervix are so very strong as opposed to the normal which are shown in the green and the relationship to the discovery of various microorganisms in the cervix. And this, so what is this picture? This result basically show what is the distribution across the bacteria across normal, invalid, and diffuse. What are common and what are unique? What is shared and what is not shared? And that's what it is indicated. Here, there are quite a bit of bacteria which is shared, but there are some unique bacteria which is in fact present in the normal, malignant, pre-malignant tumors, which have the potential to be used as by not only as a biomarker but also for special treatment. Next slide, please. So it is not very visible. What we did here is basically we took the tumors from the women, normal pre-malignant and malignant tumors, and we look for the bacteria inside the tumor. We did a process experiment called so-called FISH, where person things into our stations, to which we try to identify bacteria inside the tumors or by the tumor. And interestingly so, lactobacilli, we find that in abundance in the normal tissue, and other pathogenic bacteria such as Pseudomonas or Klebsiella, which is absent in the normal tissue, but it is present intra-tumor or inside the tumor, in the tumors of cervical. Next slide, please. Done now. We also classified them according to their abundance in the normal and pre-malignant and malignant tumors. Lactobacilli, Gardnerella, Pseudomonas, all of them have been mapped and their potential location as far as the tumor classification is concerned. We show here, next slide please. And then we also look at their antibiotic resistance pattern. What we did was basically we sequenced the entire microbiome specifically for the antibiotic resistant genes and look for the difference because it is another indication of what type of microorganism can be present, whether in the normal, pre-malignant or malignant tumor. And this is one of the indication how the antibiotic resistant genes which are present in this microorganism would tell you that merely by treating with the antibiotic is not going to take care of the problem. There is need to become much more than by treating the antibiotics. What is shown here is the definitive classification of co-segregation of antibiotic resistant genes as they occur along the microorganism which has been classified to be able to present across the normal pre-malignant what is shown here is primarily again the extent of microorganisms and their associations with the antibiotic resistant genes shown as a marker either in the blue or the red. Red is present, blue is absent, and then we were able to classify them in relation to their presence. And the job doesn't get there because many times, as I said, even if you give antibiotic treatment, it just doesn't work. And why it doesn't work? Because these antibiotic resistant genes, we have seen in our sample that accumulate mutations. These mutations are activating mutations in the sense that these mutations are actively metabolize or degrade the antibiotics in the patient so that they become ineffective, especially tetracycline resistant genes, which we find is mutated, which can affect the activity of many antibiotics which are commonly given for the antimicrobial treatment. Next slide, please. Again, this is a table to show resistance profile across 150 samples from 150 women, where we were able to classify the type of antibiotic resistance they have, the bacteria, and the sensitivity which occurs in these women. Next slide, please. And this slide shows various subtypes of viruses, as I mentioned, that HPV 16 or 18 alone is not sufficient. There are 14 different high risk, there are 24 different medium risk, and many more low risk HPV. And it is important to know polyviral infection of these tumors, and and that also is conducted performed along with the EBV and EBV and HBV viruses in these tumors of the brain of the cancer patient of the brain. So, what is the interaction between this HPV and the various viruses which are present? The first question to ask is whether the co-infection with the EBV or HPV would have any influence on the abundance of HPV or vice versa. What we should, as I simply mentioned before, the normal woman would have 40% approximately higher HPV, while in the tumors you would have 9% of the women who have HPV. But then, when we look at the normal women with or without HPV, we find that other viruses, EBV and HPV, are also present. And in relation to those which are HPV positive, we find abundance of other viruses as well as microorganisms. So, whether it is a coincidence or whether it happens simultaneously, or any one of these organisms that is responsible for such a phenotype is still not clearly understood. Experiments are on the way, and hopefully we will have an answer very soon. Next slide, please. And here it shows what HPV is a co-infection either with the positive or negative of HPV and microbial abundance in this stuff. Whenever we have HPV, whenever we have HPV and HPV, you have very high abundance of microorganisms as opposed to those which the lower panel which shows lower abundance of microorganisms. And it is shown here for EBV. EBV you are particularly interested because it is an oncogenic virus, and this oncogenic virus can potentially interact with the HPV and the mechanisms that are operating inside the tumor and then give rise to this viruses. And this virus is obvious lead to squamous cell carcinoma as opposed to negative squamous cell carcinoma. What happens then if we infect this with the bacteria? In this case, we took some of these bacteria lactobacillus and the Stenotrochomonas, lactobacillus, Stenotrochomonas, which is a non-fermenting bacteria, which is aerobic bacteria, and then we can safely infect that with the tumor cells. Nothing happens to the tumor cells. They are happy. They grow. So in other words, they don't have detrimental effect, but rather the tumors are surviving happily. But this has a significant effect on how the cells are shaping up to be able to grow. And that is what it is indicated here, where we look for generation of oxidative stress, oxidative radicals, and these free radicals have an effect on the DNA damage, and we could demonstrate that infection with the pathogenic bacteria would worsen the state of DNA, increase the DNA damage, thereby promote the oncogenic state of the DNA cell. Similarly, we also look, this is just a quantitative estimation of what is being shown in the previous slide to indicate that the clinical strain which we have isolated and when we compare to the standard ADC strain, there is a significant difference. In other words, there is a plasticity which occurs. That's why we always use a standard ADC strain as a control and then check their activity against the clinical strain to demonstrate that how functionally they are different because of the plasticity that exists between the bacteria, and we show that the clinical strain has more effect as opposed to the standard laboratory strain. And we also show in which we already save the fluorescent labels with bacteria with the fluorescent G, GFP in this case, and take them to the flow cytometry to estimate how much bacteria is invaded in the cells, and we can demonstrate Stenotrochomonas in this case that it invades inside the cells and it resides inside the tumor. And the DNA damage, as I mentioned, is one of the hallmark for the cancer progression, and this is a very bacterial Stenotrochomonas with respect to clinical strain as it was created strain is able to significantly bring down, significantly increase the DNA damage that would occur, which is detected in the form of the foci in the left-hand panel with the green dot which you see, as opposed to the control lactobacilli which does not give the same effect. We went on to sequence and Stenotrochomonas to understand what exactly is that which is making it benefit to the studies of the next slide, please. And then we have how the bacteria interact and get with the cells are bringing about arthroarticle changes, especially by modulating the function of actin filaments, which can be specifically detected with the specific pyruvate state, which shows that irregular active pyruvate state, suggesting that morphology of the cells are completely clear for its invasion, especially for invasion and metastasis. Next slide, please. There is also significance in the proliferative marker, where this bacteria was able to, Pseudomonas was able to activate number of genes which are responsible for proliferation of cells. Here and include, we have a normal tissue in which the activity can occur and which can stay latent for more than a decade, and due to the colonization of microbes, which may be pathogenic, which may be infectious, would change the population density and different type of bacteria given this climatic condition, and which leads to dismembering of the cells contact. In other words, there is a rupture which happens due to the dissolution of junction and captation complexes, leading to dysbiosis and inhabiting many more pathogenic microorganisms. And these dysbiosis keeps on intensifying, and they were promoting the tumor formation in situ women. Next slide, please. So we have been looking at it for many, many years now. Our team has done significant work in this work. But then what we learned just yesterday, last slide, that it can also come. You will be amazed at that. There are also articles, very interesting articles which appear in online as well as in the newspapers, Hindu and so on and so forth. If you have time to go through that, they have a mechanism called quorum sensing, where they can send other bacteria and they can make use of them to be able to cooperatively work along with the host to be able to promote the growth. You have a chance, so please do find the article and read it. Next slide, please. This is the team for whom we could not have conducted this work over the years, especially thanks to clinicians and PhD scholars who have spent significant amount of time in generating this work, and also to several funding agencies. Thank you very much. Thank you for your patience.
HDMI वर्क नहीं कर रहा.
Okay, no questions? I'm available. Sir, yes.
Is the blood cancer
Blood cancer.
curable?
Yes, so there are several types of blood cancer. One of them they were most successful in curing completely is the chronic myeloid. So there is a drug which is already in place, and that in fact happens because of transportation between the CRA gene, and the drug which has been done imaginary is called, which is able to kill this tumor cell by actually acting on BCR-able transported gene product. Very successful. Survival time is very high, 15, 20 years. Minimum residual disease is also very less. So that is one of the success stories which was happened during the turn of this century. Hodgkin's lymphoma, for example, not necessarily blood cancer. Hodgkin's lymphoma is another one which can be completely absolute. Non-Hodgkin's lymphoma is going to be difficult, but there are other types of leukemia like PALL or AML. They are a little bit harder. The efforts are on the way specifically to identify this. So they don't have a single mechanism unlike what my experience will become other. Thank you very much.
Very wonderful narration of
Thank you, sir.
from physical accounting. This regards SPV being the device. Vaccines are now available anti-HPV. In India, the campaign on vaccines started 2 years ago, I suppose, with the Zydus getting into the market. But I don't know the status now. Other areas become popular and more towards prevention. And at the same time, well, it has to be popularized also. And this is very much essential to prevent the PCOS problem. And at the same time, you will see the incidence of this rising HPV in the population, in Indian population.
So let me answer that two things. Right? So I don't see if I tell that look the vaccine works, then I have to close my lab and go home. So, that is just as a joke, right? But seriously speaking, as you rightly indicated, there are quite a bit of vaccine studies that have been done since about 2005, started with Glaxo, and now we have also from Serum Institute have bivalent HPV 16 and HPV 18 vaccines that have been now involved. Garrison from Glasgow is effective against 9 different types of HPV, and that is being now promoted in India. And there are very small incidences of non-compatible. People die because of the vaccine, but a very small number. This is also reported. We basically have issue with the vaccine because it's only given to adolescents, 16 years old, or maybe 20, 25, 30, women are years, and just one dose. Now, it has been very clear that if you take bivalent vaccine for 16 and 18, there are other dual high risk type viruses which are not, they start occupying. Really, you are able to eliminate this to your. Secondly, it is considered to be preventive, not preventive. It is preventive. So if you take care of two viruses, the other two viruses are growing much more rapidly. So we don't have anything right now to take care of all the 14 different. And there are plenty of reports on the positive, right? That the vaccine incidence of cervical cancer is reduced, number one, or it is prolonged. It's not related to about 10 years, now we are doing after 18 years. But we have not yet had time to see what is the outcome. Because it is only about 10 to 15 years. We really don't know what is the outcome. So we have to wait and watch. So the bottom line is, should one take HPV vaccine or not? That's to the person. Especially if the person is highly sexually active, better that the person takes it. If the person is not highly sexually active, the decision has to be done. A good force it comes to the women, saying that it is the vaccine you are taking. Not like when the kids we are being taking this vaccine, this vaccine, this is different ballgame. Thank you. Yes, ma'am. So, mobile, there has been quite a bit of studies since I'm not an expert, please, but whatever I have read in the scientific literature, whatever I remember, I will be able to tell you that Government of India has these habits. Government of India has funded many of these studies to be able to understand whether the mobile towers or mobile phones do have any ill effect on the body. And so far, the decision is kind of restricted, depending on how the experiment is done, outcomes, and it looks like in the majority of the cases, having the right person. So it doesn't have the, but if you have a continuous exposure for anything, you take any chemicals or you take any drugs, we are going to have ultrasound, waves, sunlight, solar light, whatever, anything you do in nature, it is bad for you. And mobile is not like cycle, and that's also. We have that, we have that. Yes. Definitely it is problem. Not help. Recommended that everybody should have, adults should have 60 or above should have 7 to 8 hours. The children should have at least for 7 to 10 hours sleep. Minimum across all age groups is 7 hours. They should have sleep, vocabulary, the stress increases, then you have higher cortisol level, and these cortisol are not good. And they end up in having more recently, any person you talk to young population, Gen Z, you call them, they have mental health problems. So mental health problems is becoming more and more prevalent. That is because they don't get enough sleep. And anything and everything they think it is a stress, not way of life. You think everything happens as a way of life, not stress, it is just not. But number of hours in mobile, number of hours of sleep are absolutely essential for young people to be able to do good in their life. Thank you.
Thank you, sir, for sparing your valuable time to share your knowledge with us. We are deeply honored by your presence, sir. Thank you. It is now time to express our gratitude through a small token of appreciation. I request Dr. Anil Kumar KM to come forward and felicitate our esteemed resource person, Dr. K. Satyendra. Good morning. I request Srimati Ramya to thank our chairperson, Dr. Anil Kumar, with a memento as a token of gratitude. Thank you, ma'am. I request Srimati Adya Hamid, Head and Assistant Professor, Department of Microbiology, to felicitate Srimati Ramya. Thank you, ma'am. Once again, we extend our sincere thanks to our distinguished resource person for the enlightening session and to our respected chairperson and co-chair for guiding this session so effectively. Thank you.
Ladies and gentlemen, there will be a short tea break for 15 minutes. So I would like to say the people who are participating in the poster presentation to submit your posters and be back to the venue by 12. Thank you.
Black and media. Lots of currency.
ನೋಡಿ ನಾನು ಅದನ್ನ ನೋಡೋದಕ್ಕೆ ಇದೆಲ್ಲ ಇರ್ತೀರಾ ಏನ್ ಅಂಕಲ್ ನೀವು
Show me my face now. Or else I'll send you another picture you edit that.
What picture you want to see? Canvas picture? It's good only, bro. Enough.
Because I got a good one.
We'll put that good only. What else? You sent to Anunya. Yeah. ಇರ್ಲಿ ಬಿಡು ಬ್ರೋ ಸಾಕು ನೀನು ಎಡಿಟ್ ಮಾಡೋಕ್ಕಾಗಲ್ಲ ನನಗೆ ಪ್ಲೀಸ್ ನನ್ಗೆ ಯಾಕೆ ಆ ಥರ ತಾಪತ್ರೆ ಆಗ್ಬಿಡ್ತಿದ್ದೆ ಎರಡು ಸಾಕು
I uncle bad.
ನಾನು ತಗೊಂಡೆ ಎರಡು ಮೂರು ದಿನ ತಲೆನೋವು ಬಂದಿದ್ದು ಮನೆ ಹೋಗಿ ತಲೆನೋವು ಮಾಡ್ಕೊಂಡು
Nice joke.
The HDMI cable is not working on my laptop. Yeah. Valid way. I am out of here by 4.
Now, if you don't ask question and drag it off, it will complete at 5:20.
ನಾನೇ ಅಂಕಲ್ ಮಾಡಿದ್ದು as soon as 20 minutes is done, you can file right now.
They should have kept some vendors in the participants. Or 6:30. 6:30. Because two chairpersons are going to speak. One chief guest is going to speak for valedictory, and our principal is going to speak. Principal is going to speak.
What PPT? Yeah, Payal ma'am told that sirs, I think sir will connect his laptop. I'm not sure of it, but then maybe we'll ask. I think HDMI is not working on mine. ಅದು ಕನೆಕ್ಟ್ ಆಗ್ತಿಲ್ಲ ಇನ್ನು ಮುರಿದೋಯ್ತು ಅಂತ ನನಗೆ ಅನಿಸ್ತಿದೆ ಒಂದ್ಸತಿ ಇನ್ನೊಂದ್ಸತಿ ಚೆಕ್ ಮಾಡಬೇಕು ಆಮೇಲೆ ನೋಡ್ತೀನಿ ಈಗ ನೋಡಲ್ಲ
ವಿಂಡೋ ಓಪನ್ ಮಾಡ್ದಾಗ
ಅದು ಬಂತು ಅದೇ ಈಗ ನೋಡ್ತೀನಿ
ವಿಂಡೋ ಓಪನ್ ಮಾಡಿ
ಸದ್ಯಕ್ಕೆ ಇದ್ದಿದೆ ಇವ್ರು ಕೊಡ್ತಾರ better I don't one correct Wi-Fi we need bro. It is should I just download the PPT on my laptop? It is not working. ಸುಮ್ಮನೆ ಕುತ್ಕೊಂಡಿದ್ದಾನೆ ದುಡ್ಡು ಮಾಡ್ತಾ ಇರ್ತೀವಿ ವೈಫೈ ಕೊಡಿ ಅಂತ I look studious now. I'm studious. I'm not. Yeah.
Who downloaded source? Are you sure?
Yeah. I downloaded, I saved, I saved. What bro?
There are more.
ಹಿಡ್ಕೊಂಡು ಹೊಡಿತೀನಿ ನಿಮಗೆ They put patent rights, but the checks are not on me. ಯಾರ್ದಿದೆ ಇದು A35 ಅಂತ ಯಾರ್ಯಾರ್ದು ಫೋನ್ ಕನೆಕ್ಟ್ ಆಗ್ತದೆ
Password ಬೇಕಲ್ಲ.
It is connected. Everybody's Wi-Fi is on my laptop.
No.
I will ask the password about I want password free Wi-Fi.
No, I was.
ಅಲ್ಲಿಗೆ ಆಗ್ತಿಲ್ಲ ಇಲ್ಲಿ ಹತ್ತು ಸತಿ ಇದು ಡಿಸ್ಕನೆಕ್ಟ್ ಆಗ್ತಾ ಇದೆ ಇನ್ನೂ ಚೇಂಜ್ ಮಾಡಿಲ್ಲ ನಾನು ಇನ್ನೆಷ್ಟು ಸರಿ ಹೇಳಬೇಕು ನಿಮಗೆ ಅಂತ ಕೇಳಿದ್ರು My move me download and then he comes and he says you have to edit edit ಮಾಡಬೇಕು you shut up please Wi-Fi is there some Nidhi's is there tell her thank you on my behalf I thought we have someone's Wi-Fi with us too yeah she came and said it thank you so much dear shut up ಹಿಡ್ಕೊಂಡು ಹೊಡೆಯೋಣ ಅಂತ you get up Here in Arundel. You come with me, no? We'll go to Niral sir and ask for Wi-Fi. ನೀನ್ ಮಾತಾಡ್ಬೇಡ ನಾನೇ ಮಾತಾಡ್ತೀನಿ ತಾಯಿ Come. This is who's
Please put up your order in the order. Process answer, please pay your order in the order. According to your phone number, please pay your order.
password change ಮಾಡ್ಬೇಕು. Why? Why? I cannot do that. ಏನೋ ಒಂದು ಕನೆಕ್ಟ್ ಆಗಿರ್ಬೇಕು. ನಾನು ಅದನ್ನ ಕೂಡ ಕನೆಕ್ಟ್ ಮಾಡ್ತಿರಕ್ಕೆ ಅದೊಂದು ಕನೆಕ್ಟ್ ಆಗ್ತಾ ಇಲ್ಲ. That's not the only ಒಂದು ಹಾಕಿಟ್ರೆ ಇನ್ನೊಂದು ಹಾಕ್ಬೇಡಿ ಅಂತಾರೆ. ನಾನು ಕಾಲ್ ಮಾಡಿದ್ದೆ ಅವನಿಗೆ ಒಂದ್ಸತಿ ಎತ್ತಾನೆ ಇಲ್ಲ. ಒಂದ್ಸತಿ ನೀವು ಕಾಲ್ ಮಾಡಿ ನೋಡಿ ಅವ್ನು ಲೋನ್ ನಂಬರ್ ಅಂತ ಎತ್ತಾ ಇಲ್ವಾ ಅಂತ. ಈಗ ಎರಡು ಸತಿ ಕಾಲ್ ಮಾಡಿದೆ, ಬ್ರೇಕ್ ಇತ್ತಲ್ಲ. ಬೆಳಿಗ್ಗೆ ಒಂದ್ಸತಿ ಕಾಲ್ ಮಾಡಿದೆ, ಎತ್ತಾ ಇಲ್ಲ ಅವನು ಹಳ್ಳೆಲ್ಲ. ಹಾಂ ಸರಿ ಸರಿ. ನೀನು ಹೇಗುಂಟೀಗ? ಮಲ್ಕ ಸರಿ. ಎಷ್ಟು ಮಾಡಿ ಮೂಗಿಗೆ ಹಾಕ್ಸ್ಕೊ ಆಗಲಿ. ಇದು ಹಾಕ್ಸ್ಕೊಳ್ತೇನೆ ರೆಗ್ಯುಲರ್ ಆಗಿ, ಅದಕ್ಕೆ ಎಲ್ಲ ಶುರು ಆಗ್ತಿರೋದು. ಸರಿ ರೆಸ್ಟ್ ಮಾಡಿ. ಇವತ್ತಿತ್ತು ಉಪ್ಪಿಟ್ಟೆ ತಿಂದೆ. ಹಾಂ ಜಾಗ್ರತೆ. ಪಾಸ್ವರ್ಡ್ ಸೇಮ್ ಇದೆ ಬಟ್ ಕನೆಕ್ಟ್ ಆಗ್ತಿಲ್ಲ. ಅವ್ರೇನು ಮಾಡ್ತಾರೆ, ಪ್ರಾಬ್ಲಮ್ ಏನು ಗೊತ್ತಾ, ಅವ್ರು ಏನು ಮಾಡ್ತಾರೆ, ನಮ್ದು ಇದು ಇರುತ್ತೆ ಅಲ್ಲಿ ಪ್ರಾಬ್ಲಮ್ ಇರುತ್ತೆ. ವಿಂಡೋ ಹತ್ರ ಅವರು ವೈಫೈ ಅದು ಸಿಗಲ್ಲ. ಕನೆಕ್ಟ್ ಮಾಡೋಕೆ ಆಗಲಿ ಟ್ರೈ ಮಾಡ್ತೀವಿ, ಕನೆಕ್ಟ್ ಆಗ್ತಿಲ್ಲ. ಅದಕ್ಕೆ ಯಾರ್ದೋ ಫೋನ್ ಕನೆಕ್ಟ್ ಆಗ್ತದೆ ಅಂತ ಬಿಟ್ಬಿಟ್ಟಿದ್ದೀನಿ. ಎಲ್ಲರ ಫೋನ್ ಕನೆಕ್ಷನ್ ಇಲ್ಲೇ ಇದೆ ಈಗ. ಅದು ಆಟೋಮ್ಯಾಟಿಕ್ಲಿ ಕನೆಕ್ಟ್ ಆಗ್ತಿತ್ತು, ಅದಕ್ಕೆ ಬಿಟ್ಬಿಟ್ಟೆ ಇರಲಿ ಅಂತ. ಏನಿಲ್ಲ, ಬರೀ ಕ್ಯಾನ್ವಾ ಮಾತ್ರ ಸದ್ಯಕ್ಕೆ ಸಾಕು. ಕ್ಯಾನ್ವಾ ಇವಾಗ ಓಪನ್ ಮಾಡ್ಕೊಳ್ತೀನಿ. ಅವ್ರು PPT ಒಂದು ಓಪನ್ ಮಾಡಿದ್ರು ಅಷ್ಟೇ. ಥ್ಯಾಂಕ್ ಯು ಸೋ ಮಚ್. ಇಲ್ಲಿಗಲ್ಲ, ವೈಫೈ ಕೊಡ್ತಿರಲ್ಲ.
ನಾನು ಕೊಟ್ಟಿದ್ದೀನಿ ಎಲ್ಲ ಕೊಡ್ತೀನಿ.
ಆ್ಯಕ್ಚುಲಿ ಟೇಕ್ ನನ್ನ ಪ್ಯಾಕ್ ಎಕ್ಸ್ಪೈರ್ ಆಗಿದೆ. 18 ಸೆಪ್ಟೆಂಬರ್ ಬರಲ್ಲ. ನಾನು ಮಮ್ಮಲ್ಲಿ ಐ ಹ್ಯಾವ್ ಅನ್ಲಿಮಿಟೆಡ್ ಪ್ಯೂರ್ಲಿ ಐ ಟೇಕ್, ಬಟ್ ದಿಸ್ ಟೈಮ್ ಇಸ್ ಫಿನಿಶ್ಡ್. ನಾನು ಆಲ್ರೆಡಿ 18 ಸೆಪ್ಟೆಂಬರ್ ಆಗಿ ಮತ್ತೆ ಫುಲ್ ಅನ್ಲಿಮಿಟೆಡ್. ಸೊ ಲೈಕ್ ಏನು ಹಾಕ್ಸಲ್ಲ ಬಿಡೋಣ. We'll do one thing with take ahead, go ahead with this only. Only PPT ಅದು ಕರೆಕ್ಟೇ, ಬಟ್ ಇದರಲ್ಲಿ ಕನೆಕ್ಷನ್ ನೆಟ್ ಇದಿದೆ, ಎಥರ್ನೆಟ್ ಇದೆ. ನಂದ್ರಲ್ಲಿ ಎಥರ್ನೆಟ್ ಹಾಕೋಕ್ಕಾಗಲ್ಲ. ಇವ್ರು ಎಥರ್ನೆಟ್ ಕನೆಕ್ಟ್ ಮಾಡ್ಕೊಂಡಿದ್ದೀನಿ. ಎಥರ್ನೆಟ್ ಕನ್ವರ್ಟರ್ ಬೇಕು ಬ್ರೋ. ನಂತರ ಎಥರ್ನೆಟ್ ಕನ್ವರ್ಟರ್ ಇದೆ. I have which camera, but it is dirty as fuck. Nice bro, ಚೆನ್ನಾಗಿದೆ. ಅದಕ್ಕೆ ಕೂದಲು ಕಟ್ಕೊಂಡಿದ್ಯಾ, ಅದಕ್ಕೆ ಹೌದು. ಯಾರ್ದೋ ಕನೆಕ್ಟ್ ಆಗಿದೆ ಒಟ್ಟಿನಲ್ಲಿ.
Dr. Payal Roy, Chairperson Dr. Sidney Gaurapriya, and hosted person Srimati Adya Kami to occupy the dialogue. I'm delighted to welcome our respected resource persons, Dr. Payal Roy, Assistant Professor, Department of Biochemistry, Indian Institute of Science, Bangalore. We welcome you, ma'am. I request Chairperson Sidney Gaurapriya, Professor, Department of Microbiology, to welcome our resource person, Dr. Payal Roy, slowly. Thank you, sir. I also welcome the chairperson, Sidney Gaurapriya, Professor, Department of Microbiology, to welcome your I request co-chair person Adya Kani ma'am, Assistant Professor, Department of Microbiology, to welcome the chairperson Siddhagowda sir closure. I would like to extend a warm welcome to our co-chair person Srimati Adya Kani, Assistant Professor and Head Department of Microbiology. We welcome you ma'am. I request Srimati Kandan, Assistant Professor, Department of Biochemistry, to felicitate Adya ma'am closure. Thank you, ma'am. Now I call upon Tahari of Second BDPM to formally introduce our esteemed chairperson, Dr. Sidney Gaurapriya. Good afternoon, everybody. It's an honor to introduce our esteemed guest, Dr. Sidney Gaurapriya. Dr. Bhodaya Srimati Gaurapriya, Professor and Head of the Undergraduate Department of Microbiology, also at the Government Science College in Hyderabad. Professor Gordon 20 years of teaching experience and over 7 years of R&D experience in areas related to biomedical and microbiological aspects of it. In addition, he has also industrial experience close to 22 years. His noteworthy contribution in the field of determination of animal processing byproducts, especially from fish processing, to biotechnological approaches, and utilization of underutilized marine resources. He is a recipient of teacher fellowship from the University Grant Commission and the Best Poster Award Prize for his research accomplishment in the area of shell technology. He has granted short-term studies from the Karnataka Science and Technology Academy, Government of Karnataka. He has been bestowed with the Best Scientist Paper Award 2023 by the Society of Fisheries Technologists in India for the research article for the fishery technology. Dr. DSS has published 30 research papers in peer-reviewed international journals with significantly high impact factors and has presented over 20 research papers at national and international conferences. He has delivered numerous special lectures on food hygiene and safety, health and microbes, and environmental solutions in many academic institutes. He is a vice member of various professional bodies such as AMI, and KSCI. He has been instrumental in framing the syllabus of ADSC 2018 to 2019, NC 2020 to 2024, and SCP 2023 to 2025. For the undergraduate microbiology of the University of Texas and undergraduate affiliated with the microbiology. Recently, the Association of Food Scientists and Technology India founded Dr. Madam Joanna and Food Science and Technology. Best paper award 2024 in food technology during ICFO in 2025 held on system control center. Thank you, sir. It's my pleasure to have you here. Thank you, Sally, for your introduction. I request Dr. Sudhir Gaurav to introduce our esteemed resource person, Dr. Payal Gaurav, Assistant Professor, Department of Biochemistry, Indian Institute of Science, Bangalore, for the session.
Yes.
I thank the organizers for giving me an opportunity to share technical session on introductory knowledge. I'm very much delighted to introduce the speaker of the technical session, Dr. Bayer Roy. Dr. Bayer Roy is an assistant professor at the Biochemistry Department at Indian Institute of Science, Bengaluru. He obtained an integrated BSc MSc degree in biotechnology from St. Eliot's College, Calcutta University, India.
ಇನ್ನು ಮೈಕ್ ಇತ್ತಲ್ಲ ನಾರ್ಮಲ್ ವರ್ಕ್ ಆಗೋದು ಆ ಮೈಕಲ್ಲಿ ಒಂದ್ಸತಿ ಇವರು ಸೆಷನ್ ಶುರು ಆದಮೇಲೆ ಆ ಮೈಕ್ನ ಈ ಮೈಕಿಗೆ ರಿಪ್ಲೇಸ್ ಮಾಡು ಆ ಮೈಕ್ ಮೋಸ್ಟ್ ಪ್ರಾಬ್ಲಿ ಕರೆಕ್ಟಾಗಿ ವರ್ಕ್ ಆಗಲ್ಲ ನೋಡಿ ಯಾವ ಸರಿ ಯಾವ್ದು ಸರಿ ನೋಡಿ ರಿಪ್ಲೇಸ್ ಮಾಡಿ
molecular biology, biochemistry, computational and behavioral approaches, and the mechanistic basis of malignant mutations in non-canonical NFKB pathway and drug resistance in human multiple myeloma, a digital cancer. To further improve our knowledge and expertise in understanding the role of human cells in human disease, he joined the Hoya Institute for Immunology, San Diego, USA, for a postdoctoral training with Dr. Lars Lee. She worked under his mentorship for more than 7 years, first as a postdoctoral researcher in the Hoya Institute for Immunology, and then as a junior faculty at the Immunology Center of Georgia, and new research center co-funded by Dr. Lars Lee and Dr. Lee. During her postdoctoral tenure, her work focused on understanding how immune cells can modulate disease progression and outcomes in atherosclerosis, a pathological condition that underlies most cardiovascular diseases. By combining immunological assay with next generation sequencing techniques, Dr. Rai identified the signatures of dysfunctional immune regulatory T cells and operantly activated autoreactive CD4 T cells that correlated with severity of coronary artery disease in patients. These studies have yielded important signatures that can serve as valuable tools to probe the functional dynamics of T cells that show disease-associated activations. Her overall training provides her with an excellent background in multiple biological disciplines, including basic and clinical immunology, sequencing techniques, molecular biology, biochemistry, signaling, genetics, and computational analysis. She is currently focused on understanding the triggers and mechanisms that lead to maladaptive immune cell activation and their disease-related functional adaptation in the context of chronic inflammation and dietary imbalance. A special emphasis of our lab is to delineate the antigenic targets, phenotype, and the dynamics of autoreactive T cells in metabolic and chronic inflammatory diseases in humans. Thank you. So, with this great introduction, I welcome Firefly. I request that to take over the session. Thank you.
Can you hear me? Okay, thank you very much for the kind introduction. And thank you to the organizers for inviting me here. So my topic is introductory immunology, so I'll try to keep it as simple as possible. I know I stand between lunch and this lecture, so bear with me for the next 20 minutes or so, okay?
Everybody here are familiar with the immune system, and the immune system is our body's defense mechanism. And defense is something which is very, very fundamental. It's something that we try to keep out from anything that's foreign. If you take your college, for instance, you will have a physical barrier like say the boundary wall that will ensure that nobody from outside can enter. And similarly, you will have some security guards who will stop anybody who doesn't have an ID card to enter or any permission to enter the college premises. It's the exact same thing that happens with the immune system to keep out invaders from entering our body, and even if they do enter, then there is an active mechanism that tries to clear them away. Now, this is what keeps us protected. The inflammation is the war that takes place. So the fighting process is what is known as inflammation. And if you see the graph here, the blue graph, so ideally, once the danger is eliminated, the war should stop. Then a new set of immune cells, which are the regulators and not the fighters, they should be called in so that the fighting stops and the damages are repaired. So this is the fundamental basis of having a protective response, but also controlling that protective fighting so that damages are repaired and body returns to what we call as homeostasis, this normal condition. Next slide. Now, as I mentioned that regulatory cells is a concept that we often overlook because immune cells, we think that their only job is to fight. We forget that fighting is good as long as it is well timed. So there is a special set of cells called regulatory cells, and this year the Nobel Prize in Medicine was given to this team of researchers who had discovered regulatory immune cells. Okay? So I wanted to highlight in this talk that whenever we talk about immune cells, we mostly think about the fighting part. We don't think about the stopping part. But that stopping is an essential mechanism which is a highlight of what I do in my lab. Okay? So, as we all know that the immune system is a very powerful system. Anybody that has the potential to kill, damage, is anybody with a weapon has a lot of power. And that gives them the ability to protect, but also they can cause damage. So protection, of course, they protect us against bacteria, viruses, parasites, fungi, toxins, pollutants, pathogens. We know about all of that. But if they keep fighting too much or unnecessarily, that's when you get allergy, autoimmunity, asthma, and many other inflammatory diseases. In fact, during COVID, one of the major reasons that people were dying was not always because they were not able to fight, but also the immune system was fighting either too much or inappropriately. So therefore, current research is focused on boosting the protective functions and blocking the damaging effects of the immune system. So this is what my lab is focused on, the changing phases of the immune system, which is called reprogramming. Guys who should be doing a good job trying to control, regulate, are switching and acquiring a fighting phenotype. So today's talk, I'll give a general introduction to immune cells and maybe briefly mention what my lab does. So the immune system, it's spread out throughout our body. There are many different organs that are involved in the immune system. So this is a very unique property because, say for example, a skin cell, it is born here, then it dies here. But immune cells are mobile. Their birthplace, their place of training, and their place of activation were all very unique. Like, say, for example, I was born in Kolkata, then I went to Delhi for some training, then I came back and I'm doing a job in Bangalore. So I've been moving around. And when you move around, you acquire properties born with, but also what the microenvironment teaches you to be. So if you look here, the bone marrow, the tonsils, the thymus, you can see it is spread all across the body's organs, which are involved in either making the immune cells, or training the immune cells, or activating the immune cells. So this is a typical profile of some of the well-known immune cells that you will come across. So they are the granulocytes, T cells, B cells, and their frequencies in the blood have been well characterized, and there's always a normal number or frequency of these immune cells that you can detect in blood, and often their changes in these numbers or their phenotypes is what the doctors use to diagnose an underlying condition. Okay? So as I mentioned that the immune cells are born as very distinct things. So immune cells are found in the blood. If you may have heard of RBCs and WBCs and platelets. So RBCs is the reason why our blood is red. WBC is what the immune cell compartment comes from. Okay? So this is a very well-programmed system known as hematopoiesis, and that is what results in the formation of all the type of blood cells that you get. And these are very distinct programs that you have progenitor cells going into a distinct cell type. So you have the red blood cells, the immune cells, and then as you can see that immune cells have different properties. So two subtypes that we will be talking about are the innate immune cells and the adaptive immune cells. So once the pathogen is trying to enter our body, the first thing that our body does, there are certain barriers. So say for example, skin serves as a physical barrier. So we don't let the pathogen enter. That's the first step. Or there can be some chemical barriers like saliva has some enzymes that can evade the pathogens. So, but if these barriers are breached, then the pathogen does enter, and the first line of defense, the cellular defense that fights against this pathogen, they are known as the innate immune cells. So the innate immune cells, they find the pathogen and they mount the initial attack. After that, if the innate immune cells are able to stop and clear the pathogen, very good, it's done. But if they're not able to take care of it, then they trigger the next layer of defense, which is known as the adaptive immune cells. And the adaptive immune cells, they are the two major subtypes that are very well known to us, the T cells and the B cells. You may have heard of antibodies. Antibodies come from the B cells. So these are cell types that are very specific for certain kind of pathogen. So say for example, when we get a vaccine, our major focus when we get a vaccine is to boost this adaptive part of the immune system because this is specific and it remembers. So if you have a polio vaccine, you will be immune to polio but not to dengue. But if you have a vaccine against influenza, you will be immune to influenza but not to COVID. So this is the whole concept that you have a specific activation and you would remember it. So the immune system has a memory of it. So this is the outline of the differences. As I mentioned that innate immunity is quick because it's non-specific. It tries to identify certain patterns and that's how it tries to stop the invading pathogen. But it does not have any memory of it. And it is found in nearly all forms of life because defense is a very fundamental concept and every form of life has some degree of defense. And in innate immunity being the most general kind of defense, even lower forms of life, they have this innate immune system active in them. On the other hand, adaptive is a very specialized form of immune response and you find it only in vertebrates. So from jawed fish and above, that's where you have the adaptive immune system active. And so this, as I mentioned, is pathogen and antigen specific. So what do I mean by pathogen and antigen? So pathogen is, say for example, influenza is a pathogen. And a particular protein in the pathogen, in influenza, that is triggering the response, we call that an antigen. And that is the fundamental basis of how vaccines work. The next thing is there is a lag time between the exposure and the maximal response because it's a training system. You are being taught to only fight against one specific thing, and that training takes time. And the third thing, as I mentioned, is memory of it. The memory makes sure that next time you see the same pathogen, it is very quick. Okay? So the main reason that innate is not specific and quick is because of the way it recognizes the pathogen. So it recognizes certain broad patterns. Say for example, you may have done gram staining before, right? So gram staining, a gram positive bacteria and gram negative bacteria, the wall that it has is different in composition. So gram positive will have more bacteria active. Gram negative will have more lipopolysaccharide. So this is what the innate immune cells recognize. The chemical pattern that exists in a bacteria or virus. It can be a DNA pattern, it can be RNA pattern, it can be their capsule layer as I mentioned. So it does discriminate between one kind of bacteria versus another or fungi versus bacteria, but it is not specific for only one species of bacteria. So that way it has a broad spectrum of identification. So this is known as pathogen associated molecular patterns. This is called PAMs. These are chemical structures in the pathogen. On the other hand, you can also have damage-associated molecular patterns. This can be nucleic acid structures, this can be certain chemicals like cholesterol or ATP that can trigger a sense that the body is under stress. So these are called danger-associated molecular patterns. And the receptor on the innate immune cell that recognizes these patterns are known as pattern recognition molecules, which are the PRRs. So you can have a family of innate immune cells which recognizes a series of these pattern recognition receptors, helping them identify the spans or dams in the system. One of the most classical examples of a pattern recognition receptor is a toll-like receptor. Toll-like because tolls were discovered in Drosophila, and when they were discovered in mammals, they were named as toll-like receptors. So toll-like receptors, as you can see, there can be many different varieties of it, and interestingly, they can function as a homodimer or an heterodimer, making them more diverse. So depending on what their receptor, the extracellular domain is, recognition ability for a pathogen pattern changes. And you can have them on the surface, as you can see that there are some surface toll-like receptors that helps them to detect a pathogen which is outside. Say for example, bacteria. But imagine a virus. A virus, the only time a virus is alive is when it is inside the cell. So it has already entered the cell. Then the cell must be having a recognition receptor inside to be able to detect a virus that has been entering into the cell. So that is why, as you can see, there are some endosomal toll-like receptors that help to detect viral RNA or say CTG-guided DNA. So these are specific for different kinds of bacteria or virus. So this is an example of receptors in the innate immune family. So next we come to the adaptive immune system, and this is the main focus of the research that I do. This is on the left you see a B cell, and on the right you see a T cell. These are the most famous adaptive immune cells. So they have receptors which are specific for a unique protein that only a particular bacteria or virus will be having. So if you see the structure, there is an area which is written as BCR recognition or TCR recognition. So this recognition is like a lock and key, and only a specific key will fit a specific lock. So this specificity is the fundamental basis for how an adaptive immune system is specific and has a memory. So how this works? So the way it works is, so let's take an example. So say for example, we have a bunch of keys to all the doors that will open every door in this building. Okay? So you want to open the door to this particular room. You need a key that will open only this room. But you have all the keys. So if you have 100 keys, you will have to one by one try every key to see find the one that is fitting this particular room. And that is the entire concept. The T cell receptors or B cell receptors are made with a huge amount of diversity, and once a pathogen comes, it screens for all the potential receptors that can bind the pathogen of interest. Once it does, then the receptor that is recognizing that particular antigen, that cell proliferates. And when that cell proliferates, then in the population, you will see that the T cell or B cell having a receptor against antigen X is growing in number. And that is what you see. You have a mix of many different cell types, and the red cell is specific for antigen X. It saw antigen X that gave it the signal that the selection process should occur. That red cell now expands and gives rise to many other red cells, creating an army of immune cells that can now fight any pathogen that has antigen X. So the next slide, the next figure shows the basic concept of vaccination. So in vaccination, you may have heard that initially there is a priming dose, and then you get multiple booster doses. So what this booster doses does is helps the immune cell remember what it had seen during the priming dose. So this antigen X that I mentioned, so when you have a priming, the immune cell against antigen X that grew in number. It is now red. Next time you have a booster that will give a peak of response. On the other hand, if you now introduce another antigen Y, which the immune system has not seen before, then it will again have to go through the whole cycle of screening for which receptor will be able to bind antigen Y. So this is the whole concept of the primary response and a secondary response and how vaccines work. So now, this is what we try to understand is the T cell receptor, and what is it that the T cell receptor is seen as an antigen in the context of different diseases. Now, we work mostly on T cells, and T cells need help from innate immune cells to tell them that there is an antigen that requires recognition by a T cell receptor. Now, in this case, one thing you have to remember is two things are very, very important. One is that there has to be a tri-molecular complex of the innate immune cell, of the antigen, and the T cell receptor. This has to happen. And the second thing, you have to make make sure that this antigen cannot be a cell protein. Because if it is a cell protein, then your body's immune cell will start fighting against your body's components. So these are two things that the body tries to ensure. So, as I mentioned, the first thing will be positive selection. As you can see that three cells have been crossed out. The reason those three cells have been crossed out is because the tri-molecular complex could not be formed by these three cells. So the body eliminates these cells that could not form a tri-molecular complex. The next which you can see is a negative selection. If you see here, one is crossed out. The reason being it was binding a cell protein. So this is something that the body tries to eliminate. Again, I'm saying tries because it is not 100% accurate, and because it's not 100% accurate, we get diseases. So as I mentioned that there are distinct areas that do different things for the immune system. The bone marrow is where the immune cells were formed. And the staining process that I said, positive selection, negative selection, this is the school for the T cells, and that happens in the thymus. And next we will see where the activation takes place. So the activation happens in secondary lymphoid organs, which can be spleen, lymph nodes, and one of the very famous examples which you often notice are the tonsils. So when we get a throat infection, the first thing that happens is that tonsils get swollen, and that is a very clear indication that our immune cells are getting activated and they are trying to fight. So you may have heard of the circulatory system, which has the heart and the heart pumping blood all across our body. But I don't know whether you're aware of the lymphatic system. So in parallel to the blood circulatory system, there is another system called the lymphatic system. Because your blood is not entering your tissues directly. So it's the fluid part that extravasates from the blood and goes into your tissue. That is what makes lymph. Because we know that the tissue is sitting there isolated from the blood, but it requires nutrients, it requires oxygen, it requires different kinds of fluids. That is not supplied directly by the blood, it is supplied by the lymphatic system. And this lymphatic is what the immune cells use as a traffic mechanism to move in and out of the blood to the tissue and back from the tissue to the blood. So, as you can see here, the lymph nodes is the unique area which is specialized for the immune cells to detect these pathogens that enter. They have structured areas that make them convenient for this fight.
There's no time limit now. Yeah.
So this is what the structure looks like. It's like a mesh work where the immune cells are ready and waiting to trap the antigens when they enter. So this is, as you can see here, those are the pathogens that are coming in, and the immune cells are waiting to trap them and undergo activation. Once the lymph nodes help to activate immune cells, then they are ready to supply activated immune cells to go and fight at the tissue area. Okay? So you can see on in the figure there that there is kind of a roadmap where the immune cells can move around between the heart and the different tissue areas and the lymph nodes. So what they do is they use the blood as a roadway to move across our body. And this gives us a very unique opportunity to try to study immune cells in humans. The reason being is if we want to know what particular tissue, what kind of immune response is going on. It's not very easy for us to get access to the tissue sample from a human being. But the blood is where the roadway is, and those immune cells that have exited out of the tissue are moving through the blood. So that is the fundamental thing that we are trying to do in the lab. We are trying to use human blood as our resource to study immune cells to give us an indirect reflection of what is going on at the tissue. So this is kind of the flavor of what we're doing, that there can be an imbalance between the fighter cells and the healer regulatory cells. And this imbalance is what gives rise to an inflammatory process, which is a warlike state that is not resolved. And this is now being proposed to be involved in many, many different diseases. So, and this, in my opinion, is a very modern problem. We now live a very long life, but all throughout that long, unhealthy life, we are exposed to different kinds of environmental, chemical, or dietary stresses, including neurological stresses. And all of this are now being implicated as major triggers for immune cell reprogramming. And this is the scheme of what we try to do. We try to, and that's the kind of work that I have done during my postdoctoral studies, is we have people who are in different gradation of diseases, either no disease, intermediate severity, and severe disease. We try to get blood from these different groups of people. We look at their immune cells using different kinds of techniques, and we try to get an understanding of what are these immune cells trying to tell us about the disease state. And we specifically look for the dysfunctional immune cells. Because they're very rare, we use very high resolution assays so that we can detect them even when they are very low in number. Because the hypothesis is if you can detect diseases early on, then there can still be some potential to inhibit the developmental process of the disease. And the way we try to do it is by trying to find these rare dysfunctional immune cells in human blood. What we try to do, as I said, we try to look for the antigens and other triggers that could be activating these criminal immune cells, and then we try to measure their properties to try to see whether are they fighter-like or are they healer-like. And depending on what disease you're studying, you would either need the fighter-like or the regulatory-like. That's the flavor we try to determine. We use, as was mentioned, different kinds of tools and techniques. It could be the human system. Sometimes we have to use model systems like mice. We use computational tools, and we use some sequencing techniques, as well as different kinds of biochemical and immunological assays. So that's kind of the crux of what we do, and if anybody's interested, I will be happy to explain more. So that's the premise of what I do is we know that there is a failure in the regulatory mechanism in immune cells, and that leads to chronic diseases in humans. But what I'm trying to understand is why they fail and what are the different triggers, how does that control disease progression, and most importantly, which pathways can be targeted to restore them. So that's the crux of what I do or what I want to do in the future. That's it.
Now the session is open for Q&A. Can you please, any queries related to the concept on immunology, please come.
Thank you very much for organizing this.
While immunize us with the concept of unity. Because many a time we simply think about how the disease will appear in our system and how exactly our immune system can fight back the same. But this is the story behind the same. How exactly our system works against the pathogenic microorganisms, which are all the components which play a major role in providing the immunity to our system. So, thank you very much for input and we need to take the concept on the complex things. Thank you very much.
Thank you, ma'am, for sparing your valuable time to share your knowledge with us. Now, it is now time to express our gratitude through a small token of appreciation. I request Chairperson Dr. Siddhe Gowda to come forward and felicitate our esteemed resource person Dr. Payal Rao with a memento as token of gratitude for this session. Thank you, sir. I request co-chairperson Srimati Adyasamy MD to thank our chairperson Dr. Siddhe Gowda GS with a memento as a token of gratitude. Thank you, ma'am. I request Srimati Meghana M. Ed and Assistant Professor Department of Biochemistry to felicitate Adyasamy ma'am. Once again, we extend our sincere thanks to our distinguished resource person for the enlightening session and to our respected chairperson and co-chairperson for guiding this session so effectively. Thank you. I request our resource person, Dr. V. Nagaraja, the chairperson, Dr. Sardar Hussain, and the co-chairperson, Srimati Meghana M. to occupy the dais.
Will it go? No.
No, sir.
Then what will you do?
Is your PPT on the pen drive, maybe?
You may have sometimes. This, I can't do. No, this laptop doesn't work that well. You don't have adapters. But you didn't tell me. I thought it was after the relaxing. Yeah, it's okay. You.
Yeah. Extremely sorry, sir, for the
I would have already
inconvenience.
loaded. No, you should.
Oh, yeah, yeah.
I mean, what is the point?
Which one is it, sir?
Huh? Yours. No, but I know what. I know what the it can be there. No, no. This is your. ಯಾರು ಹೇಳಿಲ್ಲ ನೀವು ಯಾರು ಹೇಳಿಲ್ಲ
Is it a file you put it on, maybe?
No, it is the PPT. Wait. You know, wait, it's coming. They are using no time, 50 minutes and all. They have not seen the program. You're students, you should be. Email. NMK. Yeah, ಅದೇ ಮಾಡಿ. Okay. I'll share that with you. Share ಮಾಡೋಣ. ನಾನು ಎತ್ಕೊಳ್ತೀನಿ. Share ಮಾಡೋಣ. Caller ID. Caller ID. Caller ID.
Call your mic, sir.
ನಮ್ಮ ಕಂಪನಿ ಇದೆ ನಮ್ಮ ಪೆನ್ ಡ್ರೈವ್ ಮರಿಬೇಡಿ ಇವತ್ತು ಮ್ಯಾನ್ಯೂ
ಅಪ್ಪ ಮಾತಾಡ್ಬೇಕು, ನಾನು ಮಾತಾಡ್ತೀನಿ. ಇಲ್ಲಿ ಹಾಕಿ ಹಿಂದೆ ಪಿನ್ ಮಾಡಲಿಕ್ಕೆ ಹೊಡಿ. ಅಭಿಷೇಕ್ ಸರ್, ಅಭಿಷೇಕ್ ಸರ್.
Good afternoon. Good all. I'm privileged and blessed to introduce the speaker of this session, Professor V. Nagaraja. Professor V. Nagaraja is a distinguished microbiologist and professor in the Department of Microbiology and Cell Biology at the Indian Institute of Science, Bangalore. He had his graduation and post-graduation from Bangalore University. He does his PhD from Indian Institute of Science in the year 1981, where his work on Mycobacterium 13 and ESC Gyrus on mycobacteria laid the foundation for his research. He worked as a research associate at the University of Basel, Switzerland during the year 1981-85 and at the University of Manchester, USA. Before joining ISC in 1988-89 as professor and contributing to the establishment of the Center for Genetic Engineering. He later became associate professor, and also has served as a chairperson for the department. In 2015, he was appointed as president for the Jawaharlal Nehru Center for Advanced Scientific Research. His research focuses on immunoisomerization and the molecular biology of mycobacterial tuberculosis, leading to the significant contributions in biotechnology, including the development of restricted enzymes and potential therapeutics for tuberculosis, and he holds international
international patents. He has guided over 45 students and mentored numerous researchers. He has also contributed several research papers to national and international repute. He has also presented 600 presentations for various institutions. Professor Nagaraja is a fellow of INSA, IAS, NASI, and PWAS, and a recipient of several prestigious awards, including the Shanti Swarup Nagar Award given in 1999, Rantaxi Science Foundation Award 2003, JC Post Fellowship 2008, PWAS Prize in Value 2011, and Sir Vishweshwaraya's State Award for the Government of Karnataka. So we have so many credits to his profile. So with this brief introduction, I welcome you, sir, for the talk. And sir will be presenting a talk on how to tackle the resurgent drug resistant tuberculosis, which is a growing burden across the globe. I welcome you, sir, to this presentation. Thank you.
Day-2. Date of body. Yeah, yes, sir, sure. Last one, yeah. Shall have a phone.
Can you hear me in the back? Is it audible? Can you hear me in the back?
I think it's off, sir. Only one.
I think you are hungry. So come forward.
Give him this mic.
Mic is not working. Hello? Hello?
Yes, sir. Yeah, no.
Hello, can you hear me?
Yes, sir.
Yeah. Okay. So, good afternoon. I was very thankful and appreciative for the introduction. I was comfortably sitting in the chair and listening to clients very nice basic function of how defense system works in our system. They implemented that after lunch is quite long. So all this connecting and everything happened because I was not told that. Oh, I think in many ways it's better because after lunch many of you made honestly, including me. So it's good that I speak now. Having said that, the second thing I want to say is I should thank Professor College, Professor Satyamurthy, who is instrumental in making sure that I come here today. He tried me last night. I had some health issues, allergy, running nose, and back pain and whatnot. But the Satyamurthi is Satyamurthy. So I had to come. So also I always like to talk to students. So I don't like students hanging around, volunteer or otherwise. Sit down. If you can learn something, it's fine. Because this is, I know what overall my research work. It is important for you to learn. I am standing here, so is quiet, so is so other teachers. We stand here because we are students like you. We are students like you, we are scholars like you, and we have inspirational teachers. Because of that, we are standing here. And that's why it's important for you to learn, and not only learn, and there are people I have talked to with different things. You certainly learn something as you use when you take up the career as a teacher or a scientist or technocrat, whatever it is. So what I'm going to talk to you today
general idea, but I will not talk much of anything about research. Maybe it is passing mention. Otherwise, it's going more general. And there are possibly misconceptions about the most ancient disease of the mankind, one of the most dangerous diseases, but nevertheless still continues to be the most major disease troubling the mankind. A global number one infectious disease killer is tuberculosis, caused by the organism Mycobacterium tuberculosis, a pathogen found in Egyptian mummies. You know what are Egyptian mummies? 5,000 years ago, they, those mummies were buried in the ground, and in there you find the lesions, tuberculosis lesions. So it's a very ancient disease. So, but it still persists. Millions die, more than millions, several get infected. Still, so many people die. Our country, government, wanted to eradicate tuberculosis 2025. We people were working on this tuberculosis research, knew it was not possible. Now they have shifted the mandate to 2035. But I am not sure it will be achieved. So what I'm going to talk to you about is about the tuberculosis. Stay tuned. Of course, we have done research. On the left side are the students who did the work. There are actually more students. And then on the right side are the very important collaborators and the funding. You can move to the next slide. So this is a more recent pattern from a newspaper, Deccan Herald. That's the newspaper I read. I'm sure it comes different ones. It says India tops global TB burden again. Drug resistance is a hurdle. Personally, you end up seeing newspaper articles about this. During COVID, you didn't hear much about tuberculosis. Every viral disease, forget about COVID, every viral disease, you end up front line news all the time. Rightfully so, but tuberculosis is common at that time. But more people die of tuberculosis all the time. COVID, there was a search, now it's called COVID virus, that's happening, SARS and Delta variant, whatever it is, they have become endemic now. It is the population, like the flu. But tuberculosis still continues because it is one of the cleverest bacteria mankind has ever faced. And this co-evolved learn to live in the humans and with the other animals. So let's move next slide, please. If I show the hand like this, you are not bad. So you are an ancient area of mankind, which I said, you are a pointer business?
No, sir.
Okay, you are a long stick, not use human. That no? As said, it is one of the ancient diseases. But the problem, boys and girls, it is not just a tuberculosis disease. Currently, we have drug-resistant tuberculosis. I will just tell you in a few minutes what are the drugs used for tuberculosis. Some of the front benches are on more about the drugs, but I will elaborate a little bit. So, what you have something called multiple drug-resistant MDR, name itself means it's resistant for certain amount of drugs. I will not do the definition, but if you ask me, I will give you. Then we have XDR, extensively drug resistant. And subsequently, few years ago in a hospital in Bombay, they called it TDRP, totally drug resistant. Means there is no drug for the TDR. And I will elaborate on this point. Next slide, please. Very tricky diagnostics. And I will show you slides 2. We always think, a lot of coffee and chest x-rays, but even chest x-rays, rest of the best doctors, they will not be able to identify TB, because sometimes the lesions do not show up. This is a fact. Diagnostics is a challenge. There has been an evolution in diagnostics. There are newer ways. Yet, childhood TB or disseminated TB, miliary TB is a challenge to detect even now. Let's know the best that is newer diagnostic was. That's a separate lecture if I have to go. And with that also, it is not going to, it's not 100%. At the most, we reach up to 80-85%. In fact, I am collaborating with the Danish group and the IIT Euro to detect TB on the saliva and whether to see whether we can make it better. So we can move and child TB rampant. In India, it's considered between 5 to 10% child TB. And then it's better. So if you are not waiting for the chest x-rays of that, you shouldn't be doing that anyway. Then you have next slide. And why I could say this is small, so I know a professor in English science. Yes, I work in English science. I'm officially retired, but not totally retired. Still, I have research lab. So this professor had a ear infection, very high fever and ear infection. So not getting cured with the regular drugs. Finally, doctors cured anti-TB drugs. Got cured ear infection. Yes, sir. And I give you another example. Bone marrow transplantation. You know when it is done. When you, after bone marrow transplantation, people come down with very high fever. When they come down with very high fever, treatment something doesn't work. Anti-TB drugs work. So the St. John's Hospital, young doctor came to me. I want to collaborate. I think this is a reactivation of TB. I will tell you what it is. I said, yes, fine. We set up a collaboration, but next week, after a month, we went up to US to got a fellowship. So that research never took place. So, but the idea of TB bacteria which is not able to sting is a fact. And TB bacteria hides in our body. It's called dormant TB. Very, very difficult to detect. I can go on. I don't want to take too much time, but like while I am standing before the lunch, I'll try to finish in next 45 minutes. But and you can ask any number of questions during lunch time. So basically, your first line is second line is then and then you have the one side of one population. TB, 9 million new cases, 1 million chronic new cases of active TB globally, and 2 million associated deaths. Million disseminated TB makes up 10% of extracontinental extremes. So, TB bacteria is very clever. Not just the lung infection what you see. Practically every organ is infected. I just talked about possibly ear TB. Look at here. It's a brain TB. Pulmonary is what you know. Bone TB, gastrointestinal TB, eye TB, lymph node TB, extracontinental TB. The ear TB can be anywhere. Skin to anything and anything. And then gentle TB. Any other organs. This one the representation. Next page. So, how do you treat TB? Normally, when you have infection, especially diarrhea or some other bacterial infection, you go to the doctor, they prescribe, depending on the doctor, either 5 days or 7 days antibiotics. One antibiotic, sometimes 2. And after 3 days, you stop because you treat the flu, and that's the reason the resistance can come. But TB 7 days. Ladies and gentlemen. It's a 6 months treatment for blood cells. That is called multi-drug therapy. Multi, and that for blood resistant TB is a different drug, and it's the longer treatment. Next slide, please. The short course chemotherapy. This short course is cost 6 months. Initially, for 2 months, you have 4 drugs. Rifampicin is R, H is INH, isolated hydroxide, Z is pyrazidamide, and E is pyrazidamide. The combination of the other 4 months is 4 of those drugs. So 6 months for blood sensitivity, and next, yeah. But for blood sensitive, blood resistant TB, you can't use because these drugs are resistant now. Then you have the second line drugs. Chloroquine and amino glycosides. So this is under those conditions, treatment is for 18 to 24 months. It's about just about 50% for India. So XDR TB to be even more complicated. PDR I am not going to talk about. Next slide, please. Next slide. So why it's so? Because mycobacterium is a very formidable, very difficult pathogen to deal with. Unlike many other bacteria what you have, and which can be huge. Of course, they are all become that resistant. That's it. We are talking for another day. If you call another time, I'll talk about that resistance. So mycobacterium tuberculosis likes nothing but getting ingested by macrophage. Like human mycobacteria has come from more from monkeys, and the cow mycobacteria or mycobacterium form is very similar. So this kiwi bacteria is being there long ago. There is a kiwi bacteria even for fish or the mycobacterium marina. That also has very good similarity for humans. So they change over. So this very old bacteria, what likes is to infect immune system. Macrophages are the immune system, right? That's what she explained so nicely. So, what do? They infect the macrophage. They love to do it. Make their home. Whether they make their home, from there they move other cells. They destroy the macrophage. I will quickly go through it. I am not going to case. We can do that because antivirals has a tough organism protected by vaccine lipid, complex array of polysaccharides, and an ability to counter immune and chemical attacks. The host is always putting immune attack, or it is by described as chemical attacks. Reactive to oxygen, reactive to nitrogen, loss, all those things. When you study biochemistry, you will know all those things. You don't need today, but all these points go basic level. Next. So, quick recap of what TB bacteria does. TB infection, all of us travel in public transport, we get exposed to this TB bacteria, but we don't come down with the disease, simply it can go down. Most of us carry it. Otherwise, how do you explain a young Swedish or American state flat down in India, spread to Ladakh and elsewhere, when gone back, when they turn 60, 65, they end up with psychiatric. Because when you get old, the immune system is down. Many such cases have come up. The TB reactivation, when their immune system is down. So we don't get, because all of us are healthy, I think we stay, that's why our immune system suppresses that resides like in the dormant state, persistent dormant state. And most of us have such TB bacteria in our body. But we can't detect it under that conditions. So, it infects, I quickly go through, but basically what happens is, this is one macrophage, allular macrophage, it finds a way out outlining, out competing our system and makes this mass of cells, I don't have time to explain it. This itself can be one of simply makes a mass of cells, multiplies them, and there got a granuloma. This granuloma, when it becomes an acid granuloma, as a time we are coughing, because the granuloma bursts open, and this bacteria released, it can go to infect neighboring tissue. Sometimes it gets out of the other cells. It can infect anything and everything. Okay, next slide. Yeah, just go back to the previous one. So basically, engages the host with the diverse strategies for survival and multiplication, inhibits oxidative nitrosamine stress, as I said, suppresses the apoptosis. Apoptosis is a process, it happens all the time in our cells, our body. The programmed cell death of unwanted cells or whatever it is. That kind of apoptosis process is suppressed and it makes cell killing by process called necrosis. And then it suppresses immune response, manipulates host to other host systems, manipulates host epigenetics. This is our recent study. I will not spend time on how manipulate the whole chromatin and there's a body study. Let's move on. So granulomas, you know what see before lunch or after lunch. This is how the granulomas also different granulomas. The lungs is down the solutions. So the thing is, this slide shows how just one tip of the iceberg how the free radicals are back. Once the virus gets into the bacteria, let's say, it's engulfed inside and it goes into a vacuole. In that vacuole, what you have is you have enzymes. Then in that vacuole is fused with something called lysosome, another organ. Lysosome and vacuole fusion, and then the virus is infected by proteases and nucleases by the phosphorase, lipases, whatnot. Even bacteria, though it is inside the vacuole, prevents the fusion between lysosome and the that starts supporting the whole thing. And this itself is been studied for well over 20 years. I will not spend time. Simply it doesn't get degraded inside the vacuole. That's what is. Then if it is degraded, then you will have the adaptive immunity, as you said. But that doesn't happen. These bacteria survives. As I said, the granuloma and such like. Next slide, please. So, what is it so called? It's an ancient bacteria, one of the cleverest bacteria, has strong ways to survive, and stay dormant inside the healthy individual, difficult to handle, very slow growth, unlike the Escherichia coli and many other bacteria. Perhaps your twice must multiply faster than many bacteria. This emergence of MDR and XDR, and there is a kinetic collaboration between the HIV, AIDS causing virus, and bacteria. The HIV/AIDS patients die of tuberculosis because the immune system goes down. The majority of at least 50% of the HIV patients die of tuberculosis. So there's a healthy collaboration. In fact, one of my colleagues is looking at both HIV and tuberculosis because one challenge is to find drug which can, because anti-HIV drug does not work for TB and let me what do you call it? It should be sensitive. It's not antibiotics. So finding the right kind of combination is a challenge that people are working on. And it's a global challenge. Strategies counter the subject. Now it has already come back. The XPR, MDR, XDR, PDR. So importantly, as I said, MPV becomes dormant and it hides inside the cell. And when it develops
cause multiple stresses. And in our body, defense system is a stress for the bacteria. And guys, no available those 4 drugs or whatever else I said can handle the TB bacteria. Expect this. So, 6 components for TB conflict. Pursue the direct observed therapy properly in Karnataka, in Tamil Nadu, in many states. This was done a very good job of that. Some of the other states, they don't do such a good job. But during COVID, all these healthcare workers involved in TB, government gives the free drugs actually, and pursues the therapy, and that is good. They are all for COVID. So you would have seen in the newspaper report that during COVID, TB cases went down. So, when you would address TB, HIV, all these, you can read those things, but where do I come in? Where you want, and when there are many research, enable and promote research. So we can, that's why we have done in class. Before you guys have gone and doing research, I still do research, but I still have some research activities. Right? So, right there. What we do is study the TB bacteria. We try to find the TB bacteria weakness, means an enzyme or whatever it is, then find a drug inhibitor which can inhibit essential enzyme, then kill the bacteria. So we do that in that research. I will not talk about research, but I will tell you the journal stuff next time. So, there are 2 ways of treating pathogens. Either you have a vaccine or drugs. Mainly, there are other ways for treating. Of course, all hygiene and other things are. When you have a disease, you have to have hygiene. There is an ancient vaccine. All of us got vaccinated to BC. This is a killed TB bacteria. It is not very effective in Indian population. For white-skinned Caucasians, it's more effective. So, 3 billion people infectable vaccinated with BCG, effective in early childhood activity, but not in adults. The most prominent reason antigen is missing. So that's one of the reasons. There is no direct link when all hospitals are to skip was spread. Next one. That depends where actually. So you can skip this. Natural immunity, I will not immunity now, we'll go next slide. So, vaccination strategy. Vaccination strategy can be very careful. This is the difficult one. That is like a vaccine to prevent infection. When the child is only zero. That is the first one. Then, you can also conventional protective vaccine. Then you can have booster vaccine at different times. Or you can have a therapeutic vaccine. Or facilitate chemotherapy by vaccine. Or vaccine to prevent reactivation, physical reactivation. As I said, at the old age. Different such strategies are followed for TB vaccine development. One ago, there are many cases. India was a few. India was from Professor Parmar developed he identified a new mycobacterial species called Mycobacterium sphaeres. Parmar, he considers it as a vaccine. But multiple approaches are there for vaccine. Again, this will come up with a few clinical vaccine immunology. I do not elaborate on it. This is not a time to elaborate. Next slide, please. So this is just a partial table of different vaccines. I'm not going to explain it because if I explain, you will see the vaccine. This will form the front pages of the board of it. They will say that. I don't want to do that. So I want to go for a TB research and this and that. Next slide. So, but vaccine is not necessarily a solution, according to me. I'll tell you why. Because before the new vaccine, it will take 20 years
then also success rate is already, it's not that way. Because chemotherapy is very clear. How do that? It's very heterogeneous. There are chemotherapy, so much differences in their genomes, their DNA, genetic makeup. So, as I said, BCG vaccine doesn't have some of that important antigens needed as a status or whatever. Don't worry about it. So, the drug is very important. At least we know that chemotherapy, if used properly, can handle it. And it's been quite a success actually, with all the problems. To shorten the total duration, right now 6 months for drug sensitivity. If anyone of youngsters, our time is over, I understand. And if anyone of you, youngsters who start the research, find out if EV treatment for 2 to 3 months, you will get Nobel Prize. Because it's not happened all the research. Not only people like me that work hard, not smart enough, but people who are smart enough, intelligent ones, have designed things, but it has not happened. So, there is lot of hope. Not only this, even the cancer was a too talk and other talks, there is hope. So, science is a way of life is fantastic. Telling you, I can tell you stories, but there is no time. Otherwise, I tell you a very beautiful story how science, by doing science, you have become more. You have got some money in the market. So it is pretty good. You also get to see all over the world. Ask people something. Then ask me where he hasn't gone. Go to conference. So science is a way of life. Now, coming back, short-term absolutely necessary. 6 months. We don't take one for 5 days, 7 days. So, who will take for 6 months? We have take. That's the drug sensitivity. Then emergency of blood resistance, we have already talked about it. That's a serious problem. So, why should I take it for lunch? And people get latent infection. Latent infection is a challenge even now. Anything I need to elaborate, lunchtime you ask me, I will elaborate. Not focus in vaccine development, which I mentioned already. TB vaccine, BCG, more than 85-year-old, provide some protection against severe, and there are no vaccines which give protection, but we don't know how effective it is. And even newer ones are being developed, as I said. It's like this. So, wherever there is interest to develop programs, big consortiums. This top one is by accident was 70% discovered. Johnson and Johnson has the headquarters called Janssen Pharmaceutica in Belgium, in Europe. There they are looking for compound and came up, published in this journal, and that is now all is let us believe. But drug resistant TB, where it becomes successful. Really good drug, inhibits ATP production, ATP is the energy molecule, and that has no, again, almost there or already this is developed by a European consortium called New Medicines for Kidney. This is successful. Next. Then, after the success, there's a new project, European, multi-billion-dollar project called Japan for Kidney, more medicines for kidney. So that is my first one. From India, I was the representative. India, there are hundreds of young people, but they invited me there and it's for new blood. Right now, my last student is trying that new blood what we found there, a combination with the existing drugs. You know what? We are just doing those experiments for that. And of course, if some company takes it up, I don't know. Earlier such was that patent and of course, Ukraine #1 company, we take it further. So I don't know what that, but we do know. So these are the different drug pipelines, and you know, drug anticiple by itself is a long process, but not as long as vaccines. From the time you identify a molecule, it can take anywhere from 7 years to 10 years to make success. But most of the time, they go down. That's why pharmaceutical, Indian pharmaceutical is not. Next. That's what it shows. All of our work, that's all of our right. So this is a 2024 global period drug pipeline. Many of them are slow promise, and 2-3 of them will benefit and other from a drugs. But that is next slide. So what we do? This is a slide for presentation when the prime minister came to background and this slide. So we do many things. Your patents and now you are finally, I'm not we do a combination of microbiology, biotechnology, biochemistry, cell biology, little bit immunology, also structure, biology, structurology, every possible areas of biology. And then we do our interact with people, type 1. Basically, I understand the essential components of vaccine. Expect this. And there are ideas, there are molecules, and so what we done? We tried the essential function of the bacteria. Not some means if you use an essential enzyme, if you disrupt it, if you inactivate it, then the bacteria will die. So if you test the essential, and you should play important role in the central processes, replication, transcription, such processes. No, our cell division is very important for that. And like this several slides here, that's what we find. Next slide. So what we work on? We work on many enzymes. We work on enzymes for DNA topoisomerase, DNA gyrase, and other enzymes. We work on enzymes which makes RNA polymerase. We work on enzymes which work on DNA, their transcription elongation factors, they are not just enzyme proteins, or enzyme called CORE, which is a transcription termination factor. When the RNA synthesis starts, it will promote that it has to stop, that happens, it's called to stop it is a goal. Like this, but whatever we work on is an essential component of it, like what? So, we study a biochemistry, we can try to develop it. You can also understand how the bacteria functions. That's the whole concept. So many things. One after the other, we have done published papers, and we learn from that how bacteria function. And develop some knowledge. So, I will quickly go through these days. Whatever idea is repurposing drugs. Hello, no crosstalk in the phone there. Hello. No crosstalk in the phone, please. I'm a teacher. You ask me any question, I can immediately answer. I'm a teacher. So, no interaction, no crosstalk. It's boring, you can go out. So, what I'm trying to say is these days, major pharmaceutical companies have found so many compounds. And those compounds, they put it in the shelf because they thought those were not useful. But now when they are running out of the idea, they bring back the cold store molecules and they start examining for other diseases. This repurposing drugs. Expect this. I'll just give one example. This LM4TB consortium. This consortium I was a member of. So there was a guy who did the blocking of the enzyme we work on. But now you know the structure. You solve the structure of target protein. And before these molecules sitting on the enzymes, what is work? These are antidepressant molecules, and in 1950s, in TB sanatoriums in Switzerland, they treated imipramide. 1950s, even before I was born. You were not 50 or not. But at that time, they treated for because of a different these happened, people came out. But I find this now, on ethics and rights, we find that this possible. So we have done studies and now we are trying. And so if it works, it's a case of repurposing. What is existing drug as well? It's typically used as active. That's it. So that was going on in the lab, as I said. We have done a lot of writings, published papers, patents. I don't want to discuss that today. And closing now, as promised, what I want to so hopefully it will work with the combination with the reforms in the INH, the front-end thinking drugs, this is what we are interested in that. And I'm showing that more guidance. Now, so what is the disadvantage drug profile? And another close by thought. Sterilizing vacuous activity. Sterilizing means, if you do 6 months treatment, the bacteria completely gets cleared. If a few bacteria decided after 1 month or 2 months of treatment, they come back and they become virus. So you should have sterilizing vacuous activity. And preferably oral delivery. That's the easiest part, taking the capsule. So you know what almost many things. Effective on persisters is cells which like government. Then it should have a mechanism of action which works, it is drug resistant and the extension of the sensitivity. And it should not be antagonist to existing drugs like drugs, direct absorbed therapy as I said. Or compatible with the dosing compatibility after using. Then it has to be compatible with the antiretroviral therapy. If it is used for TB and proximately acceptable, beyond 2 months, because many drugs you can't take after 1 week. Even 1 week you get irritation, nausea, whatnot. It's not the situation. And therapy ideally results within 4 or 2 months. And as I said, anyone of you challenge 2 months, you might get magic bullet, magic drugs. Let's go. Next slide. It's a big challenge. So the barriers, financial incentives. The major pharmaceutical companies are not interested in this. Because you say this is from India and other countries. So they also have a problem. I didn't elaborate. And many major countries withdrew from TB research. AstraZeneca had a big TB research in India. They broke down. Nobody has pulled out from Singapore and elsewhere now. And more of the political people are producing and producing years. So limited financial support compared to cancer, cancer or cardiovascular, diabetes or other things. Limited financial support. So these are the limitations. There's more money these days. Expect this. So this is the last slide. So TB bacteria is very similar to MPT. MPT is a main battle tank. TB is just one word. But main battle tank can be used. The TB is very similar. That has been the major problem. People continue to try as long as but hopefully we'll have a good if that works, people continue to work. Next slide, please. So I'm done. Thank you for your attention. Next slide, please. So I didn't do any work. I don't all this and talk. No, my hands are asleep. I can't do the full picture. But these are the students who are doing, and this what you see, I pretend to be doing, but they are doing the work. So I speak. So asking questions, anything, no question is called on the topic or related topics, asking questions. But during lunchtime, you can ask me questions. I am available after lunch, I think you may have a plan. Thank you. Your mic is not working. You will have to ask that.
Are there any safety Yes, absolutely. Dr. Subramanian asked the right question. Is there a code that is done? There is a lot of research on that. There are things have come up, but they are not effective. Like, I know I really work in that line, but it has not gone too far. I know that. For me, I really yes, but I don't know how successful they are. See, many times I know, and I think again, I will what's good for me, what is that? I go to when it's really effective. There are so many success stories for various things. Could be there. I need to.
Sir, I have a question.
Please.
70% of Indians are second to 70% of the Indian population are second workers.
Yes.
And there is less of communities.
Yes.
Less of communities.
What about sometimes yes.
How to overcome this? Whether we can total.
So that's just the newer curve is there. See, one time the doctors, the current all is trying to, newer technology, they were all that doctors were very good. And they knew by just doing the stethoscope, these days they will test. Diagnostics has improved. I gave a picture of this. It's not that much. That a gene expert and other technologies, but there is still a good experienced doctor when they say, but still pure, they come up with the antidote. That's good. But it's still a problem for them. Yeah.
However, immunotherapy is treating the tuberculosis.
It's actually, yeah, vaccine is very immunotherapy, right?
Adaptive CD8 cells are very well protected.
This approach is still there. That's where the vaccine is one way of doing it. Newer vaccines, targeting I think they had those kind of approaches using immunotherapy. But vaccination is immunotherapy. That's it. Right?
They are using the CD8 lymphocytes or not in the immunotherapeutic hospital.
In some few of them are taken.
The literature says CD8 lymphocytes are more effective than CD8.
That is, that the control C within CP community CD8 was more.
Are there any monoclonal studies?
Are there any?
Monoclonal antibodies.
Well, in my lab itself, we have done monoclonal antibodies for different proteins. And we showed the new way of doing there was a peptide inhibitors. And from peptide inhibitor onwards, looking and joint ECT taken with them. But instead of taking it further, they put it in a course because they money is happening. But then many different kind of monoclonals are there. But that kind of monoclonals directly as a therapy is not in the immediate vicinity. I don't see application.
Thank you, Professor, for the most insightful and engaging presentation on how it is emerging and re-emerging, and how it is evading the immune system, and with the emergence of NDR-PD. I thank you, sir, for this wonderful presentation on behalf of the organizers and on behalf of the students. Thank you very much.
I thank our resource person, Dr. V. Nagaraja, and chairperson, Dr. Sardar Hussain. So, it is now time to express our gratitude to a small token of appreciation. I request Dr. Sardar Hussain to come forward and felicitate our Team Resource Person, Dr. V. Nagaraja, Coordinator of Program. I request Srimati Meghana and to thank our chairperson Dr. Sardar to the moment of access to open of gratitude.
Yeah. We have online posters also. Yeah, we do. Yeah. Here.
Thank you, ma'am. We request Srimati Adya Soumya MD, Head and Assistant Professor, Department of Microbiology, to felicitate Srimati Meghana, ma'am. Thank you, ma'am. Once again, we extend our sincere thanks to our distinguished resource person for the invited session and to our respected chairperson and co-chairperson for guiding this session perfectly. Thank you. Ladies and gentlemen, we have a lunch break right now. We will be in the room T314 till half an hour, that is till 2:15. Following, we will be having the poster presentation. All the participants kindly assemble soon, as soon as you have your lunch. We'll start with the poster presentation. Thank you.
ಒಂದೇ ದಿವಸ Mine is at the last, and the judges are separate. So obviously they'll come at last for the judgment. Do you expect me to get ready in 5 minutes? Not even 5 minutes, 2 minutes. Before presidential
Wait
speech,
one second.
there's another.
Wait 1 second. Wait. Bro, if you are bringing, bring bro for me also.
Hey, ನಮ್ಮಿಬ್ರಿಗೂ ಒಂದು ತಗೊಂಡು ಬರ್ತೀರಾ ಪ್ಲೇಟ್ ಪ್ಲೀಸ್ ಯಾರ್ದು
Oh, this lady hasn't seen his face.
I heard it as Shobha. I didn't.
Shobha is my mother.
Shobha, I didn't.
If that lady goes with the phone,
Anu,
like connect the
Anu,
phone you keep and go, ma. It is updated, bro. I think I'm editing somebody. One ನಿಮಿಷ ಅಂಕಲ್
ಅಂಕಲ್ ಹಿರಿಯ ಅಂಕಲ್ ಎಡಿಟ್ ಮಾಡಬೇಕು ಲಾಸ್ಟ್ ಮಿನಿಟ್ ಏನೇನೋ ಹೇಳ್ತಾವ್ರೆ ಅವರು
why is the song here?
at last they have to play the song
Song ಇಲ್ಲಿದೆ ಏನು ಚೆಕ್ ಮಾಡಲ್ಲ ಬ್ರೋ ಹಂಗೆ ಎತ್ತು ಬಿಸಾಕ್ತೀರಾ ಪಿಪಿಟಿನ
ಇಟ್ಸ್ ನಾಟ್ ಮೀ
ಅವ್ರಲ್ಲಿ ಶಿವಂಟನು ಒಂದು ನಿಮಿಷ ಅಂಕಲ್ ಒಂದು 5 ನಿಮಿಷ ಕೊಡಿ ನನಗೆ ಸಿ ವ್ಯಾಲ್ಡಿಟ್ರಿ ಮರಿಯಾ
ಸಂಜನಾ ಟೀಮ್
ಕಲ್ಚರ್ ಪ್ರೋಗ್ರಾಮ್ ಇದೆಯಾ ಸೇಮ್ ಒನ್ ಸೇಮ್ ಥಿಂಗ್ both of them are there ಕಲ್ಚರ್ ಪ್ರೋಗ್ರಾಮ್ ಇಲ್ಲ ಇಲ್ಲ ಅಂಕಲ್ ವೆಲ್ಕಮ್ ಬೈ ರಾಜೇಶ್ವರಿ ಮ್ಯಾಮ್
ವೆಲ್ಕಮ್ ಬೈ ರಾಜೇಶ್ವರಿ
introduction of chief guest by Srimati Meghna and then address of Yogita Rao and then introduction of chief guest by Abhishek sir address sir Ramesh ಮಾಡ್ತಾರೆ ಇಲ್ಲಿ ಮಧ್ಯದಲ್ಲಿ ಹಾಕ್ಬೇಕಾ ಪ್ರಕಾಶ್
is coming and she's
ಬರಲ್ಲ ಅಂದ್ರಲ್ಲ keep quiet bro who cares what he looks like ನಂಗೆ ಆಗ್ತಿಲ್ಲ ಮಧ್ಯದಲ್ಲಿ nothing of what we do properly is done by this girl ಹರ್ಷಿತ್ ಅಂತೂ ಬರಲೇ ಇಲ್ಲ least concerned she is ಏನಿರೋ you know what we are ಇಲ್ಲ ಇಲ್ಲ ಅಂಕಲ್ ಪ್ಲೀಸ್ ಅಂಕಲ್ ನಾನು ಕುಡಿ ಒಂಚೂರು ಇಲ್ಲ ಯಾಕೆ ಗೊತ್ತಾ ಅಂಕಲ್ ಬೈತಾರೆ ಅಂಕಲ್ ಅವರು ಬಂದ್ಬಿಟ್ಟು ಏನ್ ಹಾಕ್ಬಿಟ್ರಿ ಅಂದ್ಬಿಟ್ರ ಏನಂದ್ರೆ ಅಂಕಲ್ ಓಡಕ್ಕೂ ಆಗಲ್ಲ I don't give you know why why does she wear in open hair all the time I don't understand it looks very shitty ಹಾಡು ಇಲ್ಲೇ ಶುರು ಆಗ್ತಾ ಇದೆಯಾ ಅಂತ presidential address
after that it's
announcement and distribution of prizes
so presidential speech concluding remarks
ಯಾರು ಕೊಡ್ತಾರೆ
ಆದ್ಯ
ಹಾ
ಫುಲ್
her over there. You
Wait,
pull
wait,
up
wait,
for,
wait, wait.
No, you pull the whole thing.
One
She won't.
ನಿಮಿಷ. She won't be presenting.
She won't be presenting this one. What prices?
What is
I'll
this?
tell you.
Concluding remarks.
She won't be doing the price distribution.
ತಡಿ ಅಮ್ಮ, ಅದನ್ನ ಬರೀ ರಿಪ್ಲೇಸ್ ಮಾಡ್ಬೋದು. ತಡಿ ಒಂದು ನಿಮಿಷ.
ಅದೆಲ್ಲ
2 seconds bro, some editing is there last minute. ಆಯ್ತು ಮುಗೀತು, ಬರ್ತೀನಿ. 2 minutes. ಹಾಂ 5 minutes please. ಹಾಂ after concluding remarks,
principal address.
presidential address and conference report ಇವರೇ ಕೊಡ್ತಿದಾರಲ್ಲ.
Conference report. This is concluding this remark.
Concluding remarks now.
She told me to do everything.
Next announcement and distribution.
Prices Rajesh.
Again. Nobody else to know about.
ಇವಾಗ ಹೇಳ್ತಾ ಇದ್ದಾರೆ.
ಇದನ್ನು ಪ್ಯಾರಲಲ್ ಆಗಿ ಎಡಿಟ್ ಆಗ್ತಾ ಇದೆಯಾ ಅಂತ. ಸರಿ, ಆಮೇಲೆ ವೋಟ್ ಆಫ್ ಥ್ಯಾಂಕ್ಸ್. ನಂದೀಗ ಪ್ರಾಬ್ಲಮ್ ಏನು ಗೊತ್ತಾ, ಮ್ಯೂಸಿಕ್ ಎಲ್ಲ ಹಾಕಿದ್ದೀರಾ ಕಣ್ರೋ. ನೀನು ನನಗೆ ಹೊಡಿಸ್ತೀಯಾ ನನಗೆ ಅನಿಸ್ತಿದೆ.
ಪ್ರೆಸೆಂಟ್ ಮಾಡಿದಾಗ ಅದು ಅಲ್ಲಿಂದ ಅಲ್ಲೇ ಇರಲಿ ಪ್ಲೇ ಮಾಡೀಗ. ಅದಾಗಲ್ಲ ಎಕ್ಸ್ಟೆಂಡ್ ಆಗಲ್ಲ, ಇಷ್ಟೇ ಅದು. ಅಲ್ಲಿ ಹೋಗಕ್ಕಾಗಲ್ಲ ನಿಜ, ಒಬ್ಬೊಬ್ಬರೇ ಕೊಟ್ಟಾಳದಲ್ಲಿ ಅದರ ಬೇರೆ ನಮ್ಮದಿಗೆ ಸೇರ್ಕೊಂಡು ಎಲ್ಲ ತಗೊಂಡ್ಬಿಡಿ.
ಫುಲ್ ಬರುತ್ತಾ ಈಗ? ಒಂದು ನಿಮಿಷ. ಇಲ್ಲೇ ನಿಂತ್ಬಿಡ್ತಾರೆ ಎಲ್ಲರೂ. You wear saree now only. Have lunch, wear saree now, and then go to principal station.
Can't be rushing everywhere. Morning and rushing. You're just calling me randomly. Yeah, ma'am. But still you must know that I've done the work.
ಕೇಳಿಸ್ಕೊ, ಫುಲ್ ಬರಬೇಕಿದ್ದು. ಕೇಳಿಸ್ಕೊ, ಒಂದೊಂದು ಪ್ರಾಬ್ಲಮ್ ಏನು ಗೊತ್ತಾ, ಇದನ್ನು ನಾವು ಒಂದು ವೇಳೆ ನೆಕ್ಸ್ಟ್ ಹೊತ್ತಿಗೆ ಡೈರೆಕ್ಟಾಗಿ ಜನಗಣಮನ ಸುಧಾರಿಸ್ಬಿಟ್ಟೆ ಕೇಳ್ಕೊಳ್ಳೋಣ ಅಂತ. ಅದು ಬಂದು ಕೊಡ್ತೀನಿ ತಡಿ. ಇಲ್ಲಿ ಡೈರೆಕ್ಟ್ ಬರ್ತದೆ ಏನು, ಫಸ್ಟ್ ಒಂದೇ ಮಾತರಂ ಇರೋದು, ಅದಕ್ಕೆ ನಾನು ಬರ್ತೀನಿ ತಡಿ.
Both we are standing over here. I'm not saying any sentences in between.
You have to say, please stand up for the national anthem.
Like for national anthem, no, I'm saying about this one.
ಅದು ಕಂಟಿನ್ಯೂಸ್ ಆಗಿ ನ್ಯಾಷನಲ್ ಅಂತಮ್ ಹೇಳಿ ಸಾಕು. ಆಮೇಲೆ ಅದನ್ನು ಪ್ಲೇ ಮಾಡ್ತಾರೆ.
ಒಂದೇ ಮಾತರಂಗೂ ನ್ಯಾಷನಲ್ ಅಂತಮ್ ಒಂದೇ ಸ್ಪೀಚ್ ಕೊಡ್ತಾರೆ, ಟುಗೆದರ್ ಆಡ್ಸ್ಬಿಡಿ. ಸರ್ ನನಗೊಂದು ಡೌಟ್ ಇತ್ತು ಕ್ಯಾನ್ವಾದಲ್ಲಿ ಸರ್ ಈ ಥರ ಒಂದೇ ಮಾತಾಡುವಂತೆ ಜನಗಣಮನ ಪ್ರೆಸೆಂಟ್ ಆಗುವಾಗ ಮಾತ್ರ ಪ್ಲೇ ಆಗ್ತಾ ಇದೆ ನಾರ್ಮಲ್ ಆಗಿ ಇಟ್ಟರೆ ಪ್ಲೇ ಆಗ್ತಿಲ್ಲ ಮತ್ತು ಡೈರೆಕ್ಟಾಗಿ ಈ ಥರ ಪರವಾಗಿಲ್ಲ ಈ ಥರ ಡೈರೆಕ್ಟಾಗಿ ಸ್ಟಾರ್ಟ್ ಆದರೆ ಒಂಚೂರು ಗ್ಯಾಪ್ ಕೊಡ್ಬೋದಾ ಸರ್ 1 ಸೆಕೆಂಡ್ ಗ್ಯಾಪು ಇಲ್ಲ ಹಾಂ ಒಂದೇ ಸೆಕೆಂಡ್ ಗ್ಯಾಪ್ ಬೇಕು ಎಂಡಿಂಗ್ ಮಾಡೋದು
ಅಷ್ಟು ಗ್ಯಾಪ್ ಬೇಕಂತ
ಇಲ್ಲ ಈ ಹಾಡಿಗೆ ಪ್ಲೇ ಮಾಡೆ
ಸ್ವಲ್ಪ ಗ್ಯಾಪ್ ಸಿಗುತ್ತೆ ಗ್ಯಾಪ್ ಸಿಗುತ್ತೆ ಇನ್ನೂ ಅಷ್ಟಾದರೆ
ಇನ್ನೊಂದು ಚೂರು ಕಮ್ಮಿ ಮಾಡು ಒಂದೇ ಸೆಕೆಂಡ್ ಕಮ್ಮಿ ಮಾಡು ಸಾಕು ಇಷ್ಟು ಸಾಕು ಕರೆಕ್ಟ್ ಇದೆ ಸರಿ ಸರ್ ಕ್ಲಿಯರ್ ಆಯ್ತು ಅದೊಂದೇ ಡೌಟ್ ಇತ್ತು
ಏರಿ ಆಗುತ್ತೆ. ಇದಕ್ಕೆ ಬೇರೆ ಥರ ಬರುತ್ತೆ ಎಚ್ ಡಿ ಎಮ್ ಐ ಕನೆಕ್ಟ್ ಮಾಡಿದಾಗ ಅದಕ್ಕೆ ಮಾಡಿದಾಗ ಒಂಥರ ಬರುತ್ತೆ.
ಅದಕ್ಕೆ ನಾವು ಆನ್ ಮಾಡಿದೀವಲ್ಲ ಹಂಗೆ ಮಾಡಿ ಬಿಟ್ಬಿಡೋಣ. ಆನ್ಲೈನ್ ಬರೋದು
ಪೋಸ್ಟರ್ ಪ್ರೆಸೆಂಟೇಷನ್ ಅಂಕಲ್ ಅಲ್ಲಿ ಕಾರಿಡಾರ್ ಅಲ್ಲಿ ಪೋಸ್ಟರ್ ಪ್ರೆಸೆಂಟೇಷನ್ ಕಾರಿಡಾರ್ ಅಲ್ಲಿ 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 ಹಾ ಅಂಕಲ್ ಹಾ ಹೌದು 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 ಅಂಕಲ್
Hello. ಹಲೋ, ಕ್ಷಮ ಎಲ್ಲಿದ್ಯಾ? ನನಗೆ ಒಂಚೂರು ಲ್ಯಾಪ್ಟಾಪ್ ಬೇಕಿತ್ತು. ಇಲ್ಲೇ ಇದೆ, ನಾನು ಒಂಚೂರು ಏನೋ ಟೈಪ್ ಮಾಡಬೇಕು. ನೀನು ಲ್ಯಾಪ್ಟಾಪ್ ಮುಂದೆನೇ ಕೂತಿದ್ದೀನಿ. ಹಾಂ ಪ್ಲೀಸ್. ಇಲ್ಲಾಂದ್ರೆ ಮೇಲ್ ಐ ಡಿ ಓಪನ್ ಇದೆಯಾ?
ಇದೆ, ಲೈಫ್ ಸೈನ್ಸ್ ಇದೆ.
ಲೈಫ್ ಸೈನ್ಸ್ ಇದೆಯಾ? ಓಕೆ.
1355 3156
I'll forget, bro.
ನಂಬರ್ ಲೈಫ್ ಸೈನ್ಸ್ ಇದೆ. ಅವಳು ಪೋಸ್ಟರ್ ಮಾಡಲಿಕ್ಕೆ ಇದು ಮಾಡ್ತಿದ್ದಾಳೆ, ಮಾಡಲ್ವಂತೆ ಅವಳು.
ಯಾರು?
ಮರಿಯಾ. ಮುಂಚೆ ಹೇಳ್ಬಾರ್ದ ಸರ್?
ಈಗ ಹೆಂಗೆ ಶೋ ಮಾಡೋದು?
ಪ್ರೆಸೆಂಟ್
ಇದೊಂದ್ಸಲ ಹಾಕ್ಲಾ? ನೆಟ್ ಬೇಡ. They're coming to our college, Purnima and all. I'll organize the talk here.
Yeah. She agreed.
They are only asking us. No virus. No, I have to talk to Shweta.
ಅವಳಿಗೆ ಮಾಡಕ್ಕೆ ಇಂಟ್ರೆಸ್ಟಿಂಗ್ ಅಂತ ಸರ್.
ಯಾರಿಗೆ?
ಅವಳನ್ನ ಅನನ್ಯ ಮತ್ತೆ ಅನುಷ್ಕೋಟಿ ಮಾಡಿದ್ದಾರೆ. ಆಯ್ತಾ? ಅವಳನ್ನ ಮಾಡಲ್ಲ ಅಂತ ಇದ್ದಾರೆ.
ಕೆಳಗೆ HP ಸ್ಕೋರ್ ಅಂತ ಏನಾದರೂ ಇದೆಯಾ ನೋಡು. ಬಿಡು. ಎಷ್ಟಿದೆ? ಅಷ್ಟು ಬೇಜಾನ. ಅಯ್ಯೋ, ಶ್ರೀದೇವಿ ಇನ್ಯಾರು? ಅವಳೆ ಹಂಗೆಲ್ಲ ಮಾಡೋದು. ಸ್ಕೋರ್ ಇಲ್ಲ ಬ್ರೋ. ಹಾಂ,
ಹಂಗೆ ರಿಫ್ಯೂಸ್ ಮಾಡಬೇಕಾಗಿತ್ತು. ಹೋಗಿ ಹೇಳಿದ್ರು, ನಾನು ಹೇಳಿದೆ ಬೇಡ ಅಂತ. ಬೇರೆ ಡಿಪಾರ್ಟ್ಮೆಂಟಿಗೆ ಕನ್ಫರ್ಮ್ ಮಾಡಿ ಹೇಳ್ತೀನಿ ಅಂತ ಕನ್ಫರ್ಮ್ ಮಾಡಲಿಲ್ಲ. ಅವರೆಲ್ಲ ರೆಡಿ ಆಗಿದ್ರು, ಮಾಡಿಬಿಟ್ರು. ಅವಳೀಗ ಮಾಡಲ್ಲ ಅಂತ ರೆಡಿಯಾಗಿ ಅಲ್ಲಿ ಕೂತ್ಕೊಂಡಿದ್ದಾಳೆ.
ನನಗೊಂದು ಇದು ಕಳಿಸ್ಬೇಕಲ್ಲ, ಹೆಂಗೆ ಕಳಿಸ್ಲಿ? ಜಡ್ಜಸ್ ಲಿಸ್ಟ್ ಅದೆಲ್ಲ ಬಿಡು. ನಾನು ಇನ್ನೊಂದ್ಸಲ ಕಳಿಸ್ಬೇಕು. ನನ್ನ ಇಮೇಲ್ ಐ ಡಿ ಕಳಿಸ್ಲಾ? ಲೈಫ್ ಸೈನ್ಸ್ ಕಳಿಸ್ತೀನಿ.
ಡಿಗ್ರಿ ಆಫೀಸ್ ಸೇಮ್ ಪಾಸ್ವರ್ಡ್ ತಾನೆ?
ಹಾಂ, ಇಲ್ಲ, ಡಿಗ್ರಿ ಆಫೀಸ್ ಸೇಮ್ ಪಾಸ್. ಡೌಟ್ ತಗೋ, ನಂದೇ ತಗೋ.
ಇರ್ಲಿಕ್ಕಿಲ್ಲ ನೋಡ್ತೀನಿ. ಪ್ಲೀಸ್ ಗಿವ್ ಯರ್ ವೈಫೈ ಮ್ಯಾಮ್.
ರೆಡ್ಮಿ 12 5G ಅಂತ ಫ್ಲೋರ್ 1 ವೈಫೈ
ಬರ್ತಾಯಿಲ್ಲ ನೋಡ್ತಾನೆ. ಫ್ಲೋರ್ 3 ಬಂದ್ ಮಾಡಿದ ಅಂತ ಇಲ್ವಲ್ಲ, ಮನೆಗೆ ಹೋದಲ್ಲ.
ಇದು ನಂದು ಆನ್ ಮಾಡ್ಕೊ. ಹಾಸ್ಪಿಟಲ್ ಹೆಂಗೆ ಗೊತ್ತಾಗುತ್ತೀಗ?
ಹಾಟ್ಸ್ಪಾಟಿಗೆ ಹೋಗಿ ಮೊಬೈಲ್ ಹಾಟ್ಸ್ಪಾಟಿಗೆ
ತಕ್ಕೊಂಬಿಡಪ್ಪ.
ಲ್ಯಾಬ್ ಬನ್ನಿ ನೋಡೋಣ. ಲ್ಯಾಬ್ ಬನ್ ಓಪನ್ ಇದೆ.
ಅಭಿಷೇಕ್ ABHS ನಂದೇ ಆನ್ ಮಾಡ್ಕೊ. AB ಕ್ಯಾಪಿಟಲ್ A ರೆಡ್ಮಿ 12 5G. ಹೌದಾ? ನೋಮಾ AB ಮಾಮೂಲಿ ಕ್ಯಾಪಿಟಲ್ A BHS ಚಿಕೆ. ನನ್ನ ನೇಮು
ಬಿ ಸ್ಮಾಲ್ದಾ?
ಹಾಂ ಇನ್ನೆಲ್ಲ ಸ್ಮಾಲ್. ಏನು ಶ್ರಮ. ಅಯ್ಯೋ ಇಮೇಲ್ ಆಪ್ಷನ್ನೇ ಇಲ್ವಲ್ಲ. ಇಲ್ಲ ಇಲ್ಲ ಸಾರಿ, ನೋ ನೆಟ್ವರ್ಕ್ ಕನೆಕ್ಷನ್. ಲೈಫ್ ಸೈನ್ಸ್ ಅಲ್ವಾ? ಹ್ಞೂ ಕಳಿಸ್ತೀನಿ ನೋಡಪ್ಪ.
When is this? This movie one is Thursday.
She's coming here. Probably Wednesday or Thursday.
ಅಯ್ಯಯ್ಯೋ,
You won't be there.
ನಾನು 19ಕ್ಕೆ ಹೊರಟೆ ಸರ್, 20ಕ್ಕೆ ಹೊರಟೆ.
ಬಂತ ನೋಡು.
ಗೋವಾಗ ಇಲ್ಲ.
ಓ ಇಲ್ಲ, ಅದನ್ನ ಬೇರೆಯಿಂದ ಹೋಗಿದೆ ಅನ್ಸುತ್ತೆ.
ನಾನು ವೆಡ್ನೆಸ್ಡೇ ಇದೆ ಸರ್. 19th ಇಲ್ಲ ಅಷ್ಟೇ. 19, 20, 21, 22, 23, 24 ಇಲ್ಲ. Okay, I'll come. You will meet her.
Yes, we have to.
Follow her on LinkedIn, sir. I'll not find her profile on LinkedIn.
ಇದಕ್ಕೆ ಮತ್ತೆ ಹೋಗ್ತಿಲ್ಲ, ಇದೇನು ಮಾಡ್ಬೇಕು? ಕ್ಯೂಡ್ ಅಂತ ಇದೆ. ವಾಟ್ ಶುಡ್ ಐ ಡು?
Send.
ಹೀಗಿದೆ. ಏಯ್
ಕ್ಯೂ ಆಗಿದೆ ಅಂದರೆ ಹೋಗ್ತಾ ಇದೆಯಾ ಅಂತ.
ಅಯ್ಯೋ ಹೆಂಗೆ ಮಾಡಲಿ ಇದನ್ನ?
You send again on license. The document is sent to me. I will forward to licenses.
Then fine. Shama, have sent me. Right. ಅತ್ಯಮ್ಮ ರಾಧಾರಣ ಇಷ್ಟ ಮ್ಯಾಮ್.
ಈಗಲ್ಲ 5 ಗಂಟೆಗೆ ಓಕೆ.
ಬೇಗ ಮುಗಿಸ್ಬೇಕು ಅಂದರೆ ಅವ್ರಿಗೆ ಹೇಳಬೇಕು, ಬೇಗ ಮಾಡಿ ಮೇಡಮ್ ಅಂತ.
ಪ್ಲೀಸ್ ಕಳಿಸಿದ ಕ್ಷಮ.
ಹಾಂ ಸರ್.
ಅಟೆಂಡ್ ಮಾಡ್ಕೊಂಡಿಲ್ಲ ನೀನು? ನಂದು ಆನ್ ಮಾಡ್ಕೋ ಜಲ್ದಿ. ರೆಡ್ಮಿ 12 ಅಲ್ಲೇ ಬಂತಲ್ಲ ಮೇಲೆ. ಎಂಜಾಯ್. ಜಲ್ದಿ ಓಪನ್ ಮಾಡಿಕೊಡು. ದೇವ್ರೆ ಕರ್ಕೊಂಬಿಡಪ್ಪ. ಡೌನ್ಲೋಡ್ ಇಟ್. ಡೌನ್ಲೋಡ್ ಮಾಡ್ತೀಯಾ? ಐ ಲವ್ PDF ಕನ್ವರ್ಟ್ ಇಟ್ ಟು ವರ್ಡ್.
ಇದೆಲ್ಲ ಆಗಲ್ಲ ಇದರಲ್ಲಿ. ಸರಿ, ಓಕೆ. ಏನಿದು ಸ್ಕೋರ್ ಅಂತಾನೆ ಹಾಕ್ಲಾ? ಸ್ಕೋರ್ ಶೀಟ್ ಅಂತ ಹಾಕ್ಲಾ?
ನಾವು ವರ್ಡ್.
ಅಯ್ಯೋ, ಇಲ್ಲೇ ಸೇವ್ ಮಾಡೋದು.
ಇಲ್ಲ ಐ ಲವ್ PDF ಹೋಗ್ಬೇಕು. ಪಕ್ಕ ಗೂಗಲ್. ಹಾಂ, ಅಲ್ಲಿಗೆ ಅದು ಓಪನ್ ಮಾಡು. PDF ಟು ವರ್ಡ್. ಇಲ್ಲ, ಕನ್ವರ್ಟ್ PDF. ಹಾಂ, get that PDF. Download it. ಶ್ರೀದೇವಿ ಹೇಳ ನೋಡು. ಅಲ್ಲಿ ನೋಡ್ತಿದ್ದಾಳೆ. ಈಗ ಬರ್ತಾಳೆ. ನೀನು ಅದು ಸಾಯೋರ್ಗೆ ಮುಂಚೆ ಬರ್ತಾಳೆ ಲ್ಯಾಪ್ಟಾಪ್. ನನಗೆ ಯಾರೂ ಕಳಿಸ್ಬೇಡ. ಡೌನ್ಲೋಡ್ ಇಟ್, ಕೀಪ್ ಇಟ್. ಕ್ಷಮ, ಇನ್ನೊಂದು ಕಳಿಸ್ತೀನಿ, ಅದು ಡೌನ್ಲೋಡ್ ಮಾಡಿ. ಕಳಿಸಿದ್ದೇನೆ. ಇದು PDF 2 ಅಂತ ಮಾಡ್ಕೊ. ಸ್ಕೋರೇ ಇರಲಿ ಬಿಡು. ಅದು PDF ಕನ್ವರ್ಟ್ ಮಾಡಿ ವರ್ಡ್ಗೆ. ಅದು ಕನ್ವರ್ಟ್ ಆಗ್ತಿದೆ ಈಗ ಮಾಡಿದ್ದು. ಡೌನ್ಲೋಡ್ ವರ್ಡ್ ಕೊಡು. ಫುಲ್ ತಿರುಗಿಸ್ಕೊಳ್ಳ. ಇದಾಯ್ತಾ ಈಗ? ಡೌನ್ಲೋಡ್ಸ್ ಅಲ್ವಾ?
ನೀನು ಚಾರ್ಜರ್ ತಗೊಂಡ್ಬಂದಿದ್ಯಾ ಲ್ಯಾಪ್ಟಾಪ್ದು? ಹಾಂ ಸಾರಿ, ಅವ್ಳು ಚಾರ್ಜರ್ ಆಗಲ್ಲ ಸರ್. ಅವ್ಳದ್ದು ಯು ಎಸ್ ಬಿ ಟೈಪು, ನಂದು ಆಕ್ಸ್. ಇದ್ರ ಟ್ರೈ ಮಾಡಿದೆ, ಆಗ್ತಿಲ್ಲ.
ಓಪನ್ ಆಗಲ್ಲ ಅದು?
ಆಗುತ್ತೆ.
ಅದು ಓಪನ್
They have exploited my system so much. It is on the verge of death. Open this first. Wow. Wow. ಯಾಕೆ ಸರ್ ಹಿಂಗೆ ಬರ್ತಿದೆ? ಹಾಂ ಬರ್ತಾ
enable editing. ಏನು ಮಾಡೋ ಗೊತ್ತಾ? ಯು ಡಿಲೀಟ್ ದಿಸ್ ಎಂಟೈರ್ ದಿಸ್. ಇಲ್ಲಿ ಪೂರ್ತಿ ಸೆಲೆಕ್ಟ್ ಮಾಡ್ಕೊಳ್ತೀವಿ. ಎಲ್ಲ ಡಿಲೀಟ್ ಮಾಡ್ಬಿಟ್ಯಾ? ಕೆಳಗಡೆ ಇನ್ನೂ ಕಂಟೆಂಟ್ ಇದೆ. ಕೆಳಗೆ ಪೂರ್ತಿ ಕೆಳಗೆ ಇದು ಹಾಗೆ ಇಟ್ಕೊಳ್ಳಿ. ಎಕ್ಸೆಪ್ಟ್ ದಿಸ್ ಪೇಜ್ ಯು ಡಿಲೀಟ್ ಎವ್ರಿಥಿಂಗ್ ಫ್ರಮ್ ಟು ಡಿಲೀಟ್. ಆಯ್ತಾ? ಇವ್ರದ್ದು ಡಿಲೀಟ್ ಮಾಡಿ ನೇಮ್. You come to pen drive. You come to HP. ಅದರಲ್ಲಿ 2526 ಏನಾದರೂ ಇದೆಯಾ ನೋಡು. ಹೊಸ ಫೋಲ್ಡರ್ ತೆಗಿ. ಟೆಕ್ನಿಕಲ್ ಸೆಷನ್ಸ್ ತೆಗಿ. ಸಿದ್ದೇಗೌಡ ಅಂತ ಇದರ ನೋಡು. ಸಿದ್ದೇಗೌಡ ಕಾಪಿ ದಿಸ್. ಒರಿಜಿನಲ್ ಸಿದ್ದೇಗೌಡ ಎಸ್ ನಾಟ್ ಎಟ್ ಕನ್ನ. ಸೇಮ್ ಇದಲ್ಲ, ಸಾಕು. ಸೇಮ್ ಥಿಂಗ್ ಯು ಕಾಪಿ ದಿಸ್ ಥಿಂಗ್ಸ್, ಕಂಟ್ರೋಲ್ ಸಿ, ಕಮ್ ಬ್ಯಾಕ್. ಇನ್ನೊಂದು ಇಲ್ಲ ಡೋಂಟ್ ಕಾಪಿ. ಇನ್ನೊಂದು ಸ್ಕೋರ್ ಶೀಟ್ ಐದು ತೆಗಿತೆ. ಒಂದು ಸ್ಕೋರ್ ಶೀಟ್ ತೆಗೆದಿದ್ವಲ್ಲ ನಾವು, ಅಲ್ಲಿಗೆ ಹೋಗೋಣ. ಸ್ಕೋರ್ ಟು. ಇದರಲ್ಲಿ ಕಮ್ ಟು ಡೆಡ್ ಎಂಡ್. ಯು ಕಾಪಿ ದಿಸ್. ಪೂರ್ತಿ ಅಲ್ಲ, ಪೂರ್ತಿ ದಿಸ್ ಒನ್ ಪೇಜ್ ಪೂರ್ತಿ. ಇಲ್ಲಿವರೆಗೆ ಕಂಟ್ರೋಲ್ ಸಿ, ಪೇಸ್ಟ್ ಇಟ್ ದೇರ್. ಹಿಯರ್ ಬಿಲೋ. ಅಲ್ಲ, ಒಂಚೂರು ಕೆಳಗೆ ಬರಲಿ, ಬೇರೆ ಪೇಜು. ಇನ್ಸರ್ಟ್. ಈ ಪೋಸ್ಟಲ್ ಪ್ರೆಸೆಂಟೇಷನ್ ಅಂತ ಅಲ್ಲಿ. ಗ್ಯಾಪ್ ಕೊಡು. ಕೆಳಗೆ ಬಾ. ಇಲ್ಲೂ ಅಷ್ಟೇ ಚೇಂಜ್ ಆಗಬೇಕು. ಜನಾರ್ದನ್ ಅಂತ ಇರೋ ಕಡೆ convert this into PDF. File. Export. PDF. ಅಲ್ಲಿ ಅದು ಇದು ಡೆಸ್ಕ್ಟಾಪ್. ಸೆಲೆಕ್ಟ್ ಡೆಸ್ಕ್ಟಾಪ್. ಕ್ರಿಯೇಟ್ ಆಗೋಯ್ತಾ? ಇಷ್ಟೇ ಬೇಕಾ?
ಇದು ಅಕ್ರೋಬ್ಯಾಟ್ ಸ್ವಲ್ಪ ಬೇರೆ ಥರ ಗೊತ್ತಿಲ್ಲ ಸರಿ ಅಂತ. ಇದೆ
ಕಂಟೆಂಟ್.
ಸೆಕೆಂಡ್ ಪೇಜ್.
ಓಕೆ, ಥರ್ಡ್ ಪೇಜ್. ಪೆನ್ ಡ್ರೈವ್ ಹಾಕೋದು. ಜಲ್ದಿ ಕೊಡೋರು ಪ್ರಿಂಟ್ ತೆಗಿಸ್ಬೇಕು. ದೇ ಆರ್ ವೇಟಿಂಗ್ ಜಡ್ಜಸ್. ಕಂಟ್ರೋಲ್ ಸಿ. ಇದಕ್ಕೂ ಬೇಕು ಪೆನ್ ಡ್ರೈವ್ಗೆ. ಈಗ ಅದು ಬೇರೆ ಅನ್ಸುತ್ತೆ ಕಣೋ. ಅದಲ್ಲ, ಅದಿರಲಿಲ್ಲ. ಕ್ಷಮ ಒಂಚೂರು ಜಾಗ ಬಿಡು ಕ್ಷಮ. ಯಾಕೋ ಇನ್ನೂ ಲೇಟ್ ಮಾಡಿಸ್ತಿದ್ಯಾ. ಇದಲ್ಲ, ಇನ್ನೊಂದು ಸ್ಕೋರ್ ಶೀಟ್ ತೆಗಿ. ಈಗ ಮಾಡಿಫೈಡ್ ನೋಡು, ಟೈಮ್ ನೋಡ್ಕೊ. ಸಾರ್ಟ್ ಕೊಡು. ಸಾರ್ಟ್ ಡೇಟ್ ಮಾಡಿಫೈಡ್. ಇದು ಕೊಡು. ಇದು ಈಗ ಎಕ್ಸ್ಪೋರ್ಟ್ ಮಾಡ್ಕೊ. ಇಲ್ಲ ನೀನು ವರ್ಡಲ್ಲೇ ಹಾಕೊಟ್ಬಿಡು ಗುರು. ಲೇಟ್ ಆಗುತ್ತೆ ಯಾಕೋ. ಹ್ಞೂ ಅದ್ರಲ್ಲಿ ಇರುತ್ತೆ, ಅದ್ರಲ್ಲಿ ತೋರಿಸು. ಪೆನ್ ಡ್ರೈವ್ USB 25, 26 today. Paste it. Okay. ಕೊಡಪ್ಪ. No worries. ಹಾಂ ಸರ್. ಖಂಡಿತ ಸರ್. ನೋ ವರೀಸ್. ಡನ್ ಸರ್. ಓಕೆ.
ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತಾ ಟೈಮಿಂಗ್ಸ್
ಹಾಕಿದ್ದೀನಿ ಆ ಲಿಂಕ್ ಒಂಚೂರು ನಂಗೆ ಸೆಂಡ್ ಮಾಡಿದ್ರೆ
ಆನ್ಲೈನ್ ಕೋರ್ಸ್ ಮುಗಿತಾ?
ಇಲ್ಲ ಈಗ ಅದಕ್ಕೆ ಲಿಂಕ್
ಟೈಮಲ್ಲಿ ಎಷ್ಟೊತ್ತಾಗುತ್ತೆ ಹಾಗಿದ್ರೆ?
ಓವರ್ ಆಗ್ಬೋದು ಮ್ಯಾಮ್. ಈಗ ಇಲ್ಲೇ ಕೂಡು ಸಾಕಕ್ಕೆ ಎಲ್ಲಿ ಹೋಗಿತ್ತು ಮುಗೀತಾ
ನಾನೇನು ಮಾತಾಡಲಿಲ್ಲ.
ಅದಕ್ಕೆ ಹೋಗಿದ್ಯಾ ಪ್ರೆಸೆಂಟೇಷನ್? ಅದೆಲ್ಲ ಆಪರ್ಚುನಿಟಿ ಸಿಗೋದು ಒಂದ್ಸಲ.
ಸರ್ ಮೇಲ್ ಮಾಡಿಸ್ತೇನೆ.
ಹಾಂ ಮಾಡಿ ಸರ್.
ವಾಟ್ಸಪ್ ಓಪನ್ ಮಾಡ್ಕೊಂಡು
ಬೇಡ ಕಳಿಸಿ ಸರ್, ಮೇಲಲ್ಲೇ ಕಳಿಸ್ಬಿಡಿ ಸರ್.
ಫೋಟೋ ಕಳಿಸ್ಬೇಕು.
ಸರ್ ಇಲ್ಲೇ ಇರಿ, ಇನ್ನೇನು ಬರ್ತಿದ್ದಾರೆ ಅವರು. ಪಿ ಜಿ ಮುಗೀತಲ್ವಾ? ಹಾಂ ಹಂಗಾರೆ ಬರ್ತಾರೆ ಸರ್ ಅವರು. ಹೌದಾ? ಪಿ ಜಿ ನೋಡಿದಾಗ ಇನ್ನೂ 2 ಇತ್ತು. ಇದೇ ಒಂದ್ಸಲ ಫೋನ್ ಮಾಡು. ಫೋನ್ ಮಾಡೋದು ಪಿ ಜಿ ಆನ್ಲೈನ್ ಎಷ್ಟಿದ್ದಾರೆ ಕೇಳು. ಪಿ ಜಿ ಒಂದು, ರಿಸರ್ಚ್ ಸ್ಕಾಲರ್ಸ್ 2 ಅನ್ಸುತ್ತೆ. ಅಕಾಡೆಮಿಷಿಯನ್ಸ್ ಒಂದು, ರಿಸರ್ಚ್ ಸ್ಕಾಲರ್ ಒಂದು. ಯಾವುದಕ್ಕೂ ಹೂ ಅಂತ ಹೇಳೋ. SHEK.S ಮಾಮೂಲಿ ಸರ್.
ಪೋಸ್ಟರ್ ಆನ್ಲೈನ್ PG 1,
ಹಾಂ
ಪೋಸ್ಟರ್ ಆನ್ಲೈನ್ 2, ರಿಸರ್ಚ್ ಸ್ಕಾಲರ್.
ರಿಸರ್ಚ್ ಸ್ಕಾಲರ್ಸ್ ಅಲ್ಲಪ್ಪಿ, ಅದು ಹೆಂಗೆ ಬರ್ಕೊಂಡಿದ್ಯಾ?
ಕಳಿಸಿದ್ದೀನಿ ಸರ್. ಜಾಯಿನ್ಗೆ ಹೋಗ್ಲಾ?
ಓಕೆ ಸರ್,
ಇಲ್ಲಿ ತಂದು ಇನ್ಸರ್ತೀನಿ.
ಓಕೆ ಜಾಯಿನ್ ಕೊಡ್ಬೇಕಷ್ಟೇ ಅಲ್ವಾ? ಯಾರ ನಂಬರು? ಅದಲ್ಲಿ ಹೇಳ್ತಾರ ಅವಳು, ಅಲ್ಲೇ ಇದೆಯಲ್ಲ ಅಲ್ಲಿಲ್ಲ.
ಮೊಬೈಲ್ ನಂಬರ್
ಓಕೆ ಬಿಡು, ಟೆನ್ಷನ್ ಕೊಡ್ಬೇಡ. ಒಂದು ನಿಮಿಷ ಇರು.
ಪೀಪಲ್ ಆರ್ ವೇಟಿಂಗ್.
ಆಲ್ರೆಡಿ ವೇಟಿಂಗಾ
ಅಂತ ಬಂದಿದೆ. ಹಾಂ ನೀವು
ಕಳಿಸ್ಬಿಟ್ಟಿದ್ದೀನಿ.
ಜಾಯಿನ್ ಆಗ್ಲಾ?
ನಂದು ಆ ಲ್ಯಾಪ್ಟಾಪ್ ಅಷ್ಟು ಆಗ್ತಿಲ್ಲ ಸರ್. ಇದರಲ್ಲಿ ಓಪನ್ ಮಾಡಿದ್ದೀನಿ ಕ್ಯಾನ್ವಾದಲ್ಲಿ. ಅದರಲ್ಲಿ ವಾಯ್ಸ್ ಪ್ಲೇ ಆಗುತ್ತಲ್ಲ ಸರ್, ಆ ಸೌಂಡ್ ಕ್ಲಿಯರ್ ಇರುತ್ತಲ್ಲ. ತುಂಬ ಸ್ಲೋ ಸರ್, ಚೆನ್ನಾಗಿರೋದು ಕೊಡೋದು ವಿಡಿಯೋ ಈ ಥರ ಹೋಗ್ತಲ್ಲ.
ಸೌಂಡ್ ಅವೈಲೇಬಲ್ PPTs ಇದಾ?
After that, we were told not to forward it to
ಅಮ್ಮೆ GTN ಅವ್ರಿ
ಯಾರಿರೋದು
ರಮ್ಯಾ ಜಿ ಟಿ
ಕಮ್ಯುನಿಟಿ ರಿಸರ್ಚ್ ಸ್ಕಾಲರ್ ಅದು ಬಿಟ್ರೆ
ಅವ್ರದ್ದೇ ಇನ್ನೊಂದು ಹೆಸರು, ಅವ್ರದ್ದೇ ಇನ್ನೊಂದು.
ಸ್ಕ್ರೀನ್ಶಾಟ್ ಮಾಡ್ತಾರೆ.
ಯಾರಿಲ್ಲ ಫೋನ್ ಮಾಡಿ. ಬ್ರೌನ್ ಚೇರ್
What is the number? What is the number?
Last yesterday, sir. Sir, 3 members. 3 members, Online. Online list ಕೊಟ್ಟಿಲ್ಲ.
ಲಿಸ್ಟ್ ಕೊಟ್ಟಿಲ್ಲ. ಆನ್ಲೈನ್ ಒಂದೇ.
ಮ್ಯಾಮ್ ಒಂದೇ ಲಾಗಿನ್ ಆಗಿಲ್ಲ. ರಿಸರ್ಚ್ ಸ್ಕಾಲರ್ಸ್ ಅಲ್ಲಿ ಇದ್ದಾರೆ ಈಗ. So we are just making a call whether he's attending or not. For attend ಮಾಡಿಲ್ಲ ಅಂದ್ರೆ it's no work.
ಇಲ್ಲ, you have given
ಈ ರಿಸರ್ಚ್ ಸ್ಕಾಲರ್
PGPT4 ಕೊಟ್ಟಿದ್ದೀರಾ, ಬಂದ್ರೆ ಹೇಳಿ.
ಹಾಂ ಸರ್.
ಓಕೆ, ಎಲ್ಲಿ ವ್ಯಾಲಿಡೇಟ್ರಿಕ್ಸ್ ಆಗೋಗುತ್ತೆ, ಇನ್ಯಾವಾಗ ನಾವು ಮಾಡೋದು ಅಸೆಸ್ಮೆಂಟ್?
ಆಯ್ತು ಸರ್, ಶೀಟ್ ಕೊಟ್ಟರೆ ಮುಗ್ದೇ ಹೋಯ್ತು ನಮ್ಗೆ.
ಒಂದು ನಿಮಿಷ ಇರು. Subramanya, directly share your screen and start your presentation. ಸರ್ ಇಲ್ಲೇ ಕೇಳುತ್ತಲ್ವಾ? ಸ್ಮಿತಾ ಮ್ಯಾಮ್
ಅಲ್ಲ, ಮಾತಾಡೋದಕ್ಕೆ ಮಾತ್ರ ಇದು ಅನ್ಮ್ಯೂಟ್ ಮಾಡ್ಕೊಂಡು ಮಾಡ್ಬೋದು.
ಓಕೆ. ಸ್ಮಿತಾ ಮ್ಯಾಮ್ ಸುಬ್ರಹ್ಮಣ್ಯ ಹಲೋ ಸುಬ್ರಹ್ಮಣ್ಯ ಹಲೋ ಸುಬ್ರಹ್ಮಣ್ಯ
ಇವಾಗ ಮಾತಾಡಿ.
ಹಲೋ ಹಲೋ ಅಲ್ಲ should respond, right? Please.
ಸರ್,
We are waiting. We are already in emergency. Please present ಮಾಡ್ಲಿಕ್ಕೆ.
ಓಕೆ.
ಸರ್ ಕೂತ್ಕೊಳ್ಳಿ ಸರ್. ಹುಡುಗನ್ ಕನೆಕ್ಟ್ ಆಗ್ತಿಲ್ಲ.
ಹಲೋ ಸುಬ್ರಹ್ಮಣ್ಯ ಹಲೋ ಹಾಂ
This is PGPT4.
ಎಸ್ ಸರ್.
Oh, good.
ಆಬ್ಸೆಂಟ್ ಅಂತ ಬರ್ಕೊಂಡ್ಬಿಟ್ಬಿಟ್ಟು ನಾನು
ಮಾಡಿ. ಬಂತಲ್ಲ.
ಸರ್ ದೊಡ್ಡದು ಮಾಡಿ.
ಒಂದು ವಾಟರ್ ಬಾಟಲ್ ಕೊಡ್ತೀರ?
ಇಲ್ಲಿ ಬರ್ತಾ ಇದೆ ಎರಡು ಕಡೆನೂ.
Hello, ma'am, is my voice audible? Hello,
Yes, yes, you are audible, Subramanya. You can directly present.
ma'am, is my voice audible, ma'am?
Yeah, yeah, your voice is audible. You can present directly. 4 minutes is the timeline. Kindly keep that.
I can start. Thank you.
Hello, hello, hello. Ma'am, sir, audio ಬರ್ತಿಲ್ಲ.
ಆಡಿಯೋ ಬರ್ತಿದೆ ನಿಂದು ಬರ್ತಿದೆ ಸ್ಟಾರ್ಟ್ ಮಾಡಿದ್ದೆ
Okay, okay.
Anything is fine, sir. Like you suggest.
ಅಲ್ಲಿ ಆಫ್ ಮಾಡ್ಬಿಡಿ, ಇಲ್ಲೇ ನೋಡ್ಕೊಂಡ್ಬಿಡ್ತೀವಿ. ಚೆನ್ನಾಗಿಲ್ಲ, ಅವನಿಗೂ ಎಕ್ಸ್ಪ್ರೆಷನ್ ಆಗ್ಬಾರ್ದು.
And good afternoon, ma'am. Is my voice audible, ma'am?
ಇದನ್ನು
Ma'am, is my voice audible, ma'am?
Yes, your voice is audible. You start.
Show you posted and you
Yeah,
start. Yeah.
yeah, ma'am. I have shared the screen, ma'am.
Yeah. We can see it, pa.
Yeah.
It's
Ma'am,
visible.
yeah.
You start.
Ma'am, the work which I have done was on in silico screening of anti-cancer peptides targeting AKT1 and BCL2. These two are the proteins which were responsible for the oral cancer causing. And uh, as you know, oral cancer is one of the major health concerns in India. About 1.4 cases are registered in India. These two proteins are responsible for the oral cancer. Their overexpression causes apoptosis and induces tumorigenesis. And
What are those proteins which you are talking about?
AKT1 and BCL2, ma'am.
Okay. When they are expressed, on what conditions they are expressed?
And generally, it is expressed. If it's overexpressed, it causes cancer.
Okay. So what is your main aim of work?
Um, uh, to do in silico screening of anti-cancer peptides, we have screened the peptides, ma'am.
What are those peptides which you have screened? Have you done? Is this work done or it's a
Ma'am,
review?
ma'am, as you can see, graph is there, ma'am.
No. I mean to say, is this work done or it's a review of literature?
It's a work done by my guide, ma'am.
Okay. Fine. Yeah, please go ahead.
Uh, AKT1 and BCL2, these proteins we have screened, ma'am. In that, we have got results of 4388 peptide. That is the PDB-ID, ma'am, of peptide, which has lower docking scores and has stronger binding interactions, and which binds forming 6 hydrogen bonds. So those are the things, and which was done using HDoc. These are free tools which were used in making this interactions. And as you can in the graph, peptide 4388 peptide, that was the most favorable peptide which we got as a result, which had lower docking score and stronger binding interactions with the proteins. For in this, three peptides were considered, and 4388 showed the stronger hydrophobic interactions, and 4405 it showed moderate stable interactions, and 4403 hydrophobic interactions. These were moderately okay with the results, but 4388 this peptide interaction was considered for further in vitro and for further analysis, ma'am. This peptide, if this method is taken, then we can minimize the work in vitro and in vivo conditions.
Okay, okay.
Thank you,
Thank you.
Subramanya. Thank you.
Thank you.
Ramya, ma'am, once again we'll be calling you. Just few minutes, ma'am. Okay, done, ma'am. Research call.
ಮುಗೀತು ಸರ್.
ಮುಗೀತು.
ಜಡ್ಜ್ಮೆಂಟ್ ಶೀಟ್ ಕೊಟ್ಬಿಡ್ತೀಯಪ್ಪ.
ಹಾಂ ಮ್ಯಾಮ್.
ಹಾಂ ಸರ್. ಇದು ಎಲ್ಲೂ ಜಾಗ ಇಲ್ಲ ಸರ್. ಥ್ಯಾಂಕ್ ಯು. ಮತ್ತೆ ಜಾಯ್ನ್ ಮಾಡ್ತಾರೆ ಸ್ಟಾರ್ಟ್ ಜಾಯ್ನ್ ಆಗ್ತಾರೆ. ಇಲ್ಲ ಪ್ರೆಸೆಂಟ್ ಆಗ್ತಾರಲ್ಲ ಪ್ರೆಸೆಂಟ್ ಮಾಡ್ತಾರೆ. ಹೋಗಿ ಬಿಹೇವಿಯರ್ ವೆಂಟ್ they come here only. Yeah, I thought you were present, so that's why I checked back. That is not audible, yeah. It is unmuted. Can't see me alla. ನಿನ್ ಟ್ಯಾಗ್ ಸಕ್ಕತ್ತಾಗಿ ಕಾಣಿಸ್ತಿದೆ. ಬನ್ನಿ ಬರ್ತಾರಲ್ಲಿ ಸುಯುಕ್ತ ಮೇಡಮ್ ಬರ್ತಾರಲ್ಲ ಇನ್ನರ್ ಆಪ್ಷನ್ಸ್. You don't have a photo, right? ಫೋಟೋ ಎಲ್ಲ ಅಂಟು ಮಾಡ್ಬೋದಾಗುತ್ತೆ. ಇಲ್ಲ ಎಲ್ಲಿದೆ?
ಇದೆಯಲ್ಲ ಇಲ್ಲಿ ಅದು ಲಾಗಿನ್ ಪಾಸ್ವರ್ಡ್ ಗೊತ್ತಾ?
ಎಲ್ಲಿ?
ಏನು? ಅದಲ್ಲ, ಇಲ್ಲಿ SDM ಇದೆಯಲ್ಲ
ಅದು ಗೊತ್ತಿಲ್ಲ. ಅದು ಅಕ್ಷಯ ಸಲ ಬರ್ಬೇಕು. It's not right when he's working
a short announcement. I would like to request all the participants and students to occupy your seats. We'll start the collaborative process in 5 minutes.
Without this finishing. I don't know. Everything is only in here. Every single thing. Whatever is it is going in live. They have a recorder, same computer, everything which is recording. I'm like, I don't exist, man. Like, both of us went down. ಇಬ್ರು ಮಾಡಬೇಕು ರಿಸರ್ಚ್ ಕಾಲೇಜ್ ಮಾಡಬೇಕು ಒಬ್ಬರಾಯ್ತು ಪಿ ಜಿ ಒಬ್ಬರಿದ್ರು ಅವ್ರದ್ದು ಮುಗೀತು ರಿಸರ್ಚ್ ಕಾಲೇಜ್ ಜಾಯ್ನ್ ಆಗ್ಯಾರ ಹೀಗೆ ಮಾಡ್ತಾರಾ ಅಲ್ಲಿ ಮಾಡಲ್ಲ ಅದು ಸರಿಯಾಗಿ ಗೊತ್ತಾಗ್ತಿಲ್ಲ ನಮ್ಮ ಕನೆಕ್ಟ್ ಆಗ್ತಿದಾರ ಮಾತಾಡ್ತಿದಾರ ಅಂತ ಸೊ ಜಡ್ಜಸ್ ಇಲ್ಲೇ ಮಾತಾಡ್ಕೊತೀವಿ ನಿನ್ನೆ ಚೆನ್ನಾಗಾಯ್ತು ಇವಾಗ ಸ್ವಲ್ಪ ಅಟೆಂಡೆನ್ಸ್ ಇದೆಯಲ್ಲ ನಮಗೆ ಸೊ ಅದಕ್ಕೆ ಅವರು ಬೇಗ ರೆಸ್ಪಾಂಡ್ ಮಾಡ್ತಿಲ್ಲ ಹಾಂ ಯು ಜಿ ಸಂಜನಾ ಅಲ್ಲ ಅನ್ನಲ್ಲ ಸಂಜನಾ along with ಶ್ರೀದೇವಿ and Manya. You just tell ಸಂಜನಾ ಎಲ್ಲಿದ್ಯಾ ಸಂಜನಾ gets published in the TV down there and it shows again and again it's saved from there. Why does your Adyaman stare like that? What is wrong with that lady? This R is new. You walk like a girl, not like a boy. Yeah, bro. Please, with your blouse, you absolutely like. Then the saree. Yeah. I'm not sure said that before. But me nadiತಾ ಇರೋದು you are walking like a boy. We do. The way you're walking, it looks very. No, I should do cat or something. Yeah. Ali, out ಬರ್ತಾರಲ್ಲ stage Gracia. She comes like that on the stage. Yeah. She just went from the stage once when she came. Where is your heels? She has a confidence of people who don't have yesterday. Yesterday they went with stage, with chappal on stage. Even I need to, it's just that makes me, feet are more than even with the heels, she is shorter. You know, they have Macbeth's recorded version on our YouTube page. We have Macbeth's recorded version on our YouTube page, and we won't have the bear. Devi. Devi. ಅವಳಿಗೆ ಗೊತ್ತಿದೆ ಅವಳಿಗೆ ಏನು ಮಾಡ್ಬೇಕು
ಕಾಲ್ ಮಾಡಿದ್ದೀನಿ ಬರ್ತಿದ್ದಾಳೆ ಅವ್ರು ಇದರಲ್ಲಿದ್ದಾಳೆ ಆನ್ಲೈನ್ ಏನೋ ಹೇಳಿದ್ರು ಸೆಮಿನಾರ್ ಒಂದು
ಆನ್ಲೈನ್ ಅಲ್ಲಿ ಮಾಡಿರ್ತಾರೆ ಕಾಮರ್ಸ್ ಮಾಡ್ತಿದ್ದೆ ಆಫ್ ಆಯ್ತಾ ಆನ್ ಮಾಡಿ
ಅವ್ರನ್ನ ಕಾಲ್ ಮಾಡ್ಬೋದು ವೇಟ್ ವೇಟ್ ಇಟ್ ಇಸ್ ನಾಟ್ ಕಮಿಂಗ್ Girls in Manchuranga. Enough and space you give me. Oh yeah, enough. Yeah.
ಮಾಡ್ತೀರಾ ಮಾಡ್ತೀವಿ
ನನಗೆ ಗೊತ್ತಿಲ್ಲ.
ಬೇಕು ಅಂದ್ರೆ ಮಾಡಬೇಕು
Maria, start. ನೀನು ಚೇರು
I request all the dignitaries to occupy their seats.
Very beautiful, very beautiful, very beautiful, very beautiful. Just don't give a fuck about one single photo. ये चाहिए डायरेक्टली जंप ऑन टू
A very good afternoon to our respected dignitaries, honorable guests, faculty members, dear participants, and my dear friends. I am Maria Anisha from Sir B.N. Brady, and I have been privileged to be your master of ceremony for the valedictory function of Forum 2026, the National Conference on Recent Research and Innovations in Life Sciences, organized by the Department of Life Sciences, MMK and SDM Mahila Mahavidyalaya, Mysore, in collaboration with SDM University, Dharwad. To begin this auspicious occasion, I cordially invite Ms. Sanjana and team to render the invocation song and seek the divine blessings for the successful culmination of this two-day national conference. Thank you, team, for that beautiful and so exciting information. I now request Srimati Raju Rajeshwari, Assistant Professor, Department of Microbiology, to kindly deliver the welcome address and warm welcome our esteemed dignitaries, speakers, and participants.
Good afternoon, all present here. Om Shri Manjunataya Namaha. With immense pleasure and a deep sense of gratitude, I extend a warm and cordial welcome to everyone gathered here for the one-day ceremony of the National Conference on Research and Innovations in Life Sciences 2020. The two-day academic journey has been a wonderful platform where the knowledge needed curiosity, research with innovation, and ideas transformed into meaningful discussions. As we come to the concluding session today, we celebrate not the end of the event, but the beginning of new scientific thoughts and collaborations. It's my proud privilege to welcome our esteemed dignitaries on the dais. First and foremost, my respectful welcome to our respected principal, Professor N. Bharati, whose constant encouragement and support made this conference a reality. I request Srimati Meghana N., Assistant Professor of Biochemistry, to welcome her florally. Thank you, ma'am. Our today's chief guest, Dr. Yogita Rao, Director, International Academy of Reproductive Science, Centers of Fertility and Ideas, whose presence inspires any minds to connect science with real-world medical applications. I welcome you, ma'am. I request our beloved principal, Professor N. Bharti, to welcome ma'am florally. Thank you, ma'am. I am equally honored to welcome our guest of honor, Dr. S.R. Ramesh, CEO and Academic Advisor, School of Life Science, Pooja Bhagat Memorial Mahajan Education Center, Mysore, whose academic vision guides students towards research excellence. I welcome you, sir. I request Srimati Atya Sami, HOD of Microbiology, to welcome sir slowly. Thank you, ma'am. I extend my hearty welcome welcome to our beloved guest of honor, Dr. Prakash Halabi, sir, Chief Scientist and Head Microbiology and Fermentation Technology, CSIR, CFTR Advisor, whose contribution to scientific research continues to motivate budding researchers and who is always eager for us for kidney health. Thank you for your support, sir. I welcome you. I request Dr. Shweta S. HOD of Biotechnology to welcome class slowly. Thank you, ma'am. I welcome our Chief Convener of this conference, Professor K. Satyamurthy, Director of Research, SDM University, Dharwad, for his gracious presence and timely guidance for our conference. We welcome you, sir. I request Mr. Abhishek, Assistant Professor of Biotechnology, to welcome sir florally. I request Sandraji to welcome sir florally. Thank you, ma'am. I extend my warm welcome to Srimati Sukulka, IPSC Coordinator, Head of the Department of Computer Science in our absence. I now take this opportunity to extend a very special welcome to the pillars behind the successful organization of this conference, our dedicated conveners and co-conveners. I warmly welcome Srimati Athiya Swamin MP, Head and Assistant Professor of Microbiology. I welcome you, ma'am. I request Srimati Meghana HOD of Biochemistry to welcome our second floor. Sorry, purely welcome her. I also extend my heartfelt welcome to Dr. Shweta, Head and Assistant Professor Department of Biotechnology for our constant support, and I welcome you, ma'am. I request Srimati Sandrashri Dear, Assistant Professor of Biochemistry, to welcome ma'am. A special welcome to all the faculty members, session chairs, resource persons, lecturers, resource scholars, and especially I extend my warm welcome to our sponsors, Rusa, Mr. Rajesh and Yogi sir. I request Sandrashri and Meghna ma'am to welcome them thoroughly. I extend my welcome to our sponsor, IARC, Mr. Deepak. I request Meghna ma'am to welcome Deepak sir thoroughly. I extend my welcome to my dear volunteers, participants, and students who actively contribute to the success of this conference through participation, discussion, and inclusive participation. And I also extend my sweet as well as warm welcome to the DK company who has provided drinks for us for 2 days. I request Sandrashri ma'am to welcome them thoroughly. Who have made this conference today a happy conference by providing their good quality of drinks to us. Everything that was so connecting that time they had to actively. Over the past 2 days, we have witnessed engaging technical sessions, innovative research presentations, and meaningful scientific exchange. On behalf of Department of Life Science, I once again extend a heartfelt welcome to one and all present here. Thank you all. Thank you, ma'am, for that gracious welcome. At this moment, I request Srimati Meghana, HOD and Assistant Professor,
Department of Biochemistry to kindly introduce our honorable chief guest, Dr. Yogita Rao, for today's valedictory function. It's my great honor and privilege to introduce our esteemed chief guest, Dr. Yogita Rao, an accomplished embryologist, consultant, fertility specialist, and cosmetic gynec surgeon. Dr. Yogita Rao holds an impressive array of qualifications, including MBBS and DGO from Day-2 University, postgraduate award in reproductive technology from University of Berlin, fellowship in reproductive medicine from Case, Germany, fellowship in cosmetic gynecology from American Society of Cosmetic Physicians, MSc in clinical embryology, and PG diploma from University of Mysore. PG diploma in medical legal system, advanced certificate course in embryology from the American Society of Reproductive Medicine, Topic biopsy certification from Denmark, and certificate course in embryology from University of Queensland. He is the director of Sankasa Fertility Private Limited, Mysore and NASA, and also serves as director of International Academy of Reproductive Science. Her leadership and expertise have significantly contributed to advancing reproductive medicine and fertility care. Dr. Yogita Rao is a respected member of State ARD Board of Surrogacy, Health and Family Welfare, Government of Karnataka, contributing to policy and governance in reproductive health care. He has served as Executive Committee member of IFS Karnataka and Secretary of Federation of Obstetric and Gynecological Society of India Infertility Committee. Her excellence has been recognized with prestigious International Association of Stem Cell and Regenerative Medicine Research and Innovation Dronacharya Award. She is an active member of several renowned professional bodies, including Indian Society of Assistant Reproductive, Association of Reproductive and Clinical Scientists, European Society of Human Reproduction and Embryology, American Society of Reproductive Medicine, Federation of Obstetric and Gynecological Society of India, and British Fertility Society, reflecting her global engagement and commitment to advancing fertility science. With her vast clinical expertise, academic excellence, international training, and leadership in reproductive science, Dr. Yogita stands as a true inspiration in the field of fertility and women's health. We are so fortunate to have such a great personality with us today. I request our beloved principal, Professor Bhartiyan, to participate, ma'am, this moment. Thank you, ma'am, for that wonderful introduction, and thank you, ma'am, for the presentation. I now request Dr. Yogita Rao to kindly address the gathering with a few words. Thank you for the kind introduction, kind elaborated introduction, I should say. I will say a few quick words so that I don't consume much of time and it's already late for the call. First of all, I would like to thank the organizers and organizers and the teaching faculty and principal and Martina and everybody for having me here today. It's a real honor to be here with you all today. And I give my respects to all the dignitaries of the IAS and the delegates and faculty, speakers, young scientists, established scientists, everyone. the title, the conference title itself is so encouraging and interesting here. It's recent research and innovation in life sciences, which is much connected to my field, which is therapy. And I see that there are so many talks which were very interesting and really related to clinical field, like generative in AI in life science. We are actually using AI in fertility now. In pathology, that is cell-related assessment, and as well as assessing the quality of oocytes and embryos before transferring them or before using them. So that is AI in life science, and then oxygen sensing in health and the oxygen levels in IVF especially is very important for the growth of embryo. That is also much related to us, and the inflammation which is related to everything in the body, whatever happens in the body, and the rest of microbiomes and immunology. Immunology is very much related to so many diseases nowadays, including the repeated implantation failures in state on microbes, where we wish embryo transfer, good quality embryo transfer to a couple in humans, and then but still in reversing negative pregnancy or patient conceives and she does not get repeated failures or repeated abortions. These are connected to immunology, in which we check NK cells with the blood, NK cells with the endometrial tissue, and human T2 ratio, which are all related to whatever you are doing since yesterday. And I see that I went through a few of the books up there, and I see that so many of the students are trying to do some small amount of research using the Vijnan novel advanced technologies that those are like spectrophotometry or some other rounds, or all those expensive microscopes or instruments are being used by these undergraduate students and postgraduate students with the help of their guides we are sitting here. And it's really interesting, and we would love to collaborate with you in case if you are doing something related to healthcare. It would be nice to share samples and come to a conclusion or come to some sort of interpretation that might help the general public. So I would suggest you all that researchers should be led along with I mean, the people who are benefited by that. That is, like in my field, it is the public healthcare. So healthcare academics, like you can collaborate with clinicians and academicians, and then have a huge number of samples, and then use these advanced technologies which are available in the Mysore University, and try to do something very nice and research. And as the last word, I would like to tell you that research and innovation is something when it reaches people and when it helps the common man to achieve something, or to have a better lifestyle. Thank you, everybody. Thank you, Madam Bhati Madam and the organizers. I wish you all the very best. Thank you.
Thank you, ma'am, for your inspiring and insightful words. Your guidance has truly added great value to this conference. Now I request Mr. Abhishek, Assistant Professor, Department of Biotechnology, to kindly introduce our guest of honor, Dr. S.R. Ramesh, for today's valedictory function.
Good afternoon, greetings to the secretary of the dais and everyone present here. It's our highest privilege to introduce our guest of honor of today's event, Dr. S.R. Ramesh sir, Chief Scientist, Department of Life Science, Puja Bhagavat Kanori Mahajana Education Center, Mysuru, and the CEO, Academic Advisor, SPRR Mahajana, Pasre College, Mysuru.
Sir has completed his MSc in Biosciences, PhD in Zoology, and works as research officer at Vector Control Research Center, Pondicherry, ICMR, and he has served as senior lecturer in Zoology and Sericulture in University of Mysuru. Sir has served as chairman of Department of Studies in Zoology, University of Mysuru. Sir has specialized in the areas such as psychogenetics and genetic fluids and has 34 years teaching experience in the PG lesson. Sir has 41 years of research experience as research fellow, research officer, and member of faculty of zoology. Sir has research experience outside India, like Institute of Genetics, Germany, and Institute for Genetics, University of Germany. And under Ramesh's guidance, 14 research scholars have completed their PhD and have received awards. And Sir has received many awards like he is the recipient of DAD Fellowship for Postdoctoral Research in Germany, and is also recipient of CTC designation of postdoctoral work, and he is visiting fellow of at Germany. Then has delivered many special and invited lecturers as resource person in Mysuru, Madikeri, Mangalore, New Delhi, Lucknow, Kerala, Coimbatore, Narsikere, and many more. Sir has published over 150 national and international research papers in his lifetime. And many more to go for in Sir. I'm happy to have this opportunity to have him in our college, sir. Thank you all. Thank you. I request our principal, Bharati Ilma, to proceed with the momentum. Thank you, ma'am. Thank you, sir.
Thank you, sir, for the introduction, and thank you, ma'am, for the facilitation. I now invite Dr. Esther Ramesh to kindly address the gathering.
Dear colleagues and young students, what to tell after 2 days of illuminating scientific lecture? I need not elaborate on what has happened on these 2 days. You have witnessed many scientists speaking on this dais and also interacting with you on various subjects. So that is the trend of science here now. And I feel our time was different when we used to go abroad and acquire knowledge and come back and then do our career here. But now India has grown up very well, and you find most of the national laboratories which are equivalent to what is found here abroad. I hope Dr. Satapati is a great example for that. And he has been the architect of MLSC at Manipur. Now he has assumed a responsible position in Harvard, and I'm sure that he will continue similar kind of research and also encouragement towards research altogether. And this is an opportunity for uh, youngsters to keep focused on their career, scientific career, but for that, initially you need to qualify yourself. You have to improve your qualifications. Right now you are in the bachelor's, And I would suggest that you complete your master's and then complete research in India and then go abroad. Not going abroad, having PhD and come back. Our labs are equally and equivalent with the equal opportunities that are prevailing here. All the national labs and outside the universities, and that you decide based on the area of your interest in science, especially life sciences. I would like to say your life has to be a scientific way of life where you have to keep up the scientific temper all the time. And if you do that, your career is almost decided that you have to be in top positions because our period is over, next period is yours. And we have retired, I am retired, and Dr. Halami is now, even he is active, and it's an opportunity for you to get in touch with Dr. Halami, who is doing wonderful work in CSR, CFTR, a product of which all of you had today, the probiotics. He is doing a lot of research in probiotics and also human health and welfare. Well, I told you there are some challenges in front of you as a graduate student. Then you have to complete master, and also you have to qualify yourself in the competitive exams of UGC NET or CSR NET exams. And of course, during the course of your study here and also in higher studies, in masters, if you qualify yourself or if you master on one or more technology, then you will have a lot of demand. But couple of technologies which are currently used in the industry, in scientific field, if you master, then your career will be assured. But there are challenges in your life, but you have to accept those challenges. And this is the time when you mark your career in the right direction. The right direction means morning itself, Dr. As a coach, that you have to stick to your study. And while studying, you have to focus on the principles and concepts. If you are thorough with the principles and concepts of whatever you are studying, you can become master. Understanding is one. Comprehension is very important. If you don't comprehend, then you are lost. If you have any difficulties with regard to comprehension of whatever concepts that are there in your curriculum and beyond curriculum, your teachers are here who are well qualified, who are actually interacting with you all the time in the classroom, outside the classroom, looking into your personal requirements, etc., etc. You can share whatever you feel with your teachers because teachers are the ones who will always extend the helping hand. And suppose something comes from the student side, we are here ready to respond to that. If you know more things than what we teach in the classroom, then also we will respond to that. We will get to know about it. In these 2 days, please take it from me, it's a sincere feeling that I had. In these 2 days, I have learned a lot with the lectures that were delivered here. Likewise, learning is eternal and keeping it updated is very important for us. If you don't update yourself with the scientific advancements, then well, you will be left behind. So try to update your knowledge every time, wherever you go, whenever you go, and whatever you read, not only no, don't read many things online. No, that will not give you a hard fact and also information. But stick to the well-known books that are prescribed. When you study this, then you will get to know many things. But your study should be focused towards understanding the concepts. Well, Couple of takeaways, take home messages are that you tell your parents or family that you will not use mobile after some time. That means don't sleep with your mobile. Dr. Satyamurthy was highlighting the impact of mobile technology or mobile signals on your health. Yeah, please take that seriously and keep away your mobiles when you go to sleep. It will not, it will disturb your nice sleep otherwise. And at the same time, as I discussed in the morning, sleep deprivation will also trigger many oncologies. So there is a risk of triggering oncologies if you if you are deprived from the sleep. If you have a sound sleep means without any dreams. If you sleep without any dreams, that is a sound sleep. And when you have a sound sleep, all your body is regenerated. Your cells are regenerated. Autophagy then new cells will be formed. Okay? So, take that, take home message from me. And remember, behind every successful student, there is a teacher also. Yeah? And at the end, I would like to inform you, whoever is in the graduation, on 24th February, Mahajalas is holding a campus placement drive. Those who are willing to take that, please take the campus placement drive. It will be arranged in the Puja Bhagat Memorial Mahajana Education Center in KRS Road. I will share the entire information with your principal and also with your faculty. Remember, this is an opportunity for not only for BSc, but also MSc, and also MBA, not MBA, BBA, whoever it is. Now the students of BBA can also participate, and at the same time, there are more license vacancies. One company is coming to recruit people for different positions, and that is in Bangalore. One position is available in Bangalore, and the salary package starts from 4 lakhs onward per year. So I will share all this information with your principal and faculty. Please take that drive. If you are not even serious, that will be an opportunity to get to know what exactly I have to prepare for the next placement positions, etc. You will have the experience of exposition to corporates at that point of time. Thank you very much. I thank this college, authorities, and the principals, and the faculty members of life sciences for giving me this opportunity for being with you for 2 days.
So that you can manage. I request our principal, Dr. Ben Bharati, to felicitate the Dr. Thank you.
Thank you, sir, for your encouraging and motivating words. We are truly grateful for your presence. Now, we move on to welcoming our guest of honor, Dr. Prakash Nani. I request Ms. Kanta Steve to kindly introduce our Dr. Prakash.
Om Shri Manjulaya Namaha. Good evening to one and all present here. It's a great privilege and honor to introduce our guest of honor, Dr. Prakash M. Halami. Dr. Prakash M. Halami is an experienced scientist and academic leader with over 3 decades of research and development experience. He currently serves as a chief scientist and
head of the Department of Microbiology and Fermentation Technology at CFTRI Mysore, and has a professor at KCSIR. Additionally, he has the additional responsibility as head of the Department of Food, Protectants and Infestation Control from 2021 to 24. Dr. Harari was also the node officer for the CFTRI, which is Biotech Research Society India, National Environmental Science Academy, Association of Food Scientists Technologists India, and the first fellow of the Microbiologists Society India. He has been developed deeply doctored DB bacteria and the soya curd, probiotic carrot nectar, and he has also organized two webinars on Azadi Ki Amrit Mahotsav and several national and international conferences on topics such as probiotics, gut microbiota, antimicrobial resistance, fumigation, and grain storage. We welcome you, sir. I request our beloved principal, Professor M. Balaji, to facilitate service. May I now invite Dr. Prakash to address the gathering with a few words.
Thank you very much for nice introduction. Very happy to be here at the graduation and the seminar national conference. So basically, I think so. He has yet to yet is not in your family. I want to hear we organize even international conference in Hyderabad and I could actually participate in two speakers for duration and one year. And of course, several AMI activities also we organized, and I was thinking actually that AMI should come forward even this time also. We are here for this today's validity function of this National Conference on Research and Innovation in Life Sciences. I am very happy to share slides with Dr. Yogi Tara, who is a well-known medical professional in our university. Then our guest of honor, Dr. A.S.R. Ramesh, with of course I am associated with him in CFTRI for IBS, Institutional Biosafety Committee, and sometimes of course with activities of Food Memorial Center. A very dynamic principal of this prestigious college, and organizing committee conveners, the HOD of microbiology, biotechnology, and biotechnology. Actually, organizing committee actually is quite big. I see ideas for their implementation. I hope yesterday I have the conference is nowadays utilized to organize an outline. I only for the certificate purpose. That's not. I'm very happy to see Subramaniam also. He's a retired professor and very active now in the Department of Interdiversity, also committee. And the two Sardars are also there, very my close friends. And I am of course seeing them also especially first time this year. And of course I am very happy that our website has reached to SDM. So Mr. Ramesh is there with his products and sales to because nowadays because of the process inequalities, and then obesity because of the junk food, then again the processed food, and then taking large amount of the medicines. We have also replaced from the medical facilities to students actually able to get all our citizens. I was talking with Dr. Kuluna also because of this body whom we had collaboration to work on the product for providing products for serious illness. And then now that every projects materializes with the funding agency. The funding agency expects something different, investigators will have their own way, but now actually I am only representing one of such funding agencies, so I cannot be able to tell what is information. With relation to this topic, the research and innovation in life science, I am sure that our speakers must have justified from last 2 days. In fact, initially when we were talking, we had actually more speaker and just I was here. And I am sure that all the participants came across this country are now here with the English knowledge. They are much more updated with the recent research in life science than what they were before, 2 days before even. I could actually see the focus of the nanotechnology, the focus from the advanced field, very innovative ideas. This is what actually is required in the research. That is why actually is blessed with the science. This is basically the science physics, silk and sandal. Science, we have 4-5 research issues. 3 universities, 4 research universities. Right. So definitely a good research environment. Such a research environment you cannot find in university. You go back. I'm sure Satyamurthy also will agree with me that Mysore has a better academic research than Harvard. And that way actually SDR, because some of the students who did PhD or PhD, yeah, they are working with me. And they actually also say, when I asked them why did you study PhD, so they said, why? They have the punctuality and seriousness. They have actually the discipline which is being maintained. And then they have the initiative leading up this program. But they actually a more emotional. So compliments to the management of the service, compliments to the SDM yes, university Harvard. So we see such a several new research development, and as Madam was also very good to the AI and not only restricted or pertain to the medical field, but in a biology, in the technology. Is being used now in our own field, in the community. Even if they are actually even looking identify the microorganisms, design the biology for the community. So it was scientifically used for the so what actually you are actually planning or designing the experiment, now you use your machine learning approach or big data analysis that you use such designing your table and next time you perform it. Dr. Hussain will agree with me that such a large number of data or the information, you will be actually simulating one day before or your experiment next day also. So such several fields in the biotechnology are classified, which is actually surpass the requirement-based technology has just to understand several phenomena in a biology, make the microorganisms impossible into possible. But we are never usually directly put because of their CM gap. Or mitigated series somewhere in the laboratory did not reach to the control. But with genome editing, there is no restriction. It is going to come. Genome editing is not only
used in the cancer in the disease diagnosis or the treatment, but even improving the any live organisms are in a more natural. So this is what I think more important thing. That in India, after science is progressing, you will see several such new innovations, competitions are coming from this country also. You will see several new breakthroughs also. And more importantly, actually, we have a very good biodiversity. You have the different climate conditions. You have the different flora and fauna. So that actually helps you to study the subject more in detail. You see several actually researchers from India, you will see just go outside any institute, university. Among 10 years, 2 or 3 years will be there. Whether they will talk with you or not, that is also a different story. But then you can actually identify a related. It is 3 or 4 states, 70-80% actually are India. Florida, Texas, and then California. 80% is. And if there would not be much restriction, I think 50% would have been Indians. And among those 50% actually, most of them are actually involved in the army. They are actually very world class research. And most of them, of course, terminal they are also doing but more better. Rather undergraduate perspective or postgraduate perspective, I would just say research product that don't carry over with the internet or YouTube information or the goals. Definitely, it is actually important for us to be cautious. Understanding the actual textbook, the textbook knowledge, and making sure that you understand the basic principle. Right? Otherwise, your concept is missed somewhere. And now it is being very carefully and seriously handled with reference to this management, with reference to this copyright, with reference to the authenticity, very carefully actually done. So your product, your work should be unique. So what you are actually developing a product of the technology has to be unique. It is pertinent to the research product. You have to have your domain idea. And such actually gives principles for the students. You have the department of identity, every month actually concept educate one time $20. Students like I issued the contract for DPT. After working system, we actually schedule for this program. You submit your concept. Whatever you submit, with reference to authenticity, with reference to its library, and it is a novel. And if it fits, must be every month actually happening. Similarly, AI is another scheme. Very such innovative ideas and actually as programs are being launched. It is definitely development 24/7. We don't know which level it will be, but definitely it is for you to help. So cherish and understand and actually enjoy what the India in 24/7 will be. Right? And it's actually a good moment and actually CSI actually has given the vision for education with reference to the AI and business, with reference to the agriculture, with reference to the frontier point, 8 areas they have actually defined. This should be actually CSI are going to participate. My organization is going to participate. With reference to the biodiversity management, with reference to sustainable development. And that way, actually, India is much more take a step forward. With reference to sustainable development, we have two problems like the environment pollution, the traffic, and then what are the definitely a big challenge is there ahead and a very bright future for most of people. Why? Because India is going to become a developed country in 24. More opportunities, higher pay scale. All these people are going to be a principal by that. research, better opportunities are waiting for you. That's what I'd like to tell you. I take this opportunity to congratulate all the organizers of campus, very organized people, the co-organizers, the dynamic principal of this college, and I also take this opportunity to thank the judges who could spare their time. Otherwise, on holidays, but they could spare some time and be here. I know organizers are going to thank them, but from our side also, because we are actually a part of Austin's organizing committee of this conference. I take this opportunity to thank all the participants, those who came all the way, registered for this conference, presented their and really for their interest. I congratulate everyone those who won the prize. Once again, thank you very much and best wishes to you all. Thank you.
Thank you, sir, for your motivating and inspiring address. We sincerely thank you for your kind presence and valuable guidance. At this moment, we would like to hear from our participants. I request a few participants to kindly share their feedback and experience about Cornell's 2026. I request participants to give their feedback.
Good afternoon, I am present here. This is Savarika, currently pursuing MSc Biochemistry from Day-2 University School of Science, Bangalore. I would thank MFK and SDM Mahila Vidyalaya for giving me this opportunity to attend this national conference for the 2 days. Before speaking, when I was pursuing my PhD at Yuvraj sir, there was two speakers from CFTI who had spoken about the probiotic drink. So I just remembered about that. So I just wanted to tell you think about that. And the whole committee, the professors and volunteers who had been with us through all these things, and whatever doubts we had, they were very patient enough to clarify all the things to us. And we had a very two judges who listened to us very patiently and guided us. So, and about the speakers also, we learned many. I learned, especially I learned many things that so that I can excel in my research things and many more new things so that I can focus more on it, work on it. So I would thank the college for giving this opportunity to me. Yeah, thank you.
Any other participants?
Very good to one good evening to all guests on the panel and lecturers. First of all, it's a live experience of 2 years of workshop like conference with the poster. Of course, it was my first experience of making a poster, but worth it actually. It was based on work on the poster that he also blackmailed me for not giving me evidence actually, but it's totally worth it. And it was a very good experience. Worth it for coming for 2 days. I had a very good basic knowledge, very good confidence, very interesting topics, but still complicated. Few things went above my head to be honest, but few things they did go into my head. So thank you for this experience. Thank you.
Thank you all for your valuable feedback. Your words motivate us to organize many more such academic events in the future. Moving on to the much awaited moment of the valedictory function, and now invite Srimati Rajarajeshwari, Assistant Assistant Professor, Department of Microbiology, to kindly announce the right winners of the oral and poster presentation. Okay. The sessions will be continued afterwards since there is an emergency. One of the winners has to leave for Hyderabad. They have been trying by 5:20. So I'll announce only the oral presenters. The rest everything after the formal function. Please excuse me for that. So the oral presentation and the UD section. The second prize goes to Miss S.S. Lakshmi, Bhavana Vivekananda College, a study on the cost-benefit analysis of bioremediation versus mechanical oil spill response. I request Principal Ma'am and Rohita Ma'am to demonstrate and give the memento to the winners. Congratulations. Thank you, ma'am. Let the surprise be as it is. Now I'll stop. It's over to you, Maria. I now invite Srimati Arya Samin, MPs, Assistant Professor and Head, Department of Microbiology, to kindly present the concluding remarks of this national conference. Thank you, Maria. So, good evening, everyone. Respected dignitaries, distinguished speakers, delegates, and my participants. As we came to the close of this national conference on recent research and innovation in medicine, organized by the Science Department of our college in association with SDM, RIS, Harvard University, and also University Career Hub, Durga University of Mumbai. So we take a moment to reflect on the enriching journey of the past 2 days. We have been privileged to gain valuable insights from eminent scholars and researchers. The enlightening keynote address by Dr. Govindarajan Padmanabhan on the topic History plus Theory of Science at IISc. At IISc. The conference brought together a diverse spectrum of expertise and ideas. We began the technical deliberation with an insightful session by Dr. Shitish K. Acharya, who highlighted the roles of transcriptomics in developing diagnostic assays. This was followed by thought-provoking talk by Mr. Chandrakumar M. on emerging relevance of generative AI which opened new perspectives on interdisciplinary integration. In the post-lunch session, we were privileged to hear Dr. Srinivas PM, who shared valuable insights
on malaria research, reminding us of the crucial link between research and public health impact. The session by Dr. Jagdisha Koya on oxygen sensing in health and disease further deepens our understanding of physiological mechanisms and their clinical relevance. Coming to day 2 of our conference, we began the day with thought-provoking perspectives on AI as a co-founder who set the tone for innovation and forward-thinking. The subsequent session depended our understanding of crucial biomedical themes. Dr. Ajay Kumar gave insight on inflammation in inflammatory bowel disease, which emphasized the importance of fundamental research in improving patient outcomes. This was followed by an engaging talk on the role of microbiome in cancer progression, which opened new avenues for research by Dr. K. Satyamurthy, Professor and Director of Research at Day-2 University. After the tea break, we continued with an excellent session on the basics of innate and adaptive immunity, which reinforced core concepts that form the backbone of life science by Dr. Payal Roy, scientist, IISc Bangalore. The final scientific session on tackling drug resistance is a philosophy was especially significant as it connected research with real-world public health challenges by Dr. V. Nagaraj, Professor, Department of Microbiology, IISc. So each session was thoughtfully guided by our dedicated chair and co-chairs. Apart from this, we had good number of oral and poster presentations. So this conference reflected the spirit of academic excellence, bringing together diverse expertise, encouraging and fostering curiosity among participants. Such gatherings remind us that learning is a continuous process and that collaboration strengthens the scientific community. As we move forward to the validatory function, we have our chief guest, Dr. Yamika Rao, who is the director of IARS and Center for Quality at IBM, and guest of honor, Dr. S.R. Ramesh, CEO and academic advisor, Department of Life Science, Management Application Center, Mysore, and Dr. Prakash Patni, Chief Scientist and Head Microbiology and Information Technology, CSIR, CSIR in Mysore. Along with them, we also have our President, Professor N. Bharati, and Chief Convener, Dr. K. Satyamurthy, who is also a collaborator of this conference, who is the Research Director at the University of Harvard, and all organizing members, or student volunteers. So, yeah, and also we have prize distribution after this. Let us carry with us not only the knowledge we gain, but also the connection we build and inspiration we spread. So, thank you all for your presence, participation, and contribution in making this day. Thank you, ma'am, for beautifully summarizing the essence of for the 2026 and for your dedicated efforts in making this conference a grand success. Now, I request our respected principal, Professor Bharati, and to deliver the presidential address. Om Shri Manjunathaya. Chief guest for today's event, Dr. Yogita Rao, and guest of honor, Dr. Ramesh sir, Dr. Satyamurthy sir, and Dr. Prakash, and Srimati Head of the Department of Microbiology, and Dr. Shweta, Head of the Department of Biotechnology, Dr. Subramanian sir, Dr. Siddhegowda sir, and coordinators of RUSA, Rajesh and Yogi. My dear colleagues, as well as delegates,
delegates, distinguished guests of the dais, my dear students. So, 2 days you have witnessed discussions on various areas of life science. So, even though I told you I am from physical science, whatever the branch of science may be, our purpose is same to improve human life and then to protect, second is protect our planet. So, life science is closely connected with day-to-day life. Medicine, which cures diseases, maybe agriculture, pharmaceutical industry, waste disposal, and biofuel. You had discussions in detail, and they explained also each and every area, and detailed discussions about the topic about AI in life science also. So it's nice. So it is always said that technology makes life comfortable, but life science makes life possible. So the research and ideas shared during this conference show that science is continuously growing, bringing positive positive changes on the earth. Sometimes negative also, but we have to find solutions for it. So let us take it positively. And many have presented research papers. So in this conference, in the not only for presenting research papers, but to develop curiosity, passion, and collaboration through this seminar. And today's discussion may become tomorrow's discovery. And in the research, many research scholars are here. There may be some setbacks. You may not get the result instantaneously. You have to repeat the experiment several times. Sometimes whole thing has to be repeated. So you should have a lot of patience for all these things. And once Thomas Alva Edison, who discovered electric bulb, he was asked, "Please tell me about your failures." He said, no such failures as such. Just I found 5,000 ways it didn't work. It took 2 years to discover this. I found 5,000 ways it didn't work. So look at this. Failure is not opposite of success, but it is part of success, because part of success is not easy. It is always filled with setbacks, doubts, confusion, but you have to overcome it with resilience and determination. That is most important. And yesterday session we had Dr. Govindarajan Padmanabhan, who inaugurated the conference, and he is, I can say, he's 80 plus years young, because his enthusiasm towards the subject has not waned. Still he retains the same passion and vigor to mold the young minds. So I thank him on behalf of our institution and on my personal behalf in his absence. And behind every successful event, there is a team that is working with patience, commitment, and dedication. Because conducting a national conference like this requires patience, teamwork, and dedication, and you have done it successfully. It is Srimati Asiya Sami, Dr. Shweta, and Srimati Rajarajeshwari, Abhishek, and Santashri, Meghana, all are there. So it is a team. They brought Prakash sir and Ramesh sir and Prakash sir, Satyamurthy sir. Our invite you all resource persons, they wrote in, all experts from different places to make this seminar a very successful one, because they guided us actually in the right direction from day one to till date. They are with us. In my chamber only we conducted 2 to 3 meetings, and wholeheartedly they were suggesting every step I was observing. So on behalf of our institution, we are very thankful and grateful to you, sir. because you people are responsible for making this conference to be a successful one. Satyamurthy sir also, our students had been to Dharwad last month and they had interaction, discussions with him and they were very inspired by your motivational speeches. And here also Ramesh sir and Prakash sir, today we are very thankful and our students are inspired by you, motivated by you to your presidential address. So thank you very much, sir. And on behalf of our institution, I thank all the resource persons who have traveled from different places and shared their knowledge and expertise with us, and they have inspired our students and they have enriched our academic atmosphere of this our institution in general and conference in particular. So I thank all the resource persons on behalf of our institution and also on my personal behalf. And our IPAC coordinator, Srimati J.S. Disputa, I appreciate her. She takes keen interest in all the activities of our college. I also thank her in her absence. And I also appreciate all the participants, especially students and research scholars who have presented papers and posters, and also Saurandika and Akshobya who have given feedback, positive feedback about this conference, and your efforts, enthusiasm reflects a bright future for science and research in our country. So as we conclude, the conference is not, it is an ending, but it is actually a beginning of innovation, new ideas, and new innovations in a new direction and new research direction. So as we conclude this, so I thank all the participants and I wish you all the best on your academic and professional journey ahead. And thank you, Ananda.
Thank you, ma'am, for your visionary words and constant support towards academic excellence and women's empowerment. Moving on, I now invite Srimati Rajarajeshwari, Assistant Professor, Department of Microbiology, to kindly announce the prize winners. The name goes, success is not hard, failure is not failure. It is the courage to continue that counts. Said by Winston Churchill. By this note, I would like to congratulate all the participants, whether you will not know, if people have had a thought of participating, that itself is a winning one. A round of applause to all the participants. The oral presentation, the first prize goes to UG category, Gautami V from St. Philomena's College, a review of sustainable agricultural and United Nations Sustainable Development Goals. Congratulations. I request Satyamurthy sir and Kapal sir to take the winners. Third prize, it is a special appreciation prize which is being given by Dr. Prakash Alami sir to encourage the UG students, one for poster from Prakash Alami sir and one from Dr. Ramesh sir, to encourage the UG students, they have initiated special applications like apart from 5 1 and 2. So in this category, Sanjana HD from SDM College, a review article on synthesis and emerging applications of biocells, biomaterials. And it is Dr. Sir and Ramesh sir to participate. And the PG section oral presentation. First place go to Sagamika from Jain University. A molecule reaction and first paper on Rasaika retrovirus Bangladesh from India. I request Lovika and Raki to participate the winners. Second place goes to Sendal Kumar KK from Jain University. Evaluation of and evaluation potential of Manjaro Bhattacharya Bangladesh. I request Raki and Lovika to participate. Research scholars and academicians. First prize goes to Arpita CS, University of Mysore, Maharashtra, Gangotri, for adopting elimination of physical using sustainable nano biocide. I request Satyamurthy sir and Ramesh sir to participate in the second place goes to Amrita KS from Davangere University. Microbial consortium based on nanopricing as a synthetic strategy for enhancing chili growth in the plate. We congratulate in our absence due to emergency, she left us yesterday. Next, PG category poster presentation. First place goes to, we are very proud to announce, who is also our alumni, Usha M. from Jain University. Education of the 90 effect of ethanol reaction system for multi-drug resistant of candida isolates. I request Prakash sir and Bharti ma'am to participate. Second place goes to Anupama and Devashri from Jain University for isolation of marine microbes with plant growth promoting properties and optimization of the growth of plant. Congratulations. I request Satyamurthy sir and Ramesh sir to participate. Research scholars and academicians, Ashita Hart from CSIRT Pathways, for synthesis and characterization of thin film of cellulose, zinc nanoparticles for seeding water treatment. I request, sorry. I request Prakash sir and Natya ma'am to felicitate. Second place goes to Pramod from GSS AHR. The polyphenol article from AutoCAD is distributed and domestic data immediately activity. I request Prakash sir and Rajan ma'am to illustrate the winner. Beauty category, poster presentation. First place goes to Kushi and Sanjana, SDM, children for volunteering processing of experiential production biology, practical purification, capitalization, and industrial applications. I request Abhimanyu sir and Yogita ma'am to felicitate. Second place, Shrita Young. Second place goes to FDR Mahajanas. The distinctness in the development of corona vaccine, the advent of conservation biology. I request Yogita ma'am and Dr. Sir to felicitate. The special appreciation prize is a category of indeed the special prize which has been initiated by Prakash sir and Ramesh sir goes to Darshaniyam and Arogya for mRNA vaccine revolutionization modern immunization from STR Maharaja's College. I request Prakash sir and Ramesh sir to felicitate. And the last category and the very important category where it has been given and it has been sponsored from Dr. Satyamurthy sir for Young Scientist Award. In the poster category, it goes to Shanuga Priya, Young Scientist Award in the category of poster for Shanuga Priya. That is accumulated and that is angiotensin to induced cardiac hypertension by immediately consuming anhydrous food. I request Satyamurthy sir and Ramesh sir, all the dignitaries on the dais to honor her. And she is also a proud alumni. In the OD category, Saujanya M from Jain University, Young Scientist Award in the category of oral presentation for biofunctional attribution of dairy derived probiotics lactic acid bacteria with anti-cancer potential. Saundanya M from Jain University. Anyone can cite from Jain University? I request Satyamurthy sir and Ramesh sir to participate. Congratulations to all the winners and the participants. Thank you. As principal, I also thank coordinators from SUSA, Rajesh and Yogi, for being with us today and supporting us in each and every aspect. Thank you. Hearty congratulations to all the winners, and I would also like to congratulate all the participants for their enthusiastic participation and excellent research presentation. Thank you, ma'am, for announcing the results. And now request Dr. Shweta, HOD and Assistant Professor, Department of Biotechnology, to kindly propose the word of thanks. Om Shri Gajnatha Namaha. Good evening, everyone. It's my proud privilege to propose the vote of thanks on this memorial. All the prize winners, please stay back. We need a photo session with you all. Soon after the vote of thanks, there will be a photo session. It's my proud privilege to propose a vote of thanks on this memorial occasion of the valedictory ceremony of National Conference on Recent Research and Innovations in Life Sciences organized by Department of Life Sciences and University of Mahila Mahavidyalaya in association with STMS, ST University, Harvard, under the aegis of at the outset, we express a deep sense of gratitude to our chief artist, Padma Vibhushan awardee, Pujya Dr. Virendra Hegde, President, STM Educational Society, Ujjain, whose visionary leadership and commitment to education continue to guide and inspire. First and foremost, I express a heartfelt gratitude to our esteemed chief guest, Dr. Yogita Rao, Medical Director and Chief Immunologist, International Academy of Reproductive Sciences, Mysore, for gracing this occasion and for delivering inspiring address that has set the perfect academic tone for the conference. Your profound insights into research and scientific advancement have truly motivated young researchers and students. Thank you. We extend our heartfelt thanks to our President of today's program, Professor Bharati M, Principal, MND and ST Mahila Mahavidyalaya, for constant encouragement, administrative support, and motivating presence, which have been instrumental in organizing this national conference. Thank you, ma'am. I'm extremely thankful to our Chief Advisor, Professor P. Satyamurthy, Director of Research at STM University, Harvard, for the listing proposed speakers for the conference and for his guidance and ordinance service. Thank you, sir. We greatly acknowledge our distinguished advisory board members, Dr. Prakash Karani, Chief Scientist and Head, Microbiology Foundation Technology, CSIR, and Dr. Ramesh, CEO, Chief Scientist, Sudha Prakash Memorial Mahala Institute, for their knowledge, guidance, and valuable service, shaping the academic framework of this conference. Thank you. We also had eminent speakers from senior institutions and industries who are happy to share their knowledge and expertise during the conference. I thank all the jury members for their excellent and transparent judgment. I kindly request the jury members to come on the stage, and I request dignitaries on the dais to present token of gratitude. Jury members, please come on the stage. Dr. Siddharth and Dr. Sudhavaran. Dr. Giri. song makes an ocean. A special note of thanks to all our sponsors and supporting organizations, industry and institutional partners, RUSA, International Academy of Reproductive Sciences, IRS, Srivadara Blood Bank, Therapeutic Innovation Hub, Hyderabad Society, Rajiv Life Sciences Private Limited, Bengaluru, GKUNI, Karnataka Fine Technos, Pooja Ananda for their generous support and encouragement towards student development and research promotion. I request the RUSA sponsors, GKUNI sponsors, and IARS sponsors to come up on the stage. I request our beloved principal to present the token of appreciation for our sponsors. We have here the RUSA sponsors, CKU and IARS.
What did he do? What did he sponsor? What is there? What is there? Wow, I heard of it.
Thank you all. My sincere thanks to every participant who has traveled from different places and joined us to make this academic gathering meaningful. We had around 90 external participants, all over from Lucknow, Hyderabad, Bangalore, Davanagere, Shivamogga, etc. We also had STM alumni faculties Dr. Vetru and Dr. Chaitanya as online participants and Srimati Pallavi as offline. We also had student alumni from STM and 90 international participants. Totally 180 participants for the conference. Thank you so much. You all made this Valentine's Day highly special, excluding without the platform. Thank you all. I thank the faculty members, teachers, scholars, teaching and non-teaching staff members, and student volunteers who dedicated efforts behind the scenes ensure flawless arrangement. All volunteers, hard work, were so elegant, so beautiful, and like a wow. Huge applause to them, please. Also extend sincere appreciation to all the conveners, Srimati Amya Sami, Srimati Meghna, Srimati Raja Rajeshwari, Mr. Abhishek, Mr. all organizing committee members, student coordinators, Ms. Khushi, Ms. Madhya, Ms. Shama, and executive members whose tireless planning and teamwork made this program smooth and successful.
The text, yeah, I brought it for the chat.
I also appreciate the technical team members, Sri Dinesh Babu and Sri Akshay, for managing the hybrid mode of the conference and enabling wider participation. Finally, I thank each and everyone present here for your valuable time, enthusiasm, and contribution in making this conference a grand success. Thank you all. Thank you, ma'am, for the heartfelt vote of thanks. May I now request all our respected dignitaries on the dais and esteemed judges, dear faculty members and participants to kindly raise from your seats. Let us come together in a moment of unity and pride as we invoke the spirit of a nation.
Vande Mataram, Vande Mataram, प्रज्योत्स्ना पुलकितयामिनी पुल्लकुसुमित रुमदलशोभिनी सुहासिनी सुमधुरभाषिणी सुखदां वरदां मातरं वन्दे मातरम् वन्दे मातरम् कोटि कोटि कण्ठ कलकल निनादकराले कोटि कोटि भुजे धृत कर करवाले अबला के नोमा एतो बले बहुबलधारिणी नमामि तारिणी रिपुदलवारिणी मातरं वन्दे मातरम् वन्दे मातरम् तुमि विद्या तुमि धर्मा तुमि हृदि तुमि मर्मा त्वं हि प्राणाः शरीरे बाहुति तुमि मा शक्ति हृदये तुमि मा भक्ति तोमराय प्रतिमा गणि मंदिरे मंदिरे वन्दे मातरम् वन्दे मातरम् त्वं हि दुर्गा दश प्रहरणधारिणी कमला कमलदला विहारिणी वाणी विद्यादायिनी नमामि त्वां नमामि कमलाम् अमलाम् अतुलां सुजलां सुफलां मातरं वन्दे मातरम् वन्दे मातरम् श्यामलां सरलां सुष्मितां भूषितां धरणीं भरणीं मातरम् वन्दे मातरम् वन्दे मातरम् वन्दे मातरम्
All the members, I request you to come on the stage backside to have a photo session. All the members, please come on the stage. You hear the first part of Tamil Nadu. All the participants, please stay.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
I request all the participants to occupy their seats. And I also request all the members to occupy the dais for conversation. All the members, please come on stage.
Please, only the prize winners come on the stage. Sir, your team members, Dr. Subramanian, Dr. Sidek and Dr. Dinesh.
Thank you, sir.
Jemput ya, jemput ke sering ya.